Welcome everyone to the Regional Science Consortium's 16th Annual Research Symposium. I'm Jeanette Schnars, the Executive Director of the RSC. We are excited to start our second day of symposium presentations. We had a full day of talks yesterday, including topics on snails, amphibians, river otters, waterfowl, bees, gardens, invasive periwinkle, invasive species, and including the invasive Japanese lilac. And a great session from our formal and non-formal educators discussing the impacts COVID has had on teaching and teaching styles this academic year. We also had a fantastic poster presentation last night, and thank you for everyone that participated on the Discord platform to make it a great success. Last evening, we also had our guest presenters, David Frew and Jerry Skripczak, talking about their new book, Accidental Paradise, referring to the natural, political, and social history of Presque Isle. Please tune in tonight for our guest speakers, Brian Gula and John Laskos, PA, DCNR, Environmental Educator Specialists, talking about the natural resources at Presque Isle State Park and how they are managed. Tomorrow we will be continuing our talks in the morning with the student awards after lunch. Please be sure to tune in at 1 o'clock to watch those awards. I'm excited to listen to today's talks. We will hear about ecotoxicology, invasive species, fisheries, including kinematics and chemosensory in fish, harmful algal blooms, and zebra mussels. In addition, this afternoon we'll have a session on the wetland restoration at Presque Isle State Park, which has been an awesome team of collaborators, and we've been working on that project for nearly a decade. And we'll continue today with a session, or we will conclude today with a session on physiology and medical studies related to the auditory senses, microbiology, and antibiotics. So I think we're in for a full day of talks, and please tune in as much as you can, as well as encouraging your students and colleagues to also tune in to these different talks. If you want to see the schedule for today or any other information, please look at our website, www.reachsciconsort.com, and click on the Symposium tab to find all the details. I would like to go ahead and introduce Sarah Magian from the Regional Science Consortium, and she'll be chairing our first session today.
Hello everyone, I'm Sarah Magian. I'm the Education and Outreach Coordinator with the Regional Science Consortium, and we're just going to get right into it today. So our first speaker is Pooja Mehta. She is presenting to us today on the selection preference in Hylela azteca, development of a behavioral assay of ecotoxicology. So Pooja, whenever you're ready, go on ahead.
Thank you. Hi, everyone. My name is Pooja Mehta, and for the past year, I have been working on selection preference in Hylela azteca to see their behavioral development assay for ecotoxicology. I worked on this project with Kyle, Dr. Nutile, Dr. Simpson, and Dr. Beatty. To start off, you may be wondering, what is ecotoxicology? Well, ecotoxicology is the study of adverse effects of pollutants on ecosystems. In this, we look at common pollutants, pesticides, industrial chemicals, and pharmaceuticals that make their way into the environment. We also try and understand the effects they have on an individual's populations and communities. Traditionally, in ecotoxicology, biologists look at the lethality, growth, and reproduction of organisms. Lethality looks at the organism being alive or dead for its endpoints. Other endpoint definitives include how big the organism grows and how much its reproduction rate is. Being able to easily identify those endpoints allows us to assess specific concentrations to levels of effect. For example, a company can run an environment and determine what concentration of a chemical impacts the organisms living in that environment. The company may determine that a concentration of a certain chemical or insecticide has a lethal concentration 50, meaning that this concentration causes 50% of the organisms to die. The level of effect that this prescribes to the particular concentration is nice because it creates a defined endpoint. This is helpful because after there's a definitive number, regular organizations can go out to other companies that are dumping greater concentrations that exceed that number, and they're able to say that they're polluting the environment. They look at experiments that have been defined before by specific endpoints so they can be easily translated to population effects. All this is fine, but what happens below the definitive concentration? This works most of the time and allows for more regulatory standards for pollutions, since they are based on the endpoints discussed earlier. Like I said, this is great and all, but sublethal endpoints like behavior of animals is often overlooked. Some sublethal endpoints include behavior and space use. One behavior in particular, avoidance of contaminated areas, would be likely to have some kind of endpoint on an individual's by increasing their exposure to predation. These sublethal endpoints are equally as important as the other ones, but they're hardly ever incorporated into the process. This is what made this research come to life, because we wanted to explore the effects that are often overlooked, the sublethal effects and behavior. When organisms are in a clean environment, they're able to just live in the water freely. However, when a chemical is added, they move to the surface to get away from the contaminated area. When they go to the surface, they face a greater risk of predation and being eaten by other organisms in the shared environment. The main objective of the research was to determine if Hyalexteca exhibit preferential selection to substrates as an estimate of this species' behavioral response to the contaminations. Going deeper, we wanted to see if they have a preference. And based on that, we wanted to then expose them to contaminants and see if they can detect the chemical and avoid it. Hyalexteca are amphipod crustaceans that feed on algae and diatoms. They are the favored prey of fish. They made a good target group because of their lifestyle and reproduction cycle. They were kept in a tank with deionized water and were fed every 3 days. The 3-part chamber was brainstormed by the team and made with the help from the Penn State Barron Engineering Department. In the chamber, one side had sand and the other side had sediment, with the middle part being empty. There was altered substrate on either side, and we monitored how much time the Hyalexteca spent on each side. The specifics were changed for selection experiment and contaminations. Our materials included beakers to allow the Hyalexteca to rest, a camera tracking their movement using ethovision, pipettes, and the 3-part chamber. When I say trial, it means a total of 3 runs with sand and sediment on certain sides. When I say run, it means one individual Hyalexteca's preference. After the trials were run, the videos were manually analyzed and recorded in a notebook. In phase 1 of the experiment, we focused on the first objective. Do the Hyalexteca exhibit a preference to sand or sediment? To find our answer, we ran many trials. In each trial, we ran 3 Hyalexteca. After acquiring and putting them in their respective beakers, the trial started. We filled one side with sand and one side with sediment. We used sand against substrate as a test to see if the Hyalexteca are even able to detect the different substrates. One Hyalexteca was dropped in the middle, and so it was able to decide where it wanted to go. The ethovision program was then started and recorded the video for 10 minutes. Once the 10 minutes were over, the first one was replaced with the second Hyalexteca, and the process was repeated. Once the 10 minutes were over, the second one was replaced with the third, and the process was repeated. This allows each one to have a 20-minute rest period between each run. Once one run was done, the chamber was dumped and the placement was physically flipped, meaning the sand and sediment were dumped and refilled on opposite sides. This process was repeated 2 more times for a total of 18 trial runs in one sitting. The videos were then manually analyzed and recorded to see what type of Hyalexteca preferred and if there was a side bias. I'm now going to show a series of figures that are going to show the Hyalexteca preference, time spent on what side, and side bias. The x-axis represents the substrate type, sand and sediment. The y-axis represents the time spent in seconds. This figure shows the average time that Hyalexteca spent in arena zones containing either substrate or sand. Points represent the mean plus or minus standard error of 10 individuals, each tested 6 times. This figure demonstrates how the Hyalexteca clearly prefer to be on the side with the sediment compared to the sand. This figure shows the reaction norm showing individual Hyalexteca time spent in an arena zone containing either sand or sediment substrate. Each set of points connected by a line represents the behavior of an individual hyela. Points at the end of each line are individual means. It looks like all but one like to spend more time on the sediment than on sand. The degree of preference for sediment as a substrate appears to vary between individuals. The positive line going from sand to sediment shows the preference of wanting to be in the sediment. This figure shows the average time that hyela spent in arena zones with sand or sediment substrates, while that substrate was either on the left or right side of the arena. Points represent the mean plus or minus standard error of 10 individuals, each tested 6 times. From this figure, there does not appear to be much of a side bias. From the trials run and the videos analyzed, there's a clear representation that the hyela prefers sediment over sand. So what do we do now? Our next step was to contaminate the sediment and do the trials over again with contaminated sediment and clean sediment. To go back to our objectives, in phase 1 of the experiment, we focused on the first one. Do the Hyalexteca exhibit a preference to sand or sediment? Now, in phase 2 of the experiment, we focused on, can the hyela detect a pollutant and avoid it? Like I mentioned before, the only difference between phase 1 and phase 2 methods was that this time the chamber was filled with sediment on one side and contaminated sediment on the other side. The contaminated sediment used was a commonly used insecticide, bifenthrin. Bifenthrin was used because it's a commonly used insecticide. It is a potent neurotoxin that's quite commonly used in urban agriculture because it's really toxic to pest invertebrates, but not toxic to humans, birds, or mammals. If you have a dog, you may recognize this insecticide in your dog's tick repellent. This is also a common ingredient in bug sprays that you may spray in your house and is commonly used in bee spray as well. To find our answer to the objective, we ran many trials. 3 days before starting these trials, we put contaminated sediment on one side of the 3-part chamber to get the full contamination effects. In each trial, we ran 4 Hyalexteca. After acquiring and putting them in the respective beakers, the run started. We filled the left side with contaminated sediment and the right side with sediment. One Hyalexteca was placed in the middle and the EthoVision program was started and recorded for 10 minutes. Once the 10 minutes were over, the first was removed and replaced by the second, and the process was repeated. Once the 10 minutes were over, the second was removed and replaced by the third, and the process was repeated. Once the 10 minutes were over, the third was removed and replaced by the fourth, and the process was repeated. The Hyalexteca were tested 3 times under each condition. This allows each one to have a 30-minute rest period between each run. Now comes the different part of the steps from phase 1. Once one run was complete, the chamber was physically flipped. There was no dumping and filling the sediment and contaminated sediment. We decided to just flip the chamber because the contaminants are hydrophobic, and it's likely they could leak into the chamber, and so we did this to avoid potentially contaminating both sides. This process was repeated 3 more times for a total of 16 trial runs in one sitting. The videos were then manually analyzed and recorded to see if the Hyalexteca were able to detect a pollutant and stray away from it. I'm now going to show a series of figures that are going to show the Hyalexteca preference and time spent on what side. This figure shows the average time that hyela spent in arena zones containing either sediment or contaminated sediment. Points represent the mean plus or minus standard error of 10 individuals, each tested 6 times. This figure demonstrates how the Hyalexteca clearly prefer to be on the side with the contaminated sediment compared to just the sediment. This figure shows the reaction norm containing individual hyela time spent in arena zones containing either contaminated sediment or sediment substrate. Each set of points connected by a line represents the behavior of an individual hyela. Points at the end of each line are individual means. The flat lines represent individuals that demonstrated no preference. In this figure, it looks like fewer individuals prefer contaminated sediment, but a good amount did show preference for the clean sediment. Based on the variance of 0.89, we feel that we need to run more trials. There could be a preference in the hyela, but we need to run more samples to prove or disprove this theory. In conclusion, the Hyalexteca preferred the sediment over the sand in phase 1 of the experiment. From these results, we tested their preference between sediment and contaminated sediment by bifenthrin. There will need to be further samples done to prove which side they preferred, due to some having no preference, some preferring the contaminated side, and some preferring the clean side. This type of experimentation is overlooked, but it does exist. It is known as fringe science, and potential application to community structure or population effects is relevant in terms of how we understand the effects of contamination and how we regulate it. We hope to get more answers soon and how we can apply ecotoxicology in the future. Thank you for your time, and I will now accept any questions.
Thank you so much, Pooja. We're now going to wait and see if any questions come in. We're just going to give them a couple of seconds for those to load through. Great presentation.
Thank you.
Trials, will we have a separate container for every trial instead of?
All right, we do have a question coming through. It says, in your future trials, will you have a separate container for every trial instead of flipping it?
So our future trials, we want to next do contaminated sediment against sand just to see how sand is affected. Chamber, we have not thought about doing it separately, but the possibilities for this experiment are pretty much endless. So we can do that if possible.
All right, and then I have a second question for you. It says, how did you decide the concentration of the insecticide?
So it was the lethal concentration 50, meaning it was 0.52 micrograms per gram organic carbon. We decided it just because it was the lethal concentration, so that's like the maximum that we are able to do.
And then I have a third question here. It says, do you plan to use different concentration levels in, I assume, your future experiments?
Yeah, like I said, the possibilities are endless. So we do plan on, after we're done doing the contaminated sediment versus sand, we do plan on differing the concentrations just to see how it's affected.
All right, thank you very much, Pooja. That's all our questions.
Thank you.
Great presentation. And now we're going to take a short break before our next presentation, which will be at 10:30. we're going to get started here with our next talk. Up next we have Ivor Knight. He will be talking to us today about environmental DNA for early detection of ballast-mediated invaders in the Great Lakes. Now, judges, this is not a student presentation, so please don't worry about judging this one. And Ivor, you can go ahead and get started whenever you're ready.
Well, thanks, everybody. I'm glad I'm not being judged. That's good. Appreciate your time, and glad to talk to you about our work, the work of myself and my colleagues on environmental DNA for early detection of ballast-mediated invaders in the Great Lakes. So, outline of the talk today, I'm going to talk about ships, going to talk about shrimp, and I'm going to talk about science. So, first, the ships. 80% of the goods transported globally are transported in ships, and that includes in the Great Lakes. And there are a variety of types of ships that are move goods around in the Great Lakes. The most common are the bulk carriers, but there are also some tankers that carry liquids around. And we don't have container ships in the Great Lakes. Those are marine ports. But one of the things that is true about ships is that they not only carry goods, when they're not carrying goods, when they're empty, they're carrying a lot of water from one port to the next. And they carry that water in the tanks that are on board the ship. So this is a bulk carrier general ballast tank plan. So the cargo hold is the most important thing where all the stuff gets put. But the water gets put in various tanks in the ship that are located around the cargo hold. So when a fully loaded ship is carrying cargo, it sits down in the water pretty low, and it does not include any ballast water that would be transported from one place to another. However, when the ship unloads its cargo, it rises up in the water, and to compensate for the lack of weight in the boat, and to increase the stability, it takes on water. It doesn't take on as much water as it would cargo, but it's still a significant amount of water. In this particular vessel, it would be about 10,000 tons of water, which it carries to the next port, where it's going to pick up cargo and then unload that water. So that's how water gets, lots of water gets transported around into the Great Lakes and around the Great Lakes. So one of the things that we're concerned about in the Great Lakes is aquatic invasive species that might be transported around the Great Lakes through ballast water. And within the Great Lakes, we are concerned about ballast water transporting aquatic invasive species from the lower lakes into the upper lakes, because water, as you know, flows down from the upper lakes to the lower lakes. And about the only way that you would get a lot of aquatic invasive species, or a lot of water that might contain aquatic invasive species moving from the lower lakes to the upper lakes, is through ballast water transport. So if the ship is unloading cargo or taking on ballast water in these lower lakes and then discharging those in the upper lakes, there's this potential to move aquatic invasive species. So we're concerned about coming up with methods that we can use to help the shipping industry manage ballast water, help managers, environmental managers around the Great Lakes be able to understand where there are potentially invasive species located in particular harbors, and can we use a monitoring tool for this. So I'm going to talk about an organism now, the shrimp. The shrimp I'm talking about is the bloody red shrimp, Hemimysis animala. It's an invasive species. It first invaded in the early 2000s. Very small organism. 2 to 3 millimeters. It is an invasive species, which is very similar to a native species. And the way you tell the difference between the invasive species and the native species is a particular distinguishing feature on the tail of this little shrimp. It's these small organisms that are most easily transported in ballast water. And one of the problems of detecting small organisms is obviously being able to capture them in harbors and in ballast water, but also it is sometimes very difficult to identify them, and it takes specialists to identify a particular invasive species, and it's quite labor intensive. The Hemimysosome anomala in the Great Lakes, this is from the Glancis invasive species monitoring project. Here's a map that I pulled up just the other day. These orange places are where this particular organism is located or has been collected and put into the database. And you can see that it's pretty abundant in the lower lakes and also into some of these terrestrial areas away from the lakes in some of the, potentially some of these other watersheds that are out here. For example, in the Finger Lakes, there's these guys are located. But there's very little, if any, in Lake Superior. And there's a concern about ballast water being transported from the lower lakes and the upper lakes and transporting this particular invasive species up to the Great Lakes. So we use this as an experimental organism for us to examine some things about the science we wanted to do. Just as an aside here, we actually, our team of undergraduate students was the first to actually document the presence of Hemimysis anomala in the Pennsylvania waters of Lake Erie, and it was right here on this on the pier. Those of you who are local will be able to recognize this. And this is Kyle DeLoe, an undergraduate student who is actually fishing for these particular critters. It's best to find them at night, but you can also find them using this technology in the daytime. So let me talk about the science of how to track and monitor and how we might utilize these, use this science for helping the shipping industry as well as managers manage aquatic invasive species that might be transported by ballast water. So the technique is environmental DNA. It's actually the science and technology of using genetic information from natural environments to identify and track organisms. The concept's pretty simple. This figure shows that you can obtain samples from a variety of environments, including aquatic environments, and then process those to extract all the DNA out of that sample, whether it be DNA that's just free floating or DNA that is associated with cells that might have been sloughed off by organisms, or very tiny organisms that you might collect in a sample of either soil or water, etc. You get it into these little vials of DNA, and then you process that in a way that you can actually look at the genetic signatures in that sample, and those genetic signatures can give you information about which species are present based upon that particular genetic signature. So the way we do this in aquatic samples, here's Noel Moore, one of our undergraduate students, collecting a water sample, which is collected just in a bottle of water. And then we filter. There's two kinds of filters you can use. You can use a high capacity filter, that's the one above, or a low capacity filter, depending on how much water you want to collect. And it's a 0.45 micron filter. That's the pore size, so you're collecting any organism or parts of organisms that are smaller than 0.45 microns on average. What you do then is use commercial technologies for extracting the DNA from those filters. So you lyse the cells that would be in there and purify out the DNA. And then we have we come up with about 100 microliter sample of purified DNA. The next thing we do is use technology called polymerase chain reaction, or PCR. that has become, PCR has become well known in the last 6 months because you've heard about it in the news as the main method that is used for detecting COVID, for diagnosing COVID. So now everybody knows what PCR is. Before COVID, I had to explain that, what that was, but now I don't have to do that. Basically, you take those environmental DNA samples and you add certain chemistries, and we use a particular target gene called the cytochrome oxidase gene. And so we have a particular target in here that amplifies the cytochrome oxidase gene, which all organisms have, but are different in each organism. And we can process many hundreds of samples at once in a machine that amplifies the DNA, and then produces a curve that tells us when this curve is going up, that means that particular target is there. And we used a specific target for the bloody red shrimp or Hemimysis animala. One thing that we do in our work too is we confirm our test by looking at what we call gel electrophoresis. So we take those amplified DNA samples and run them out on a gel. You can see that there's a positive control here that we use, and then we have a negative control. And then the rest of these show where you show these little blue blobs. These are where we would, if they match the positive control, that is definitely Hemimysis animala DNA that we have detected. So the research question that we have is, can we use this eDNA as a tool to monitor harbors and ballast water for invasive organisms that could be transported by ships? It's fairly simple to use. You can get a lot of samples, a lot of bottles of water. It's a lot easier than going out and doing plankton tows and identifying these particular microscopic organisms. So it offers the potential for widespread monitoring. There's a policy implication too. We think there is value to the region in providing new tools for effective ballast management by ships and possibly reducing the cost of mitigation by allowing species and voyage-specific ballast water management. So we've begun experiments to look at the residence time for eDNA after the death of an organism. So what eDNA doesn't do is it doesn't tell you whether the organism is alive or dead. You're extracting DNA from that sample, and you don't know whether there's live or dead organisms in there. So we have been doing some experiments to look at what is the residence time for that signal after the death of the organism. And just show you an experimental design we did last summer. We took aquarium tanks, 20 aquarium tanks, and we put 20 liters of lake water into 10 of them. And then we took 10 liters of lake water and 10 liters of deionized water and put it into another 10. And the reason for that was it has been shown in the literature that DNA signal degrades faster in water that has its normal flora in it of bacteria and other things. And so if we cut that in half by adding deionized water, we thought we might be able to see a difference between these two and confirm this idea that the more biological activity you have in the water, the faster the eDNA degrades. Another thing what we did was we took some Hemimysis animala specimens, put them in water overnight to expose the water to it, and then took the animala out of it, and then inoculated all these tanks with 200 mils of that, what we call exposed water. No organisms, but exposed water. It might have cells that sloughed off from the organism, bits and pieces of dead organisms or things like that. The final thing we did was in 2 of these sets of tanks, we put actual bodies in, not just exposed water, but also included the organisms just to see how long that signal would last. Finally, we had our control tanks with water-only controls, no exposed water, no organism bodies in there, and the 2 types of waters. So we used this technique to look at our results, and I'm showing you the same gel again. This is a time series that we have right here for one particular sample. So we took samples of water out of those tanks, very small samples, 10 mil samples out of the tanks at various time periods throughout the course of the 14-day experiment, just to see how the signal degrades. And in this particular gel, you can see that the signal does not degrade. So if you look at that negative control, the T negative right there, you'll see no signal. But then at T0, after we put the exposed water and the organisms in there, we have definitely a signal. And then as time points go on left to right, you see the signal is fairly maintained pretty strongly through what we call time point 8. So to look at all the results together, we produced this plot, and I want to spend just a little bit of time on this. The y-axis here is the number of eDNA positive tanks out of 5. Remember, we had 5 replicates. So at time 0, if you see all of these dots have 5 out of 5 tanks. So at time 0, all of the ones, all 20 tanks, no matter what treatment, no matter which of these 4 treatments had a signal. And then as the experiment went on over 14 days, you can see that there's a trend between time 0 and day 4 or 5 where the signal degrades down to 0 tanks. But then something surprising happened, and as we further carried out the experiment, we would have sporadic upticks in signal. So this indicated to us that potentially there's something happening in the water where cells might be being broken down and releasing DNA into the environment again. So one of the things that we need to do with this experiment is extend the experiment longer. It's just the initial experiment, and I just want to talk a little bit about what we're doing next, the ongoing research. We're going to conduct follow-up experiments for long duration to determine the legitimacy of this sporadic resurgence of the detected eDNA. We're going to conduct similar experiments with other aquatic invasive species of critical importance to efforts to prevent the dispersal within the Great Lakes. In other words, invasive species like the invasive water flea, Daphnia lumholtzi. We're going to do 2 or 3 of these. And in the long term, we're hoping to begin with funding from the Great Lakes Protection Fund, a Great Lakes-wide project to establish parameters for use of eDNA and determining whether these target aquatic invasive species may have recently arrived or be contained in ballast water in a live condition. So this is the whole live dead type of approach. Just want to finish up by acknowledging our team, Allegra Cangelosi, Matt Gruel, Mary Balser, Ellen Jaskowitz, Emily Dobry, Kyle DeLoe, and Noel Moore. These are people who did all the work, both in terms of field work as well as laboratory work. We're indebted to collaborators who helped us along the way. Aaron Gray, who helped us with some of the primers and probes. Jeff Elliott, who helped us with getting some specimens before we were able to collect them in Lake Erie. Megan Brown, who taught us how to collect Hemimysis anomala, and Jim Grazio, who helped us identify the bloody red shrimp that we captured in Lake Erie. Our funding comes from the Great Lakes Protection Fund and Penn State University. So thank you for your attention. I'll take questions.
Thank you so much, Ivor. That was a very, very interesting presentation. I'm going to give a couple seconds to see if any questions load through. All right, we do have a question for you. It said, could you use this method as an early alert system for future aquatic invasive species invaders?
Yes, that's certainly the intent here. As we think about monitoring, it's almost like you could use this technique kind of as a smoking gun in harbors and areas where you worry about a particular invasive species. You know what it is, but it isn't there yet. You could monitor those harbors for its presence. And the advantage of this particular technique is you can do fairly widespread monitoring. As I said, you can process a lot of samples, and so that helps with the monitoring. But yeah, that's a good point.
All right, thank you so much, Ivor. We're going to move on to our next presentation. Up next here, we have Sydney White. Sydney will be talking to us about environmental contextualization of urea toxicity in aquatic environments. Judges, Sydney is a presenter, a undergraduate student, so her last name is White, spelled the normal way. Sydney with S-Y-D-N-E-E. All right, thank you so much, Sydney. You can go ahead and share your screen.
All right, awesome. All right, I do believe it's sharing now. All right. So good morning, everyone. I am Sydney White, and I'm going to be presenting research I've done I've been a part of at Penn State Behrend alongside Kristen Honhart and Sam Newtile. We are working on contextualizing the toxicity of urea in aquatic environments. Now, before I can discuss our research, I have to provide some background information. The image to the left here is depicting a de-icer. De-icers are something that any of us from Erie should be familiar with. They tend to be made with salts, therefore they're commonly known as road salt. De-icers are applied on roadways and walkways so they keep free of slippery ice during the winter months. Then spring comes along, ice and snow become liquid runoff, and runoff makes its ways to streams, rivers, and water bodies. Well, those de-icing formulations are mixed in with that runoff, and they too make their way to the streams, rivers, and water bodies. One of the most commonly used formulation of de-icers is chloride-based de-icers. However, formulations utilizing chloride are not great. Traditional de-icing formulations, like the aforementioned chloride-based ones, are harmful. For one thing, they are harmful to structures because they cause corrosion on roadways, walkways, and buildings. Corrosion is seen as crumbling and cracks, like the picture in the left here. Although not as destructive, discoloration can occur too. Pets are also at risk. If de-icers are ingested, this can cause dogs, cats, or whatever pet you own to be poisoned. If not ingested, walking along de-iced pathways can irritate an animal's paws and skin. And on the right here is a picture of my cat Phoebe, just in case anyone needed a visual on what a pet looks like. Of course, chemicals introduced into the environment have the potential to affect the environment. De-icers are no exception here. One of the many ways de-icers do affect the environment is by altering soil chemistry and therefore vegetation too. In short, plant roots tend to be salty, and through osmotic forces, the roots uptake water. However, salt and de-icers can get trapped in soil. If soil is saltier or the same amount of salty as the roots, then the roots won't uptake any water. If plants are not intaking the necessary amounts of water, their growth will be stunted if growth happens at all. Due to problems like the ones listed, alternative de-icing formulations are available. A supposed eco-friendly alternative de-icing formulation is urea-based. Such are even used to de-ice planes. So it is used on both an industrial and commercial scale. The picture on the left here depicts Safe Paw, and as the name suggests, it is marketed as being safe for animal paws, and its label right here even advertises it as non-toxic. However, the de-icer is toxic, as Safe Paw and other urea-based de-icers as a whole undergo hydrolysis in which urea gets converted to ammonium. And this leads us to the idea that started the research I'm presenting today. Previous studies have demonstrated the toxicity of urea-based de-icers, but does environmental condition, such as bedding material like sediment or sand, or temperature, such as either warm or cold. Do those cause differences in toxicity? Focusing on temperature-based toxicity as opposed to time-based gives us the opportunity to see if spring-like temperatures, when runoff occurs, induce toxicity differences. Possibly temperatures could influence the toxicity of urea due to the rate of hydrolysis or bacterial activity. A synergism, or an increase in toxicity, may exist between urea toxicity and colder temperatures, or urea toxicity and bedding materials. To figure out if such hypotheses were accurate, that brings us to the objective of the study. And that objective, to determine the effect of environmental context on urea toxicity to Chironomus dilutus. C. dilutus was the species used for this research. They are commonly used in research, allowing for adequate information availability about their life stages to relative toxicities to particular chemicals. They are also easy to culture and have both an aquatic stage and a larval and a terrestrial adult stage. For our research, second instar midges were used. That would be the ones that look like these guys. So, how is this test species used? That brings us to the methods. Toxicity tests are an attempt to measure toxicity in a sample by analyzing the results that the exposure produces on standard test organisms. C. dilutus were those test organisms. And that's what we did. Multiple toxicity tests were completed, attempting to measure toxicity of urea given different environmental parameters. Each setup had about 30 replicates with 1 liter of moderately hard water and about 200 grams of bedding. Differing concentrations of urea were dissolved in the water, ranging from 0.05 to 2.45 grams per liter, with 5 replicates allocated for each concentration and 5 controls, each with 20 individuals. Replicates were kept in tanks with temperature-controlled water. The test lasted for 10 days. Water quality measurements were recorded, and C. dilutus were fed half a milliliter of fish food mixture per replicate. Replicates' water levels were also maintained so that evaporation did not change the concentrations of urea. On day 10, the tests were broken down. Sieving through bedding material, the number of individuals alive, retrieved, and dead were recorded. These numbers were put through SPSS, which all you need to know is that that's a statistical analysis software, to find the LC50, which is the concentration of urea that is expected to kill about 50% of C. dilutus. So this was the basic setup. What changed between runs? The first environmental manipulation was bedding material. Both sand, which is at the top here, and sediment, the bottom, were used. Possibly different bedding protects differently from the ammonium present in the water, or may change the bacterial activity. The second manipulation was temperature. 23 and 10 degrees Celsius were used, with 10 degrees being a temperature mimicking that of spring runoff. An extra consideration we had was to ensure putting C. dilutus in colder temperature did not shock them. Therefore, there was an acclimation time of 2 days where the temperature was lowered at a rate of 1.5 degrees Celsius every 2 hours until reaching 10 degrees Celsius. And that was prior to starting the 10-day tox test. To find out if these manipulations cause change, we must examine the results. I'll tell you right away, neither temperature nor bedding changed or influenced urea toxicity. And what we are looking at here is experimental condition on the x-axis with the LC50 in grams per liter of Safepaw on the y-axis. For sand at 23 degrees Celsius, the LC50 was 0.505. Sediment at 23 degrees Celsius had an LC50 of 0.201, and sand at 10 degrees Celsius had an LC50 of 0.96. The lower the number, the more toxic the experimental condition is. Therefore, this was our tested most toxic experimental condition. And if colder temperatures slow the rate of hydrolysis, we would expect a higher LC50 for the 10 degrees Celsius, and that is what we observed. However, these error bars you see demonstrate a 95% confidence interval, and that's why they may look a little weird as opposed to your standard deviation error bars. And as you can see, All of the confidence intervals overlap, indicating the found values are not different and environmental context does not play a role in urea toxicity. And yes, there's supposed to be a fourth bar for sediment at 10 degrees Celsius. But the control mortality was too high, indicating other factors beyond the urea concentrations were affecting the survival of C. dilutis. And then also, upon inspecting water quality data, temperatures ranged from 6 to 12 degrees Celsius when they were supposed to be at 10. Therefore, the data cannot be considered good. And more importantly, after this presentation, my next step in research is figuring out if the cooling system is functioning properly or if it's broken. Not that I know how to fix it, but I'll figure out if it's broken. All right, moving on, this chart is displaying the average ammonium concentrations for each test over the entire testing time. On the x-axis is time in days since the test began. And on the y-axis is ammonium concentration in milligrams per liter. The portrayed values are from the same urea concentration between each of the tests. As seen here, both sand treatments, whether 23 degrees Celsius or 10 degrees Celsius, follow a similar pattern in hydrolysis. They steadily rise over the entire length of the test. However, for sediment at 23 degrees Celsius, ammonium concentration peaks sooner and even decreases after the peak. Not to mention the ammonium concentrations are much higher too. Made us wonder, does the pattern of ammonium concentration or the peak concentration play a role in toxicity? That brings us to this chart. With experimental condition again on the x-axis, but this time ammonium LC50 is plotted on the y-axis. The average ammonium concentrations are in the darker colors on the left, so this one, and this one. And the lighter colors on the right depict peak concentrations, this, and this. Again, the error bars represent a 95% confidence interval. Despite the observed differences in patterns of ammonium concentration from the previous chart, all confidence intervals overlap again. So there's still, even when looking at ammonium LC50s, there's no significant differences in toxicity based on environmental condition. With no significant difference between environmental conditions and toxicity, it leads us to believe that biological factors drive toxicity as opposed to environmental conditions. Unless regarding temperature, maybe a wider range would induce toxicity changes where we used 10 degrees Celsius and 23 degrees Celsius, maybe we should be using different temperatures. Also, since the data is incomplete, there is a plan to try to run the sediment at 10 degrees Celsius again, just to see if it is in line with the results we already have, or if maybe that would look different. However, even with the rejection of the hypothesis thus far, there's still a message worth reiterating. Urea-based de-icing formulations are still toxic. The top image here is just a smaller copy of the first chart I showed you, and the bottom chart is one taken and cited from a previous study Sam Newtile did with another Barron student examining toxicity of different de-icers. De-icing formulations here on the x-axis, and the LC50s on the y-axis, where sodium chloride, traditional road salt, is the taller bar with an LC50 of about 6 to 7 grams per liter. And the smaller bar is urea-based, which is what our test was covering, and its LC50 is probably about 1. Again, the lower the number, the more toxic it is, as less of a chemical is needed to induce 50% lethality. Therefore, not only are urea-based de-icers toxic, but they are toxic at much lower concentrations. And the skulls are here to emphasize that point. Larger skull, more toxic. And despite this, such formulations are still being advertised as eco-friendly. This is a reminder to keep in mind that eco-friendly advertising is just that, advertising. Just because something is written on packaging does not necessarily make it accurate. Therefore, being conscientious as a consumer means understanding what is actually being sold, whether it's de-icers or other products, so you know what you're actually buying. And that concludes my presentation on the research involving urea toxicity in environmental context that I've been lucky enough to be a part of at Penn State Barron. And right now, I suppose I have time to answer any questions, and thank you all so much for listening.
Thank you so much, Sydney. That was a wonderful presentation. Thank you for showing us your cat. She was very, very cute. I'm going to give a couple seconds to see. Oh, I do have a question in here for you.
Absolutely.
From a fellow pet owner. Are there any de-icers that you think are truly pet safe?
I mean, from the research I've read previous to our study versus other ones, it doesn't really seem like it. But the point being that it's kind of not so much finding a safe de-icer across the board, but finding out which one is less bad. So I guess I'll just keep Phoebe and my other pets indoors and make sure that where I'm applying de-icers, or not, at least on my scale, they're not harming, my animals.
All right, and then I'm gonna see if any more come through, so just one moment. All right, that looks like it's all good on questions for you. Thank you so much for a great presentation. And we are gonna take a short break until our next presentation at 11:10. Thank you, everyone. Alrighty, we're going to get started with our next talk here. We have Grace Galinato. Sorry if I mispronounced that. She'll be talking to us about developing artificial manganese porphyrin proteins encased in a silica sol gel for sulfoxidation reactions. That is a tongue twister. All right, Grace, we will have you go ahead. And judges, this is a speed talk, so remember, please, that you do not have to judge this one. All right, Grace, whenever you're ready.
All right, thank you, Amber, for the introduction. The title is a mouthful, but I will try to break it down for clarity. So to start, I want to talk about sulfur, because this is the main element of interest in our work. Sulfur is a natural component in crude oil that is present in gasoline and diesel. It impairs effectiveness of emission control systems, and it contributes to air pollution. So reducing or eliminating sulfur content in crude oil is important. So how do we remove sulfur? Through a process called oxidative desulfurization. This method, or ODS, means that sulfur gains oxygen atoms, making sulfur more easily removed in crude oil on the basis of polarity. So unlike another method called hydrodesulfurization, ODS can be employed in moderate reaction conditions. It has high selectivity. It does not require hydrogen, which can be explosive, and it is efficient. So how does the ODS reaction work in general? So if we consider this substrate, so dibenzothiophene is a model for sulfur-containing compounds in crude oil. What happens is dibenzothiophene gains those oxygen atoms from forming sulfone. This is polar, meaning it dissolves in water, and this does not, so you can easily separate it from crude oil. And this can only be achieved in the presence of an oxidant, so the source of your oxygen, and a catalyst. A few oxidant-catalyst pairs are shown and listed over here. For our lab, we intend to use hydrogen sulfide as a catalyst as the oxidant, and catalysts derived from nature called hemoproteins, some of which are listed over here, including hemoglobin and myoglobin. Now, these proteins contain a molecule referred to as a heme or a porphyrin. So a porphyrin is composed of a macrocycle containing a metal center surrounded by these organic groups called perols. Now, it's been shown that hemoglobin and myoglobin were able to catalyze the ODS reaction, so we're not going to use them. However, we will utilize their structure as a model to make more efficient ones. So interestingly, a porphyrin that has a manganese metal in it instead of an iron, they're shown to be more efficient for catalyzing substrate oxidation, so that dibenzothiophene I showed you earlier, they do it in a more efficient manner. They have longer catalyst lifetime, and they tend to generate higher yields of products. So for what we intend to use, we want to use this molecule that has this manganese center surrounded by this macrocycle. Now, importantly, there must be this manganese-nitrogen bonding interaction. Manganese-nitrogen bonding interaction. So we achieve that by adding small molecules containing nitrogen, and they're referred to as N-donor ligands. So N for nitrogen, and ligands is just a fancy term for a small molecule. And what this does in the presence of this nitrogen donor ligand, it enhances the reaction rate, and it stabilizes the formation of an intermediate, which is required to make the chemical reaction happen. So now that we've identified the porphyrin that we want to use, we want to surround this porphyrin with a protein environment. What does that achieve? Well, it turns out that porphyrins, they tend to dimerize, meaning two units come together to form a larger unit as it forms a chemical bonding interaction. And that is unproductive because that does not make the ODS reaction happen. We intend to use human serum albumin, or HSA, which is structurally flexible, and it is an ideal template for generating artificial enzymes. So we have our manganese porphyrin. We're surrounding it with a protein environment. Now we want to make the lifetime longer. And we do this by encapsulating it in a sol gel made up of a silica network. And when we do that, it forms this glassy material. So this stabilizes the system without compromising enzyme activity. And we know this works because it does work for hemoglobin and myoglobin, our model system. So in general, we want to make our HSA manganese porphyrin. So I abbreviate it as HSA for human serum albumin and manganese porphyrin for that catalyst part. And then we encase it in the glassy material. So the first step to do is take our manganese porphyrin, insert it in the protein. And we verify the formation using absorption spectroscopy. So shown here are the absorption spectra of just HSA, manganese porphyrin, and we know that we've made the complex because published data also shows similar peaks in the ultraviolet and visible region. Now remember that manganese-nitrogen bonding interaction I told about that is crucial. Well, we add small molecules containing nitrogen to potentially generate that interaction. And shown here are the different types of N-donor ligands, so those nitrogen donating molecules that we've tested that do show subtle differences in the absorption spectrum. So something is happening, but we have to characterize them in more detail. So we have here a molecule called imidazole with the ring, and then we have amines here, which are simpler versions of these N-donor ligands. So we need to characterize this in more detail. But in the meantime, what we've been doing as a side project is we've been trying to simulate the bonding interaction of the N-donor ligand within this protein cavity to see where it might potentially bind. So here we are using a compound called 1-methylimidazole, and we're docking it into the protein structure of human serum albumin with that porphyrin. It binds close to the periphery of the porphyrin, and it has a fairly high energy of 2.3 kilojoules. Generally, the more negative that number, the more stable the complex, but it is noted that that metal center is iron instead of manganese because that's what's present in the crystal structure. So beyond attempting to make the catalyst, doing docking simulations, we also wanted to do a test on this ODS reaction where we are taking dibenzothiophene in the presence of myoglobin as a catalyst, so not yet our own catalyst, and adding this oxidant, and we want to see what happens. The goal for this part of the experiment is just to see whether we're seeing any changes in the features of dibenzothiophene versus the oxidized project, and ultimately trying to optimize the conditions for when we have our catalyst ready to use. Shown here is an absorption spectrum of dibenzothiophene, so the peaks are very prominent in the ultraviolet and the visible region. Myoglobin, on the other hand, has a prominent peak at around 420, 450 nanometers. Now, in the presence of an oxidant, that's when we would expect the reaction to move forward. And we would expect that the original features that we see from the reactants would look different. And that's what we do see here. So the defined peaks from the sulfur compound now disappear, and myoglobin's peak shifts to around 425 nanometers, which might be indicative of the formation of an intermediate. So because these are very preliminary studies, this is a new project, obviously we want to optimize the conditions in more detail. So our immediate plans for now is to develop the catalyst and then study the structure of the protein, because the protein structure may change when we insert the manganese porphyrin, and then importantly, characterize the manganese-nitrogen bonding interaction. Once we have achieved that glassy material, we want to study the topology of it before and after the reaction. And then ultimately, when we do have our catalyst ready, we want to perform the ODS reaction and characterize the product to see if it has been oxidized. And we also want to characterize yields. So with that, with my speed talk done, I'd like to acknowledge the American Chemical Society Petroleum Research Fund for funding this project, Penn State Behrend for providing scholarship money to my students, Erin Trail, who performed the ODS reaction and is currently working very hard to generate the catalysts, and Ashley Lombardo for doing the computer simulations. And of course, I'd like to thank the organizers of the Regional Science Consortium 16th Annual Symposium meeting for coming up with an awesome meeting despite COVID. And with that, I'd like to thank you for your attention.
Thank you so much, Grace. That was very interesting. I'm going to see if any questions load through here.
All right, thank you.
All right, it doesn't look like we see any questions coming through. Thank you so much for your presentation.
Thank you.
On that note, we are going to break for lunch, so we will be back at 11:50. Thank you, everyone.
Thank you.
It's no secret, education is the key to success. Serving more than 2,200 families, WQLN brings learning to life by combining PBS Kids programs with reading and hands-on activities.
PBS Learning Triangle, I feel like it's a simple step for parents to grasp. So it's not something over their head or too complex.
Watching my students for the 4 weeks develop a little bit more of their personalities, have the opportunity to read and have the opportunity to be excited.
People say it takes a village to help raise students. It also takes a village to educate them. So WQLN coming in to partner with our kids helps those kids get those strategies and the parents the strategies that they can help their kids at home.
You're part of a community and we're all here together and we all care about the children and the future of the community.
To become an EITC partner and support this program, visit WQLN.org.
ETD Photography is an Erie-based media and creative services company by Eric Dye, creating media to expand your reach and further your goals. With a diverse project history, ETD Photography brings unique and comprehensive creative media to Erie individuals, businesses, and organizations. Services include photography, videography, web design, logos, podcast production, YouTube channel production, live streaming, and more. Check out our website at etdphotography.com.
Hello, my name is Dr. Jeanette Schnars, and I'm the Executive Director of the Regional Science Consortium. And I'm here at Presque Isle State Park today exploring the priority wetlands that we've been working on with our collaborators to restore over the last decade. Now, wetlands are a critical ecosystem that help protect Lake Erie and other bodies of water that they're connected to from runoff, and that would include sediment in the runoff, nutrients, and pollutants. And then wetlands also serve as critical habitats for all different kinds of species, including plants and insects, fish, birds, and other mammals that live here. Today we're going to investigate the relationship between humans and wetlands and how this has changed over time. So wetlands, once thought of as swamps or just pools of stagnant water that were unsightly, were often destroyed as a result of construction and development. However, today their importance is realized, and a healthy wetland exhibits high plant diversity and animal diversity. However, due to invasive species and some other human interactions, some wetlands are not considered healthy, And that's where we come in as researchers to help restore these areas.
My name is Holly Best. I'm one of the assistant park managers here at Presque Isle State Park. I work with the resource management program. A large part of that program, we try to manage invasive species that are growing on the park. Some of the main targets are Phragmites and narrowleaf cattail. Phragmites are specifically important to manage because not only are they very prolific when they come into an area and take it over, they spread through rhizome, they also spread through seeds. They also are allelopathic, so they actually put poison or toxin down into the soil and prevent anything else from growing. So that makes a monoculture of that plant, which for species diversity is very detrimental. At this park, we're very fortunate that we are able to use herbicide to help control these species. We have done everything from have interns with backpack sprayers, doing a very focused attempt, all the way up to helicopters actually doing kind of broadcast spraying over some of those very large monoculture areas. We're fortunate that we're able to use that as a control technique because if you don't have that ability, it's very hard to control these species and try to return these areas back to their native environments.
My name is Jen Salem. I'm the horticulture specialist for the Regional Science Consortium, and I'm also the founder of Go Native Erie. Go Native Erie is a program that operates under the Regional Science Consortium, and it helps educate the public about the benefits of native plant species and using natives in their gardens. Native plants are actually, they're defined in two ways. They are plants that were discovered here growing in North America at the time of European settlement. But a better definition of that is that native plants are a plant that has been in an area for a long time and it has evolved with something else to form an important relationship.
We are in the Regional Science Consortium greenhouse at the Tom Ridge Environmental Center. So we use this for propagation. So we get things growing and started here.
So this is the beginning of the process. So every year I'm looking at what seeds can I grow, what did well. Every year we're trying to increase our diversity and the amount of plants that we grow, but also pay attention to things that are going to do well. It's not just about quantity, it's also about quality. So now we're down at the Regional Science Consortium's cold frame greenhouse on Presque Isle State Park. And this is used as a distribution center and holding area for all the plants that are produced in the Regional Science Consortium propagation greenhouse. So when plants grow up to be a certain size and they're ready to be planted in a wetland, they are brought down here and held at this site and cared for. Then when we have groups of volunteers and employees that can come and help, we decide what plants are going into each of the restoration locations, and we'll just load these all up on a truck and have a planting day.
All right, so I hope you enjoyed the tour of our wetlands and all the work that we're doing with our collaborators to help restore these areas. So if you'd like to learn more about this project or other projects, please visit the Regional Science Consortium's website at www.regsciconsort.com.
Hello, my name is Dr. Jeanette Schnars. I'm the Executive Director here at the Regional Science Consortium. So the RSC is a collaborative. We are made up of colleges and universities, state and federal agencies, K-12 school districts, as well as other nonprofits with similar missions. And some of the research we do focuses on water quality, especially here at our swimming beaches, harmful algal blooms, different types of fishery, including yellow perch and walleye, invasive species, wetlands, native plants, and even shipwrecks and exploration of other submerged cultural resources in Lake Erie. If you get a chance today, make sure you ask at the front desk for a tour to learn more about the different projects that we have going on back there. So we use these research projects as fuel for some of our education and experiences that we offer to students in our school district membership. So these students get to experience things with invasive species, with our native plants, with fisheries, and we even have other experiences that they participate in, including a college prep workshop series. To find out more about our research and education, and also to find out if your organization, school district, college or university is a member of the RSC, just check out our website at regsciconsort.com.
Hi, I'm Dr. Jeanette Schnars. I'm the Executive Director of the Regional Science Consortium, and I'm standing here on the shores of Lake Erie, which is known as one of the best sport fisheries in all of the Great Lakes. And two of the most popular species include yellow perch and walleye. The fishery historically has always been great here, but it has had some pressures in the past, including over-harvesting of certain species and other things like invasive species. These invasive really paid a toll on some of our best sport fisheries.
Hi, my name is Greg Andreso, and I'm a biologist from Gannon University here in Erie. And for about the last 20 years, my students and I have focused on the fish population in Presque Isle Bay behind me. A lot of our work has focused on the round goby. The round goby is a non-native species from the Black Sea, Caspian Sea part of Eurasia. And it was first introduced into Lake Erie in the early 90s by ballast water in freighters. So these freighters brought these fish across the Atlantic and offloaded them unintentionally into the Great Lakes. So a lot of the work that we've done is focused on round gobies and their interactions with two species of mussels, which are actually non-native as well, what we commonly call zebra mussels. And there's actually another species that's closely related called quagga mussels. And our work has really looked at predation by round gobies on these Dreissenid mussels, the zebra mussels and quagga mussels. Round gobies eat things like smallmouth bass eggs, so they've had an influence on the ecosystem. They're here to stay. I don't think we're ever going to get rid of them, and part of my job and the job of my students has been to try to better understand their roles in that ecosystem.
Hi, I'm Dr. Casey Bracha-Wilson from Allegheny College. I am studying the invasion of round gobies from the Lake Erie watershed into the French Creek watershed, which is a stream that ultimately flows to the Mississippi River. So this invasion is a really important one that we've been studying and monitoring since they've first been introduced, probably through some type of fishing activity, whether it was through a boat or through bait bucket transfer or something like that. We have a lot of projects going on, including what the round gobies are eating, how they're impacting native fish through behavior and aggressive interactions, also how they can change communities through eating macroinvertebrates and other fishes and their eggs and those types of things. French Creek is an extremely important stream in Pennsylvania. It houses more diversity than any other stream in the state, including many threatened, endangered, or species of concern. So round gobies have recently been known to eat native mussels in the juvenile phases when they're very small. And so a lot of the mussels in French Creek are threatened or endangered species. And so the impacts to mussels from round gobies, we don't know exactly how catastrophic that will be, but it could be really bad for the French Creek watershed.
The Pennsylvania Fish and Boat Commission regulates fishing here in Erie, Pennsylvania, and on the waters of Pennsylvania for all of Lake Erie. Lake Erie has been subject to a lot of invasive species throughout the years, all of which have unknown effects when they first get here, and it's only through their actual arrival that we're able to manage them. 2018, the Pennsylvania Fish and Boat Commission joined with our cooperators in Ohio, Ontario, and New York and started tagging walleye here off the shores of Pennsylvania, right here off of Walnut Creek Marina, looking at spawning site fidelity as well as fish movement throughout the summer months and even into the winter. To date, 41 walleye have been tagged here at Walnut Creek and released back into Lake Erie, and we've watched those fish travel for almost 2 years now. We have 21,000 detections of those fish in the last 2 years. And with that, we were able to see if these fish do exhibit spawning site fidelity and return to Pennsylvania waters to spawn. The Pennsylvania Fish and Boat Commission operates the Lake Erie Boat Angler Survey annually from May 1st to October 31st. We run a bus route style creel survey in which we sample anglers and boaters from Walnut Creek, Lampe Marina, East Avenue, Boat Launch, and Northeast Marina. In this process, we count all the boats that launch, and we count all the boats that land at those marinas during the times that we're there. And while we're there, we try to interview all the anglers that land. We try to find out some economical information from them, where they have traveled from by getting their zip code. how many folks were here, how many folks fished, how many rods they used, how long they fished, what they were targeting, and what they caught. And from this information, we can start to come up with a figure of how many fish are successful, or how many fishermen are successful, and how many fish are harvested from Lake Erie annually by our recreational anglers. The Pennsylvania Fish and Boat Commission is very interested in sustainability. So with all this information and all this work we do throughout the summer with the other jurisdictions that manage Lake Erie, we're able to make sure that the fishery is sustainable and that the anglers have the best opportunity of success every time they go out in the water.
So as you can see, our evolving relationship with the fisheries and the people that live here is so important, both historically as well as currently. And researchers will continue to work on the different issues that arise and challenge our sport fishery, and managers will continue to manage and sustain these populations for the future.
It's no secret, education is the key to success. Serving more than 2,200 families, WQLN brings learning to life by combining PBS Kids programs with reading and hands-on activities.
People say it takes a village to help raise students. It also takes a village to educate them.
You're part of a community, and we're all here together, and we all care about the children and the future of the community.
To support the program, visit WQLN.org.
ETD Photography is an Erie-based media and creative services company by Eric Dye, creating media to expand your reach and further your goals. With a diverse project history, ETD Photography brings unique and comprehensive creative media to Erie individuals, businesses, and organizations. Services include photography, videography, web design, logos, podcast production, YouTube channel production, live streaming, and more. Check out our website at etdphotography.com. The Regional Science Consortium is a collaborative of colleges, universities, state and federal agencies, K-12 school districts, and nonprofits. The mission of the RSC is to promote and enhance our knowledge and understanding of the Lake Erie and Ohio River Basin ecosystems through research and education. The RSC also provides educational lessons and experiences for students, including forensic science escape rooms, hackathons, and a college prep workshop series. Each year, the Regional Science Consortium hosts an annual research symposium to highlight local research and projects. The Regional Science Consortium's 16th Annual Research Symposium, November 4th through 6th, will be live on YouTube at regscitv.com. Presenters will highlight local research on water quality, harmful algal blooms, wetland restoration, invasive species, fisheries, and more. David Frew and Jerry Skripczak will present Wednesday evening, November 4th, and PADCNR Environmental Education Specialist will present November 5th. All are welcome. Details online at regsciconsort.com. Supported in part by WQLN.
temperature, pH, conductivity, dissolved oxygen. And so we measure this handheld unit against the buoy, and that helps us determine if we need to calibrate it or not, make sure it's measuring correctly. Temperature is 22.250 degrees Celsius. This unit right here is our weather station. Measures things like wind, air temperature, even takes data on rain. In the center of the buoy right here, we have our beacon. That light is a white light that comes on at an interval of every 2 seconds to make sure that mariners in the evening or at night are aware of the buoy and can see it. Our Garmin GPS unit helps us keep track of the buoy. If it were to ever break free, we would be able to find it. We use these solar panels here to recharge the marine batteries inside the core of the buoy. So they've taken energy from the sun every day and recharge those every night. Our antenna right here helps us with cellular reception, so we transmit our data every 20 minutes through a cell phone connection or a cellular modem back to our computer that processes the data. And then on the opposite end of the buoy, we have a video camera, and that video camera allows us to get 20 or 10-second clips of the conditions out here, the lake conditions, and update that to our website so people can see what those conditions are. Inside, in between all of these solar panels, is the wave meter. And that wave meter is measuring these waves, which are pretty decent today. And those waves are being monitored over a 10-minute interval, and then it will give you the maximum wave height as well as the average wave height during that time period. Now, under the buoy is what's called a water quality sonde, similar to what we were just using and dropped in the water. And that's going to measure things like temperature of the water, pH, dissolved oxygen, turbidity, and blue-green, the presence of blue-green algae. So the Regional Science Consortium has 2 weather stations on land and 4 buoys that they put out each year. This is our nearshore buoy, 2.5 miles off of the lighthouse at Presque Isle State Park. We also have another buoy similar to this, a little bit smaller, that we deploy off Walnut Creek to the west of here. And then we have 2 smaller beach buoys that we deploy off Beach 2 and Beach 6. Those beach buoys are just measuring parameters about the water, like temperature and the chemistry of the water, and that helps us with predicting if there's blue-green algae, harmful algal blooms, bacteria in the water, so we use that for a lot of research. The buoys are deployed in May, so as early as we can get out here, and then we usually retrieve them around October at the end of the boating season. Okay, so now we're back to my office, and this is where the information from our weather stations and our buoy come back into the computer and then get processed and eventually to the website. So this is the software that runs the data that comes in from the buoy, and it does some calculations to figure out things like the maximum wave and average wave, as well as your maximum wind, your maximum rain, and then averaging those data points as well. So right here you can see we're looking at the nearshore buoy data. We have a lot of the information you might see on the website, as well as some information that we need here just to make sure things are operating correctly. And we can scroll through any of our weather stations or buoys, and we can also make sure they're all updating when they should. So this will scroll new data about every 20 minutes. And then this data will be processed, and I'll just click on our website. This is PALakeErieBuoy.com. And then if you come into our website, you can see our weather stations as well as our different buoys. And this is the nearshore buoy that we were just visiting. We click on that and it will go ahead and load the data, and we can see how big the waves are, how fast the wind is blowing, and what kind of temperatures we have out there. So right now, looking at it, we have about a maximum of 2.8 foot waves, about an average of 1.6. And when we were just out there earlier today, we probably saw good 2 foot waves with a little bit of white capping. All of the data looks good and current, and that's how we process the data.
So another project that we work on at the Regional Science Consortium is looking at barotrauma and yellow perch. And yellow perch is a really important fishery to this area. And it's one that reproduces naturally, so we want to make sure we take care of it. And one of the issues is that late in the summer, yellow perch can be as deep as 50, 60, even more feet of water. And when anglers pull them up, a lot of times their swim bladder will expand, which pushes on their stomach, and their stomach protrudes through their mouth. And there's other symptoms that they experience as well, such as bulging eyes, hemorrhaging around the fins. And so what we're trying to do is see if they are capable of recovering from this barotrauma, the stress. And one of the ways that we do this through some of our research is by putting them in these cages. And with these cages, it's almost like an elevator, and we can sink them right back down to the bottom pretty quickly. Now on these cages, we outfit them with a GoPro camera across the top, and we can monitor what they're doing for the first hour that they're submerged. And we have found that within about 10 to 15 minutes, these perch will right themselves. So when we put them down, they're kind of belly up and bloated. So in about 10 or 15 minutes, they'll right themselves or turn themselves right side up again. Their stomach goes back into their bodies, and they're able to swim around like a normal fish. So then the next question is there any latent or delayed mortality? Did they have any injuries inside their bodies? And we found after 3 days, they are still healthy fish and able to overcome this stress. So we'll go ahead and we'll deploy this cage. So when we're out here, we'll collect perch by angling. We'll take that perch, we'll put it in the cage, we'll lace the cage up, and then we'll go ahead and put it down to the bottom. So when we pull these cages back up after 3 days, the perch are still healthy fish and they were capable of overcoming that stress. However, that's the story we found in the beginning of the season when the bottom temperatures were still pretty cool. As summer goes on, the bottom temperatures in the water get warmer, and as temperature increases, dissolved oxygen decreases. And when this happens, that's just another stress that's put on these fish. So we find that when we pull these cages up, we have about 100% mortality. And so between the barotrauma as well as low dissolved oxygen, high temperatures, these fish just can't overcome all of those stresses. So early in the season, real early in the spring, we might find yellow perch as shallow as 17 feet of water. And as spring comes on and moves on into summer, we find that these fish migrate from the shallow waters out into the deeper waters. And that's how we can see that, you know, the seasons are changing and that these fish move with the change in temperature, other fish as well, and they move out into the deeper waters by the time we get to summer.
Hi everybody and welcome back. I'm Sean Dalton. I'm the lab and field coordinator here for the Regional Science Consortium. And without further ado, we're going to get started with our next session. We have a very exciting session next, and we're going to start it off with Greg Andreso from the Biology Department of Gannon University, and he's going to be talking about the growth rates and catch per unit effort of yellow perch and white perch in Presque Isle Bay from 2016 to 2020. So Greg, take it away.
Thanks a lot, Sean. Appreciate the introduction. And before I get started, I'd just like to thank Jeanette, Amber, Eric, Sarah, and Sean, and everyone else associated with the Regional Science Consortium to make this symposium possible this year. I know we went kind of into the unknown, and I think it was pretty bold, and it looks like it's working out quite well. So I appreciate the opportunity to come talk about some of the work that a couple of my colleagues and I, Chris Dempsey and Michelle Coons, as well as a bunch of our students, including Nolan Pyle, who did a bunch of the data crunching for this presentation. So again, I'm going to talk a little bit about some work we've been doing with yellow perch and white perch in Presque Isle Bay as part of a larger project that we began back in 2016. So in October 2016, Chris and Michelle and I began a monthly sampling project on Presque Isle Bay, where we engage a lot of our students in our freshwater marine biology program, as well as biology majors. We anchor down at the same spot every month in the bay. We do some work there, and then we do a fish trawl, and I'm going to be talking about some of that work from the fish side of this long-term project. But just to give you a real quick overview, each month beginning in October of 2016, we do a bunch of water quality and water chemistry work. That's really headed up by Chris Dempsey. You can see the type of things that we record out there. And then we do some biological sampling as well. Michelle Coons heads up the zooplankton community assessment with some students. And then I kind of head up the fish part of it, but we all work together to do this work. And again, it really involved quite a few students over the last 4 years. All right, so in terms of the monthly fish sampling we use the Environaut, which is a research vessel owned by Gannon University. We use pretty standard bottom trawling gear with a single warp line. The trawl we use is about 20 feet or 6.3 meters wide across the foot line. We drag that along the bottom. We do 10-minute trawl once each month, and we record all the fish that we collect. And for several of those species that are of interest commercially, perhaps, as well as recreationally, we also take length data. All right, so the area of the bay that we sample with our fish work, I highlighted here with this oval. We tried to rule out variation by sampling in different areas and on different dates. So we pretty much hit that same path every month. We get a little bit of drift depending on the wind, but all of our sampling has occurred within that shape that you see there. All right, now there's a lot of data here. I don't plan on going through it, but if there are any kind of fish geeks out there, we could always come back to this if there are questions. This is really the summary of our catch between 2017 and 19, and what I included here are years in which we have one sampling date per month from June all the way through November. And with those data, you can see we've collected over 13,000 fish. Almost all of those have been released to the bay unharmed. We get very little mortality. They belong to 19 species and 10 different families. And I'll be focusing on 2 of those species here today. But to just kind of cook those data that you saw there between 17 and 19, here's a pie chart, and I wanted to emphasize the 5 species that make up the largest portion of that catch. The yellow perch, 63% of that catch in terms of individuals. White perch, so you can see between the 2 of them, they're making up about 84% of that total catch across that 4-year period. We see brown gobies, another one of our non-natives, spot-tail shiners, bluegills, and then the other species make up typically less than 1% of the catch each, and usually far less than 1% of the catch. And so again, the 2 species I'm going to be focusing on here today, the yellow perch, I show you a range map here, and the white perch. Of course, we have to be careful here. They both have perch in their common name. They're actually members of 2 different families, the Percidae for the yellow perch and the Moronidae, which are temperate basses actually in the case of the white perch, okay? And I just wanted to point this out. The white perch is actually a non-native to the Great Lakes region. I got into Lake Erie about the middle of the 20th century, It's an important species in estuary communities along the East Coast. I know there's a couple talks after mine talking about white perch in these estuaries. What happened, they were able to colonize this part of the country in the Great Lakes, moving up through the Hudson River Valley. And then with the help of the Erie Canal system, were able to get into Lake Ontario, make their way into Lake Erie. Notably, Lake Champlain, a lot of interest in white perch there, which have invaded very, very successfully. And then closer to home, Chautauqua Lake, you can see a bunch of data points there. Chautauqua Lake has a very large population of white perch as well. And there's been a lot of question about the effect of white perch on native species. There's been maybe a little bit of fear of that, but at least to date in Lake Erie, I'd probably argue that they haven't had a very large impact. So how does the catch per unit effort, we'll be talking about numbers of fish collected per kilometer trawled of yellow perch and white perch vary across the season and across years. So I want to show one length frequency histogram here. So we go out on the boat, we net up white perch and yellow perch, and we measure every one of these individuals, and we can plot out here numbers of individuals on the Y and length of the individual measured in total length, that's tip of the nose to the tip of the tail, and just plot numbers. And these are pretty common looking data for fish. We see a few peaks here. So this peak here would be young individuals. These would be young of the year. And then we get some separation between them and those 1 year old, or they're 1 year old plus a little bit more. So people call them 1+. And then we see a little bit of overlap here with 2 year old or 2+ fishes. So what we'll do, I'm going to focus on young of the year individuals and then those 1+ individuals. And then a similar frequency distribution here for white perch. This is very typical what we find in the bay, sort of a single peak here of small individuals, young of the year, and we get very, very few larger individuals that would be 1+ or older. We just don't find, in our sampling at least, older, larger white perch in the bay, which we think is very interesting. All right, so in terms of catch per unit effort here, I'll introduce you to this figure. So in the first year, that's what the ones represent down here, May of their first year through December of their first year, and then 2 would be in the second year of growth. So those would be fish that are 1+ year old. And then just numbers of individuals per kilometer trawled, or catch per unit effort. Always in these figures, we're going to see yellow perch with the blue bars and white perch with the red bars. I didn't do that because of our political climate, but just a couple colors I selected there. I just thought of that yesterday, actually. And we see here, when we sample in June, we don't turn up any of these young of the year fish. And then we start seeing both yellow perch and white perch start to show up in July. We typically get pretty large numbers in August, September, October, and then often a pulse of yellow perch in November. And if you notice, the white perch here are essentially gone. Those young of the year white perch are gone by November, and that is consistent across years. So you also see here that there's quite a bit of variation. These error bars are one standard deviation of the mean. So across sampling years, we get quite a bit of variation here. Out in those 1+ year old fish, you can see their numbers are substantially lower in terms of the yellow perch than those young of the year yellow perch over here. So we tend to get a drop off between that first and second year. So here I just broke down a similar figure and by year class. So I have the 2019 cohort in yellow, the 2018 cohort in purple, and so forth and so on. And you could see here that the 2018 cohort, those fish that hatched out in 2018, that was a very large cohort showing up in August. And then we see numbers dropping off a little bit in September and October, and a little bit of a resurgence in November. That's kind of a common pattern. In contrast, our 2017 cohort, you could see in green there, kind of numbers that first season, and very small numbers of those fish when we would have collected them in 2018, when they would have been 1 year old. And then this big red bar here, I just want to point that out. That was our first sampling year, that 2016 cohort. So the only data we have from them will have been in October, and then a giant pulse of them when we ran into them in November. And then we were able to see those fish kind of track into their second year, which would have been in the 2017 year. You see them in red here. Okay. Similarly, white perch, just breaking those guys down by year class, it looks like 2017 was relatively a very good year for those young of the year white perch. In 2018, 2019, much less so. But again, those 1 year old fish, in other words, catching them in their second year, we just are not finding those 1+ fish in our sampling. So we think what is going on here, it looks like those young white perch may be moving into the bay when they're a few months old, and then staying there for that first season, and then kind of moving out of the bay by November, not to come back. That's at least sort of our operating model at the moment. All right. So what about growth rates of these fish in those first two seasons? How much are they growing in that first year and then in that second year? So just to give you an idea of this, I put size frequency histograms here for the 2017 collection year, June, July, August, September, October, November. Again, yellow perch in blue, white perch in red. Those small fish, those young of the year fish, notice here in June, we're not turning up any of them, and they start showing up in July. And I built this figure to show how these young of the year fish, these distributions of those young of the year fish are shifting to the right as the season goes on. So you could see that young of the year cohort incrementally getting larger here month by month. And then again, if you look in November, you don't see any red down here. Those young of the year white perch are just gone. So I think that gives you an idea of what we're able to do here. And remember, we're able to trace the growth of these fish into their second year and through their second year as well. So this is just the summary here. Average length of yellow perch, again in blue, and white perch in their first, right, with the 1, and their second growing seasons here with the 2. And this is all fish, thousands of fish here from all of those different year cohorts with the error bars, again, 1 standard deviation of the mean. So we see nice growth here of both yellow perch and white perch through that first season, again, picking them up in July, carrying them through November. On average, the white perch ending up about 75 millimeters after that first growing season. Yellow perch, on average, a little bit less, about 70 millimeters. And then these are the winter months. Of course, we're not sampling then. Then we pick those fish up again in their second year. And by the end of that second season, we don't have many of those 1+ white perch, but the ones we do at the end of that second season reaching about 160 millimeters total length, and the yellow perch only about 125. So it looks like for that first year, the yellow perch and white perch growing about the same rates, and then our white perch kind of taken off, growing a little bit faster in that second season. So what we need to do some literature searching here, and what can we say about growth rates of these fish in the bay compared to yellow perch and white perch elsewhere. That's certainly on our agenda. So a little bit there with growth of those individuals, and it's pretty consistent across years too. I didn't show you that figure. All right, and then I know there's been a lot of talk recently about the condition or the state of the yellow perch population in Lake Erie. There's some concern about that, numbers being down. So I thought I'd at least show you one slide here. What can we say about that 2020 yellow perch cohort? How did that hatch look, at least in Presque Isle Bay? So this figure, similar in design, we have length on the x-axis, numbers of fish here on the y. Notice my scales are different year to year. I just want to show kind of relative abundance in this figure. And then our 17 cohort, 2017, is shown in the solid blue, and our 2020 cohort there in the hatched or lighter blue. And in July, we went out there and this year, and we turned up a handful of young of the year yellow perch that concerned us. We certainly typically find quite a few of them in July, like we did in 2017. Notice just literally a few young of the year yellow perch in July of 2020. We did get quite a few 1+ fish out there, right? That would have been the 2019 cohort. When we look down here in August, this is not typical, right? Our 1+ individuals, fish that have gone through one growing season, greatly outnumber our, again, light blue bars here. That would be the young of the year yellow perch that hatched out in 2020, right? Typically, we'd see a pattern more like we did in 2017, where the young of the year fish here in the solid blue outnumber the 1+ fish. Okay, and then in September, again, relatively low numbers in 2020 compared to 2017. October about kind of evened out. And then when you look, I put November here, we actually collected last Friday, so that would have been October 30th, but we're calling it November, close enough. We see, again, compared to 2017, a lot fewer numbers of those young of the year yellow perch, and our 1+ individuals pretty similar 2017 to 2020. So at the surface, our initial reaction here is that at least in the bay, it did not look like yellow perch had a very good spawning year based on the number of young of year that we are showing in 2020. And remember also, 2017 was actually the poorest year of the 17, 18, and 19 period. So even compared to that low year in 2017, 2020 appears to be sort of a leaner year for those yellow perch in the bay. So with that, I'd like to just acknowledge certainly a lot of our students in freshwater marine biology and our biology major have been involved in going out in the boat, collecting all these data, the fish work, the chemistry work, zooplankton work, support from the biology department and the Murawski College of Health Professions and Sciences, as well as Captain Sam, who's a captain of our Environaut and always doing an excellent job for us. So it looks like there might be a couple minutes. If there are questions, however those are fielded, I'm happy to entertain those.
Thanks so much, Greg. We do have one question for you. Are most of the perch that you catch in the bay year-long residents in the bay, or do they exit the bay and spend time in the lake as well?
Yeah, you know, that's a really good question. I know a lot of people have been interested in that. If you look at those size frequency distributions, the bulk of the yellow perch we get are young of the year and 1+. We do get some larger fish, like in June, for example. If we're trawling in June, we'll get some larger individuals. We think we're getting them kind of fish that are stragglers heading out to the main lake after the spawn. So it looks to me, at least, and I know people may not, we might not all be on the same page, but I think the bulk of the adult perch in the bay come in late winter, spawn, make their way back out to the lake. But that being said, it certainly looks like there are resident adult yellow perch in the bay. You know, we get some larger yellow perch, a handful of them in our trawling in July, August, September. I know there's been interest in that very question.
Another question that we got is, why do you think that the older yellow perch were not found?
Yeah, you know, sampling is always, it's exactly that. You're sampling. Larger fish in general, I think, have an easier time avoiding getting collected in trawls. So we might have some bias there towards our smaller yellow perch. But again, our feeling is that that there's just not a large population of adult yellow perch in that bay in the summer. Now you could catch some, right, and make dinner out of them, but certainly not the numbers that you would find in, for example, February or March, when you have those lake fish coming in to spawn. So we think that the bay is a really, really important nursery water for both of these species. Yellow perch spawning in the bay and their young using that for 1 year, probably 2 growing seasons before heading out into the lake. At least that's kind of the model we're working with here.
Thanks so much, Greg.
Yeah,
we're going to go ahead and get started with our next talk. We have Eric Anderson on the effect of tail and body area on swimming kinematics in fish. So Eric, I'm going to turn it over to you.
Howdy. I'm going to share my screen here for you. All right. How's that working? Great. Well, it's great to be here. Thanks to the organizers. I've come to the symposium for several years and actually only had students give talks. So this is the first time I think I'm giving a talk at the symposium, and I'm looking forward to it. And thanks for being here. So this talk is about a topic that my students worked on in my lab using a large data set that we've taken on swimming kinematics at Woods Hole Oceanographic Institution. And this particular project actually wasn't a project that we had planned to do the data taking for. We were looking at frequency and amplitude of tail beats in fish. But one of the things we realized very quickly was that this data set was very rich. And so I've gotten into the habit now of bringing students into my lab and having them sort of look through this data set and make hypotheses and try to test them. And this is an example of a project that came from that. The lead on this was my student, Anna Truman, from the Department of Mathematics. She's an undergrad at Grove City and then graduated last year. She's now at YSU studying mathematics. Alec Gaiman was in the mechanical engineering department here at Grove City, and he's now at CMU studying mechanical engineering. Micah Warren was in the biology department. And so you can see it's a very multidisciplinary team. I really enjoy that about my work because it's biomechanics. I get to work with all kinds of fun people. My background is both biology and physics with a little bit of mechanical engineering in there. So anyway, let's talk about the effect of tail and body area on swimming kinematics in fish. So the motivation of the work in my lab is looking at optimization in undulatory propulsion and fish swimming in general. We also look at things like the fins of squid. We look at jet propulsion as well in squid, but we focus mainly on undulatory propulsion. We look at biomimetic propulsion in plastic strips as well. But we always like to get back to the real organism to see if there's anything going on that's more highly nuanced than what we can see in things like flapping plastic strips. This has application to fish ecology and evolution, which I'm very interested in. For example, if we find out that fish have a resonance that depends on their body stiffness or their length, How might that help us understand why particular fish are a certain size when they're pelagic during their life cycle, and perhaps they're more sedentary at another point in their life cycle? And maybe this has to do with physics. These are some of the interesting questions we're interested in. There are also folks that are really interested in biomimetic propulsion and building vehicles that are more efficient or more maneuverable because they mimic biology. And so some of our work has application to that. So we're going to look at two questions. The first question we're going to look at is, do fish modulate their tail area when they change their swimming speed? So do fish modulate tail area with swimming speed? Now this kind of came from something anecdotal that we've watched a lot of fish swim in flow tunnels in my lab. And there's just a gut sense that sort of counterintuitively, fish tend to make their tail smaller the faster they go. Now, this was only anecdotal. This was us talking about what we thought we saw while we watched fish. And we thought, well, we better go measure that. So, so here we have a fish. We've got its tail area. It's swimming fast. And here's a fish. The tail area is a little larger while it's swimming slow. So this is our hypothesis. And we're going to take a quick look at that. And then we're going to look at our bigger question, which is, do fish with a higher tail to body area ratio? So here we have a fish that has a higher tail area to body area ratio than this one over here, little tiny tail area compared to the same body area in these two diagrams. Do fish with a higher tail to body area, like this green one, use a lower relative amplitude beat of their tail? So the beat of their tail being the peak to peak motion back and forth of the tail. That's my Y0 here. My Y-axis goes into the board. And the length is the fork length of the fish. So if basically it's the sort of percent amplitude of body length. And typically in fish, that's about 20%. Fish move their tail back and forth about 20% of their fork length. And so what we're wondering is, if you have a bigger tail, can you get away with beating your tail less in terms of amplitude? And we know in hydrodynamics that that could mean that you're more efficient. And so here's a fish with a smaller tail compared to the same body size or body length. And maybe it has to beat its tail further back and forth, which would lead to a larger wake and less efficiency. So that's our second question. Is there any relationship between tail area and body area, and the amount a fish has to move its tail back and forth? So how do we do this? I'm going to spare you a lot of the technical details, but basically we swim fish in large flumes at Woods Hole Oceanographic. We're building a flume right now at Grove City College. It's an 8-inch by 8-inch, but these are more like 18-inch by 18-inch. This is a bluefish, probably about a foot to a foot and a half long. We've used 5 saltwater species: bluefish, mackerel, scup, which is kind of like an ocean panfish if you're not familiar, dogfish, and striped bass. We use stereo imaging to get the information, and I'll show you more about that in a moment. And then we use image processing to get kinematic data and morphometric data from the fish. So this is what the flume looks like. There's a small striped bass in there. We've got the cameras lined up here. My student is collecting data. In the back, we have an LED backlight so that we see a silhouette of the fish in our field of view. We collect the fish in Nantucket Sound and bring them in a live well, and then keep them in large tanks at Woods Hole Oceanographic. And the imaging, again, is stereo imaging. We've got 2 cameras looking at this mackerel right here. You can see the resolution. We go 50 frames per second. And we've been able to figure out a way to get the data off in real time at 50 hertz so that we don't have to save images, which has been a great help. And our position error is less than 1 millimeter in our stereo imaging. So we're getting really good kinematic morphometric data. We save 1 snapshot per swimming sequence, and we do thousands and thousands of swimming sequences at different speeds. And this is a problem in this particular experiment. And again, this particular study wasn't in our minds when we were taking the data, and we would have taken more images otherwise, and you'll see where that comes in later. So anyway, we can use the stereo data to find the tail of the fish in the two fields of view, plot the fish's tail moving back and forth. So you're kind of looking down on the fish right here, and the fish's tail is moving back and forth across the flume. And we can then find the average position of the fish as it's kind of moving back and forth across the flume, subtract that out and get just the tail motion. And then we can find the peaks, calculate an amplitude of motion in centimeters or millimeters back and forth. You can also calculate the frequency, but we're interested in the tail amplitude, so that's what we can get here from this data. To get the body area, the backlighting makes life easy. We just get rid of all the white pixels and we cut off all this junk from the bottom of the flume. And the computer just tells us how many pixels are dark, meaning it's the fish. And then we cut off the tail and measure the pixels in the tail. And that's how we can get areas. We can then convert that to millimeters easily by just sticking a ruler in there. So this is our 2D method of calculating areas of the tail and area of the fish. So here's some data from our question number 1. Does tail area change? Does a fish change its tail area while its swimming speed changes? So this is for a mackerel. Here's the swimming speed down here. Here's the tail area. The magnitudes don't really matter. We have a fish swimming over a range of 30 centimeters per second up to 90 centimeters per second. And you can see that there's very little obvious change. There's a slight gradual uptick here. So it almost looks like it's against what our gut told us, that the tail area decreases slightly as it increases speed. We then took the data that all looked like it was perfectly 2D in the sense that the fish was straight in the flow. There wasn't a little bit of a curve to its tail or anything like that, and just picked out what we called the best data because of the 2D. We didn't want any 3D effects. And we saw basically the same thing. And sorry, the slopes and the R squareds aren't in here. This is my student's graph, and I don't have the spreadsheet on me. But the basic story is there's not a whole lot of change in tail area with swimming speed. But we wanted to double check this since we had stereo data. We wanted to do it in 3D and see if doing 3D gave us a little bit higher resolution on what's going on. So you can take 2 pictures of the tail, and another student of mine who's on this project, Alec Gaiman, wrote some code that allowed us to pick points around the tail on both tails from the stereo images, and picking the exact same positions on both images of both tails allowed us to, and I've warped it a little bit here so that the tail matches what's in the plots here. Here is the side view of this tail that we took that data from. And because of the stereo view, we can really this is cool, we can look from behind. So right now we're looking from behind the fish. This is the back of the tail. And then this sort of more triangular kind of looking part is the front edge, the leading edge of the tail. I've got the green dots labeled in both. And you can see there's an extreme amount of curvature in the tail as you look up from behind the fish swimming, even though from the side view, you'd figure that the tail was just flat. So certainly 3D makes a difference. So here's some morphometrics. The interesting thing here is the range. So the fish's tail is bent to such a degree that over this range, there's 1.64 centimeters. So clearly there's some 3-dimensionality. Well, the orange is the 3D method, the blue is the 2D method, and as you'd expect, the 3D method gives more area because of the curvature, the bending into the field of view. And sure enough, there's about 20% more area when you use the 3D method, and there's lots of Y depth to the tails. But we don't really see anything different here. It still doesn't look like fish are changing their tail area at different speeds. This is a bluefish. So now moving from mackerel to bluefish, I don't want to go into all the details here, but sort of same story. in fact, a little clearer picture from the 2D to the 3D. The 3D orange shows, again, not much tail size modulation. I was amazed by this. I was sure that I was seeing this when I was watching these fish swim. So it just shows that you really have to take the data and can't go by anecdotal info. So anyway, switching to the question 2, which I don't have a lot of time to dig into, but basically here is the tail area to body area ratio that I talked about, okay? And so you can see that the tail area for mackerel compared to the body area is about 10%. So the tail area is about 10%, but for a bluefish, it's more like 15 to 16%. And these are significantly different from each other. I've got scup in there, but I'm not going to talk about them. So we do have a difference between the tail area to body area of these two fish, mackerel and bluefish. These are 95% confidence intervals. And so here's the final story. So here I have the tail area to body area. Here's mackerel with its 10%. Here's bluefish with its actual like 14%. Long story, but anyway, they're different. And if you look at the amplitude that the tail moves, the mackerel moves its tail a little bit more back and forth, 21% of its length compared to the bluefish, 18%. So it's not a big effect, but when it comes to hydrodynamics and resonance and drag, sometimes a little bit of a difference can mean something. Here are those numbers comparing mackerel and bluefish, and you can see these are 95% confidence intervals. We're very close to being statistically significant here. We've got to take more data and look into that. But in conclusion, the 3D tail areas are better. Question number 1, doesn't seem like fish are modulating their tail area with swimming speed. There's some evidence that tail size compared to body size does make a difference in terms of how a fish beats its tail, and we'll have more for you on this next year. Just want to acknowledge my funding from Grove City College and the facilities at Woods Hole Oceanographic. Thank you.
Thanks so much, Eric. We're going to give it just a couple moments to see if any questions roll in.
Sure.
So we got a question. How did you sample the fish that you brought to the lab? Were they on a boat or a trawl or?
Yeah, so we catch them on barbless hook and line, which is very challenging, so that we don't damage them. And so that's how we do it. We try really hard to pick our size and species so that we can sort of fill in the different lengths and morphometrics for the different species. Yeah.
Does the ratio seem to remain the same regardless of what age the fish is?
That's a really good question. We haven't looked at age. I do have the data to look at fish length versus the ratio, but I haven't looked at that. So thanks, whoever asked that question. I'm going to add that to the list of things to look at for sure. That's great.
We have one more question that came in for you. How does this discovery play into the fish's ability to feed swimming prey?
You know, it's definitely early for me to sort of apply this to that, but what a larger amplitude can do is cause the hydrodynamics to be less efficient. And so you could make the hypothesis that fish with sort of poor hydrodynamic efficiency compared to another may have to feed more, or their ecology might be different. They might be more sedentary than a sort of more high-performing swimming fish. So sorry, that's about the best I can do for you on that one at this point.
Thanks so much, Eric. We're going to go ahead and move on into our next presentation.
Excellent, thank you.
Next up, we have Jessica Briggs, who is an undergraduate student from the Pennsylvania State University, University Park. For judges, that last name is Briggs, B-R-I-G-G-S. And she will be talking about the potential impact of toxicants on white perch, a key species of New Jersey estuaries. So Jess, I'll turn it over to you.
Thanks, Sean. Okay. So good afternoon, everyone. Like Sean said, my name is Jessica Briggs, and I am a senior at Penn State University. And my project today is called The Impact of Contaminants on White Perch, Moroni americana, a key species of New Jersey estuaries. This presentation is the final product of my virtual internship from this past summer with the NOAA Fisheries Howard Marine Sciences Laboratory in Sandy Hook, New Jersey. So unfortunately, because of COVID, I wasn't able to physically go to Sandy Hook, but that also meant that a lot of our data collection was delayed into the late summer, fall and winter months of 2020. So this presentation is going to be focused mostly on the procedures as well as the expected results of this currently ongoing project. The overarching goal of this presentation is to discuss the external morphological features of white perch and how these traits and metrics can be measured to show pollution damage to individual fish. So for this project, we are collecting fish from two different sites, first being the Lower Passaic River in northern New Jersey, which is subject to a lot of different industrial contamination, as well as the Mullica River in more southern New Jersey, which is serving as a reference site to the Lower Passaic. A little bit more background about the Lower Passaic River. It is in a very highly industrialized area in Newark, New Jersey, that has been subject to industrial pollution for the past century, if not more. But for this project, we are looking more specifically at the pollutants contributed to the system by the Diamond Alkali Company Superfund site. So that can be shown right here. The site is right on the banks of the Lower Passaic River. And from the 1940s through the 1960s, this site was home to manufacturing of agricultural chemicals such as DDT and Agent Orange. So we now know that those are very harmful to human health. So in the early 1970s, this site was shut down, and soon after, in the mid-1980s, it was listed as a Superfund site. So it was listed as a Superfund site because they found things like dioxins, pesticides, and other hazardous substances in the soil and groundwater physically on this site, as well as dioxins, PCBs, heavy metals, polycyclic aromatic hydrocarbons, or PAHs, and pesticides in the sediment of the Lower Passaic River that neighbors this site. So this project as a whole is contributing to NOAA's natural resource damage assessment of this Superfund site, and my work specifically is going into the injury assessment and restoration planning stage. So we are hoping to help contribute to that stage by answering the main research question: how has contaminant exposure damaged organisms in the Lower Passaic River? And we're going to be trying to answer that question using white perch as a model organism. So we decided to use white perch because of their ecological significance in the Lower Passaic. So white perch are generally a very pollutant tolerant organism, which means that it takes a lot of pollutant being present for them to show damage. So what that means is that if the white perch are showing damage, other pollutant intolerant organisms are probably also likely to be damaged by the pollution in this area. White perch are also the third most common fish species in the area after mummichog or killifish and inland silverside. White perch and mummichog are also the only two resident species that can be caught in the Lower Passaic River all year long. In addition to the ecological significance, these fish also provide a lot of ecosystem services to the community that they surround. So white perch is a common game fish, as people are very familiar with in Erie. Unfortunately, you're not currently allowed to fish in the Lower Passaic River because it is so polluted, but the goal is to eventually restore it so that white perch can be caught and eaten from this area. White perch are also a predator that consumes a lot of different smaller fish and insect larvae, which means they're a big part of the food chain, giving, again, another ecosystem service. So for this project, we are doing collections, like I said before, from the Lower Passaic River and the Mullica Rivers, with the goal of collecting about 10 to 20 fish from each river during each collection day, with hopefully about an equal number of male and female fish during each collection. And the collections were taking place in August, October, and December. So the collection method is going to vary and has varied based on the conditions in the river at the time. The late summer collection was using line fishing and trawling. All of the collection is partnering with local fishermen or university staff in the area that knows the behavior of these fish. So basically they're just kind of tweaking it so that they can get the best catch for that time period. After the fish get back onto land, they're going to be photographed dockside and then again in the lab, the left side, so that it's all standardized. And then the photos are going to be analyzed in NIH's ImageJ image analysis software. So the first group of metrics that we are looking at for these white perch are related to body length and depth. These size variables are shown to be impacted by TCDD or dioxins, PCBs, PAHs, and heavy metals. So exposure to these kind of pollutants can cause a lower length at age, as well as lower width to length ratios. So that length at age is going to be determined by comparing either this fork length or standard length to their age, which is determined by an otolith analysis. So an otolith is a small ear bone that can be extracted from the ear and has annuli or rings that you can count to show how old the fish is in years. So then when comparing that length to the age from the otolith, you can tell about how long it is and what you can compare it to what's normal for that species at a certain age. The width to length ratio is determined by comparing this body depth measurement to the fork length, and that's going to tell you like the fullness of the fish. So if that's a lower number, it's a less full fish, there's less biomass. So both of those things being depressed would mean a slower growth rate, and that can cause lower fecundity in the adult fish. That smaller size can also lead to increased predation. So the measurements for this group of metrics as a whole are the body depth, fork length, standard length, and an otolith analysis. The next group of things that I'm looking at are lesions and parasites. So it has been shown that fish that are impacted by PAHs, PCBs, dioxins, and heavy metals tend to show the prevalence of these lesions or get parasites a lot more commonly than fish that are not exposed to pollution. So lesions can take their form in a lot of different ways, one of which is skin reddening, another is the loss of scales, which can also be seen up here, a hemorrhaging, which is bleeding from below the surface, as well as ulcers, which is kind of the eating away of the flesh into the body cavity of these fish. It can get pretty intense. So lesions and parasite presence are both usually a sign of reduced immune system capabilities, which then allow for further disease to spread in the population. So overall, the measurements being taken for this group of metrics are a presence or absence of lesions, as well as a number if they are present, a presence or absence of parasites, as well as a description if they are seen, and a presence or absence of hemorrhages. The next group of metrics that we are looking at is coloration and pattern. So this fish right here is a very normal adult white perch that is darker yellow, a more concentrated color up top with a lighter, brighter white color on the underbelly of the fish. So we have seen in past research that when fish are exposed to pollutants, they can sometimes experience something called bleaching or blanching, where they lose concentration of their color and they tend to be a less colorful and intensely colored fish in general. So we are going to be trying to quantitatively measure that by using a measurement called the mean gray value in ImageJ. So as you can see here, this one area is a lot darker and more concentrated color than this 2 area. And when you look at the mean gray values, the 2 area has a higher number returned than the 1 area. So we are assuming and thinking that fish caught from the lower Passaic River or fish that have been exposed to pollution are going to return higher mean gray values. That's going to kind of show blanching has occurred. Another common coloration trait on white perch is a pink or purple coloration underneath their chin, on their underbelly, and on their front and side fins during spawning season. So we are also going to be using that mean gray value to look at the intensity of that coloration, as well as using a plugin called Color Threshold that allows you to pick out the different hues that you want to look at. So in this case, pink and purple, and will identify and quantify the area on that fish that is covered by that coloration and that hue. So we are then going to compare the fish from the lower Passaic and the Molucca, and we're assuming that fish from the lower Passaic are going to have less intense pink and purple coloration, and over a lower amount of area on their body. Another common thing that is impacted by pollution, specifically we found evidence from it being impacted by heavy metals, is the pattern formation. So this fish has one like normal stripe, it's called their lateral line. And sometimes when fish are exposed to pollution, their stripes can become more wavy. So we are going to be looking to see if that wavy character is seen in these fish by measuring the stripe length right along that line and comparing it to the full length of the fish, with the idea that if the line is wavier, it's going to be longer when compared to the total length of the fish. So that's just kind of because when you are exposing a fish to pollution, their immune system function can decrease, and that will stop pigment from being placed in the right spot. So overall, the measurements that we are looking at for this group of metrics are the yellow intensity, purple area, purple intensity, shape length, and shape character, or a qualitative kind of measure, just saying, is the line straight or is it wavy, so that we can validate that stripe length assumption. The next group of things that we are looking at is the craniofacial morphology. So it has been shown that fish facial structure can be impacted by dioxins as well as PAHs. So that can take its form in a lot of different ways. It can be a shortened operculum to expose the gills, a longer, more extruding lower lip, as well as something called pug-headedness, or a rounding of the skull, which is all not normal. So those abnormalities can result in reduced ability of these fish to capture prey. And so for measurements, we're basically just going to be looking at these fish and noting if they have any abnormal facial features, and then describing them if they do. The last group of things that we are looking at is fin size and necrosis or decay. So this has been shown to be impacted by dioxins, PCBs, and heavy metals in the past. So the two things that we are looking at are the length along like the fish's body, as well as the height of the fins to see if the fins are smaller, as well as a scoring system for necrosis or decay. So this fish up here would have a score of 0 because its fins are fairly pristine. That means that there's no signs of damage. A score of 1 would mean that the fish has some fraying in their fins, but only of the connective tissue and not of the rays. And a score of 2 would mean very severe damage. Not only is the connective tissue being eroded, but also the rays of the fin. So all of these things would combine to impacting the fish's swimming ability, which would then reduce their ability to evade predators. Overall, the measurements for this category that we're going to be looking at are the fin lengths, the dorsal fin spine length, and a necrosis score. Now to go into some expected results of this project. So the first that we are going to be looking at is the expected results on fish's body weights and body sizes. So this specific study was looking at the impact of dioxins on weight in freshwater fish, and was by Kleeman in 1988. So if you zoom in on one of the more representative graphs of this whole study, it shows that fish that were exposed to no dioxins or lower levels of dioxins were able to gain weight at a higher rate, with the y-axis being weight and the x-axis being time, than those fish that were exposed to dioxins at higher levels, with the highest level just not even managing to survive. Another expected result is going to be craniofacial abnormalities in these fish that we catch from the lower Passaic River. So this graph over here is the results from a study looking at adult fathead minnows after they were exposed to bitumen, which is a PAH, as larvae. So they were exposed for 21 days to this PAH as larvae, and then allowed to grow out into adulthood in clean water. Even though they were living in clean water for most of their lives, there was still a majority of them, even in this section, or maybe not majority, but a large amount of fish were seen to have craniofacial abnormalities compared to none being seen in the reference sediment. So this study kind of shows that even though these fish might not be exposed to the pollution for their entire lives, even just a small amount of exposure for a portion of their life can impact them for the rest of it. So in conclusion, I know that this is a very kind of overwhelming spreadsheet, but I promise it does make sense. So the organisms, after they are exposed to pollutants or something like dioxins, PCBs, or heavy metals, that can have a lot of different impacts on the individual fish, from things like external lesions to coloration abnormalities, body length and depth, and those all have a specific impact on the individual fish. However, if a lot of those individual fish have that thing, they are going to go and then impact the population by either having impairment of their reproductive ability or an increased amount of mortality. And that's kind of the whole point of this study, is to show that enough fish are being damaged that it is going to have an impact on the population as a whole. So especially for marine populations, quantifying a population impact is very, very difficult, but that is going to be worked on by more experiments that are happening in the Howard lab to get a more complete understanding of the impact on these white perch and hopefully the population level as well. Finally, some acknowledgements. So I would like to thank the NOAA Hollings Undergraduate Scholarship Program for providing me the funding to do this research over the summer, as well as my NOAA mentors, Chris Chambers and Ann Peterson. In addition, I would like to thank my fellow interns, Jenna Stanley, who was working on the same project, but looking at internal morphology, and Tony Slew and Dexter Davis, the other two Hollings mentored interns that were also at the Sandy Hook site, or were supposed to be. I would also like to thank the Regional Science Consortium for hosting this wonderful symposium and allowing me to present today. Does anyone have any questions?
Thanks so much, Jess. We do have a couple questions for you. The first one is, were the fish preserved in alcohol or something before the color analysis was done on them?
The goal of the initial photo taken not in the lab when they immediately got dockside, that was the goal of that photo, was so that they could be analyzed before the color changed. But when they were on the boat, they were, there wasn't a chemical fixation, but they were placed on dry ice on the boat.
We do have one other. It says, you may have mentioned this already, but how were the fish caught? And is there a way to tell if the ulcers and hemorrhaging are from pollutants instead of barotrauma?
Cool. So the fish were caught in a range of different ways. The one in August that I was around for had line catch fishing from one site, and then trawling from another. So that was one thing that I hadn't thought of. We had been looking at, especially for the fin necrosis, to see if that damage was from catching or from them actually having fin damage, but I hadn't actually thought about the barotrauma, but that would be something really interesting to look at.
And we have one more for you. Has there been any work done on population differences in morphometric characteristics in the white perch? Genetic differences in morphology, if they exist, could confound some of your analyses.
There have been more population studies done in the Hudson River and then in the Chesapeake Bay, but there hadn't been any that I had seen in the lower Passaic and Newark Bay area.
Thanks so much, Jess. We're going to go ahead and get started with the next presentation. Next up, we have Amber Stillwell from the Regional Science Consortium, and she is going to give us a harmful algal bloom update from the Regional Science Consortium. So Amber, the stream is yours.
Hi, everyone. Thank you, Sean. As Sean mentioned, I am the research coordinator at the Regional Science Consortium, and I will be sharing with you today a lot of different things about harmful algal blooms. I won't specifically be talking about fish, but this is a topic that heavily involves all of us and all of the work that we do around the waters of the Great Lakes and beyond. So I'll be talking to you about a harmful algal bloom update from us, the Regional Science Consortium, and I'll be taking you through several different research projects that we have been working on. And I'd like to introduce my special guests, my dogs, Chloe and Getty, who you may hear outside my door whining. If you do, I apologize for that. So for anyone that may not be familiar with cyanobacteria and cyanotoxins, just let me give you a very brief introduction. Cyanobacteria are blue-green algae. They're actually a bacteria that functions like an algae. So there's a lot of, I guess, murkiness around the name cyanobacteria. But for the purposes of this presentation, I'm going to stick to cyanobacteria as well as what they produce, which are known as cyanotoxins. So cyanotoxins are the toxins that are produced from the cyanobacteria, whether that is intracellular or extracellular. And these cyanotoxins are so important because they can have really detrimental effects on humans and animal and our health. Specifically, they can cause skin rashes, neurological symptoms, liver failure, but more importantly, and I think more scarily, they can cause unknown long-term health effects from chronic exposure. So what we're going to be talking about a lot tonight is exposure to dogs. I guess it's not night, it is the afternoon. I'm sorry, I'm usually presenting in the evening to students. So the toxic levels that dogs can be exposed to are much, much different than for humans. So the levels at which dogs start to experience adverse health effects are about 0.2 parts per billion of the toxin microcystins specifically. Whereas humans, on a recreational standard, just going into the water to recreate, not necessarily directly drinking the water, we don't start to see humans having adverse health effects until about 8 parts per billion. And the threshold that I have listed there for you is 20 parts per billion of microcystis, which is the no contact advisory threshold that, that we abide by. So one of the scariest things about the cyanotoxins is that they can be present even when the cyanobacteria is no longer visible. So it's really important for us to be monitoring for these toxins and to test for them because they're completely invisible in the water when the cyanobacteria has dissipated. They can also persist in the environment for several weeks. And what we have been seeing over time is that there are certain places where the cyanobacteria will persist longer than others. So that can be an issue when we're talking about monitoring and the public, because a lot of people think that we just maybe forgot to remove the advisory or forgot to lift the advisory. But in reality, these toxins are really sticking around for long periods of time. So that is just my introduction, and I'll be taking you through 3 different projects. The first one, we'll be talking about the harmful algal bloom monitoring that the Regional Science Consortium does in Erie County. The second project is our newly launched canine cyanotoxin testing program, which works with veterinarians in the local Erie area. And the third project is saxitoxin, which is a cyanotoxin, and mussels in Presque Isle Bay. So these are our HAB monitoring locations in Erie County. And you can see we monitor 6 different beaches that are labeled in the red pins in the top left map. They are labeled as zones 1 through 6. We also monitor multiple different locations that are high priority areas around Presque Isle Bay because they're easily accessible to anglers, to recreators, and to people that are walking their dogs. We also monitor all the way out to almost the Ohio and New York state lines at Elk Creek, Avonia Beach, and Walnut Creek, as well as Lake Cliff Boat Launch, Shades Beach, and Freeport Beach. Additionally, we process samples from Erie Waterworks and Northeast Drinking Water Authority to kind of serve as an early warning system to see if there's any cyanotoxins in the drinking water. All of these samples are collected at the same locations every week from the end of May to the end of October, and they're all collected at approximately 1 meter below the surface. So these are subsurface samples that I'll be sharing with you today. So some updates from the 2020 monitoring. You can see in the line graph that we had several different peaks of cyanotoxins at several different sites. This line graph is specifically for microcystins. So you can see that the gray line for Ferry Slip peaked very, very high for microcystins in the late summer, late July, mid-August, whereas other sites might have peaked a little bit earlier, and we had other sites peak later in the season. But if you look at my annual average microcystins concentration bar graph, you can see an average of Presque Isle Bay sampling sites over the last 5 years. And the reason that this is important is because we're able to look at all of the sites as an annual average around Presque Isle Bay to almost like get an average for the bay itself. And you can see in 2016 and 2017 and 2018, we had relatively average or relatively the same amount of toxins. In 2019, we had a bit of a spike, but in 2020, we had a much lower amount of toxin than in previous years. So I like to think of this as, all of the things that 2020 has delivered us so far. Harmful algal blooms were not that bad. So as I was looking at the data over the last 5 years, I just wanted to show some notable trends. So like I said before, we have 5 years of really solid data, and looking at all of the sites, separating the Presque Isle Bay sites out into the 21 testing weeks, we can take an average of the bay each week of each year. And this allows us to look at the trend of the bay as a whole over the past 5 years. So you can see that on average, we see a peak around week 10 or mid-July every single year in the entirety of Presque Isle Bay. The next step would be for me to take this data and compare it to the environmental parameters that we also test for, and particularly dissolved oxygen, relative humidity, and water temperature, to see how those changes at each of our sites are impacting each of these weeks of testing. So these are the annual averages of microcystin concentration at each site every year for the past 5 years. And this just shows a similar trend to the first bar graph that I showed, where we saw relatively similar concentrations of cyanobacteria, of microcystins the first 3 years that we analyzed. But in 2019, you can see that there is a sharp increase. And then in 2020, almost across the board, we had lower concentrations of microcystins in all of the sites. So I'm going to talk a little bit now about our canine cyanotoxin testing program. And we're really very excited about this program. And I'm excited to share it with you because it's all about protecting the dogs that are being potentially exposed to cyanobacteria. So for those that might not be familiar, dogs can be really negatively impacted by cyanotoxins. And the reason is because they are more likely to directly consume the toxins from the water, or they'll be driven to actually ingest and digest the algal crust and mats that are formed along the water edges. They can also be exposed because of swimming in the water and then self-grooming later when they leave the water body. So because the symptoms that can arise can be so delayed in being shown, there's a really big time lag between when the animal was swimming and when we actually can confirm that it was cyanotoxin poisoning. So the symptoms that animals can exhibit include staggering, difficulty breathing, weakness, salivation, vomiting, convulsions, and the convulsions can eventually lead to the death of the animal at times. So there's really no good relationship that has been built between how often dogs are being exposed to cyanobacteria and cyanotoxins in the Erie area. So that's the gap that we are really trying to close with this program. So with this program, we created CCTP or canine cyanotoxin test kits. And we recruited participating veterinary offices to take samples from canines that have a possible exposure. They then submit those samples to the Toxins Analysis Lab in Syracuse, New York, and they then obtain the results and can inform the pet owner as well as us what happened to their animal. There are very few treatments for cyanotoxin poisoning after the animal starts showing symptoms like convulsions. So there may be little that can be done to help the animal other than wait it out and hope that it clears through their system. However, this does help us build that relationship and look for potentially potential symptoms that are more related to cyanotoxin poisoning than we would have previously known. And I just want to share with you that my dogs, Getty and Chloe, have a very different way of understanding how they want to participate in this program. We had to build this entire program and put it together when we were on quarantine. So they were very helpful, as you can see, in getting all of the test kits together and sending them out, putting everything in shipping containers. So the samples that we are looking to obtain from veterinarians are hair samples, urine samples, gut contents, and in the case of a post-mortem patient, it would be hopefully a liver biopsy. All of these samples will be tested for an entire panel of cyanotoxins, including microcystins, anatoxin A, saxitoxin, and cylindrospermopsin, among other things. The veterinarians were asked to look for the symptoms in this list here, including things like extreme fatigue and hot spots that were not on my original list. But like I said before, we don't know everything about this, so we gave them a more broad list of symptoms so that we could get a broader amount of information. And when the samples were submitted, or when the samples are submitted, it is at no cost to the pet owner, which was the whole point of this program, was to provide a service that could give us more information without producing an extra cost for the veterinary office or for the pet owner. So water samples are going to be paired with this, with the submission of the canine samples. So the veterinarians were asked to ask the client if the dog has been swimming in any kind of surface water in the past 7 days. And if they answered yes, then we had asked them to get as close to the exact swimming location as possible so that we could take a sample and submit it to the SUNY lab or SUNY Syracuse lab and get that comparison of cyanotoxin presence in the water versus in the animal. So both fortunately for the animals, but unfortunately for our research, we did not get any samples in 2020. But this sort of tracks with it being a low cyanotoxin year in the Bay. So we are lucky enough to have this research extended to the 2021 HAB season. So we will be launching this again in June and it'll go until the end of September. But coming out of this is another research project that I wanted to talk to you about, and that is increasing our cyanotoxin or saxitoxin testing so that we can see how saxitoxin is concentrating in our water and if it's concentrating in invasive mussel species. So a lot of people are very surprised when I talk about cyanotoxin being present in Erie, Pennsylvania, and that's because you normally hear of saxitoxin being in other countries where analytic shellfish poisoning or paralytic shellfish poisoning is a big problem. But with our testing and analysis over the last 3 years, we have seen concentrations of saxitoxin, both detectable amounts in some years and both in exceedances for the dog safety threshold, which is 0.02 parts per billion. So what you see here are several dates that I have listed where saxitoxin was detected in high enough concentrations to exceed the dog safety threshold. And those concentrations, like I said before, are all they are all above 0.02 parts per billion of saxitoxin. And in 2018 in particular, you can see that date in September, we had many, many sites in Presque Isle Bay exceeding 4 saxitoxin all around the bay. So we need to increase our saxitoxin testing because currently we only do it once per month. So the goal will be to increase saxitoxin testing to more frequently than once per month, maybe every other week, if not every week. And we also want to see if saxitoxin is concentrating in our invasive freshwater mussels. So we will be taking water samples from our normal areas, but we'll also be taking zebra and quagga mussel samples in quadrats from chosen areas that are more suspicious of having saxitoxin presence, whether that is looking at the annual averages to see the sites that exceed most frequently for cyanotoxins in general, or looking at the areas where saxitoxin has been detected previously. So we will sample these areas in quadrats and use a random number generator to take the samples of the mussels. We will then remove the mussel tissue from the shells and send the tissues to the toxin analysis lab along with a paired water sample from the area for sampling. We hope to do this, the mussel sampling, at least once per month in order to build an understanding of how saxitoxin is interacting with freshwater mussels in the area. So I want to just give a quick thank you to our funders, which are the Great Lakes Restoration Initiative, the Department of Environmental Protection, Coastal Resilience, or Coastal Resource Management, and our partner in the CCTP program, which is the Toxins Analysis Laboratory, specifically Greg Boyer. And I can't do this without our massive team of interns and our RSC team. So thank you all. And I've included photos of all of our RSC for you guys to see how much they really contribute to this project and kind of how much fun we get to have. And with that, I will take any questions.
I'm just going to give it a little second here, Amber, just to see if anything rolls into the chat.
Okay, thanks, Sean.
So what do you think has contributed the most to the overall increase in HABs from the last 5 years?
That's a really great question. And I don't know that I want to answer it without looking more at the rest of the data. So we, along with our harmful algal bloom samples, we collect air temperature, water temperature, dissolved oxygen, all sorts of other information. We do have an indication that Erie County as a whole is warming over the last 20 years. So it could be that, I guess if I have to speculate, it could be that with the warming air temperatures and resulting warming water temperatures, we're just creating a better habitat for these species to bloom and produce their toxins.
All right, thank you so much, Amber. With that, we're going to go ahead and get into the next portion of our session, which are the speed talks for today. So our first speed talk is Madison Betts, who will be talking on the predation response to chemosensory and visual cues in wild versus cultured Chesapeake log perch. So Madison, I'll go ahead and turn it over to you.
Thank you. Let me get this up. Can you see it? All right. Okay, so hi, my name is Madison Betts, and I am a senior student at Allegheny College. And I have been working with a team at Penn State University on our project, which the title of is Predation Response to Chemosensory and Visual Cues in Wild versus Cultural Chesapeake Log Perch. And this is actually part of a larger study run by the Penn State University team. So what is the Chesapeake log perch? Chesapeake log perch are a large elongate species of darter with a pointed snout and a yellow body and zebra patterning. They were considered synonymous with the common log perch until recently. And because of this, and because of some environmental degradation, the species populations are not well known. Likewise, little is known about its life history, other than the fact that it's likely similar to other log perch. So in general, they eat macroinvertebrates, amphipods, and isopods, and they do this by flipping stones with their snout. They spawn from April to June in tributary streams, and they lay their eggs in the substrate of riffles. And then overall, their historic range spanned in the Susquehanna and Potomac watersheds and a few smaller creeks in Maryland. However, they have been extirpated from the Potomac River, and they're currently only found in the wider reaches of the Susquehanna and a few smaller streams in Maryland. So due to their restricted distribution and their small overall population, the Chesapeake log perch is considered threatened in both Pennsylvania and Maryland. And because of this, they have various conservation efforts have started to try to reintroduce the species into its historic range. The most successful of these has been the Stauffer Lab at Penn State University. This lab has reared a total of 1,500 log perch at last count anyway, for release into the Susquehanna River. And however, they're experiencing a few setbacks, the first of which, and the one that we're going to be focusing on, being predation, both from natural predators and invasive predators. The overall project is actually going to be looking at a few different predators, including American eel and the snakehead. But for this specific part of the project, and the one that I'm focusing on, we will be looking at smallmouth bass. So the issue is that a lot of studies have shown that all different types of reared fishes do not react to stimuli the same way that wild fish do once they're released into the natural ecosystem. And this is of concern because their predation response can be innate or learned behavior. And when it's learned behavior and they don't have exposure to natural predators, this often ends up having a low survival rate overall. Sometimes their survival rate can be as low as 5%. And the idea is that we do not know whether Chesapeake log perch, whether it is innate or learned behavior for them. So this directs how the conservation efforts move forward from here. So like I said, the predator of concern for this project is the smallmouth bass. And the smallmouth bass is a voracious predator that has been introduced to the Susquehanna watershed. It's been naturalized, it's not invasive. It eats as fingerlings, they eat microcrustaceans, macroinvertebrates, and small fish. And as adults, they eat crayfish and other fish. And they've been introduced primarily because they're a highly sought after sport fish. And although they're native to the Mississippi and Great Lakes drainages, they have now been introduced all across the United States and the world. And this is why they are in the Susquehanna and why the Chesapeake log perch could interact with them. So my research question is, are predator avoidance strategies innate or learned behaviors for Chesapeake log perch? And I'm seeking to answer this by testing the question using a series of trials, visual and chemosensory trials with smallmouth bass as the naturalized predator. So the first set of trials I've conducted, or actually Penn State has conducted since I'm remote, are visual trials. And these have been conducted in a tank with a sealed plexiglass divider. And then the prey fish, the reared or wild Chesapeake log perch, is introduced to one side of the tank, which in this picture is the left side of the tank. And then the smallmouth bass is introduced to the other side of the tank. And then the idea is that when this visual stimuli is added into the experiment, the reaction of the Chesapeake log perch will tell us whether it is experiencing, whether it recognizes the predator and how it reacts to it. So predator avoidance for us will be measured by the time taken to hide, time taken to display abnormal or erratic swimming behaviors, or the time taken to halt movement, since different fish react in different ways to predation. And then this will be quantified and then averaged between the wild log perch and the reared log perch, and then compared between the two. The second set of trials are the chemosensory trials, which are designed to test whether Chesapeake log perch react to the smell of a predator in the water. And for these trials, we've designed a tank, an experimental tank that has 2 arms and a holding area. And in the picture to the right, the PVC pipes that come in at the top of the image, that's where the water will flow through. Then you have the 2 arms, and then the white horizontal piece at the bottom, that's the holding area. And then the water will drain through the end of the tank. So conditioned water will flow through one arm, and it's conditioned by holding a smallmouth bass in a tank of water for a set amount of time. And then the other arm will have control water, which is just tap water. The log perch will acclimate for 5 minutes in the holding area, and then they'll swim freely for 10 throughout the tank. And the idea is that if they recognize the predator's smell, they'll spend significantly less time in the conditioned arm and more time in the control arm. So the time spent in each arm will be recorded and then averaged and compared between the fish. So our anticipated findings, based on the literature surrounding predation response in reared fish, I believe that the reared Chesapeake log perch will not exhibit the same behavior as wild fish, meaning that predator avoidance is a learned trait in Chesapeake log perch. And some of the consequences of this is if we were to release the fish without giving them some exposure to their natural predators, then they would likely die. So if I can show that predation response is not an innate trait, then that will direct how conservation efforts for Chesapeake log perch move forward and also improve the success of the current efforts. So those, so are there any questions?
Thank you, Madison. We're just going to give it a couple moments to see if any questions roll into the chat. It doesn't look like we're getting any questions, so we're going to go ahead and take a break for a couple minutes until 1:20 with our next talk. Welcome back, everybody. Thanks for hanging in there with us. We're going to go ahead and get started with our next speed talk. We have Steve Spots from Allegheny College talking about the economic effects of zebra mussels on businesses in the city of Erie, Pennsylvania. Steve, go ahead.
Awesome. Oop. Well, hello everyone and good afternoon. Thank you all for coming. I'm Steve Spots. I'm a senior at Allegheny College and I'm an environmental science major and biology minor. And I will be talking about my senior comp project, which is focusing on the economic effects of zebra mussels on businesses in the city of Erie, Pennsylvania. So just to dive right in, we're going to do a little background on zebra mussels here. Zebra mussels are an invasive species that was introduced to the Great Lakes region in the 1980s. When they were introduced, it was assumed that they were introduced through European vessels. When they were introduced, there were a lot of issues that came about in their first couple years of introduction. They negatively affected a lot of ecosystems as well as residents along the Great Lake region. Early in their introduction, a lot of studies were done and a lot of studies focused on specifically the region of Lake Erie. Lake Erie had a lot of issues that had to do with zebra mussels affecting the reefs, commercial and recreational boats, and as well as water and electrical facilities. Some studies have focused on these particular issues. Some studies in particular focused on the electrical and water facilities, commented that once the zebra mussels would make it into the facility through the pipes, they would reproduce and start to cluster. And that clustering in the pipes would cause a lot of damage within the facility to the point where they couldn't even run the facility at all. They would have to completely shut down and clean the entire inner workings of the facility, which would leave residents without water and electricity for long periods of time, which, as anyone can understand, is definitely not something fun to deal with. Another example with zebra mussels, they would clog up the reefs within the lakes, and they would leave other fish without any place to hide, leaving them open for predators to prey on. One of the biggest issues with this species is that there is not really a solid predator to keep their population in check. So their population numbers are always increasing, and that's why the effects of them are always increasing as well. So I want to focus on the city of Erie, Pennsylvania, and I'm going to analyze and explore the economic impact of zebra mussels on select businesses in that region, mostly because a lot of the studies that have been done in the past have focused on either the whole Great Lakes region as a whole or larger cities along the lakes. And I kind of want to see where our local city of Erie kind of falls in the big picture of impacts by zebra mussels. So the way I'm going to go about doing this is through one electronic survey. And the select businesses that I'm going to be focusing on are boat marinas and boat repair shops. This survey will be sent out and it will only be long. And the 13 questions will be made up of general business related items and zebra mussels, impact, and economic questions. The survey will be no more than 5 to 10 minutes long. And currently my survey is undergoing some review to be approved by the Allegheny IRB board, which will be hopefully approved within the coming days. So some examples of these questions, particularly the business specific questions. I have two here. How long have you been in operation, and what are your business's profits for the last 2 years? These kind of questions will allow me to understand just how big these businesses are and how long they've been around. I can hypothesize that businesses that have been around for a long period of time have come to the realization that zebra mussels are an issue, but have come to deal with them. Whereas businesses that are new, that aren't as aware of it, will see it as it's in the forefront of their mind. They're trying to figure out what to do at this point. And being able to know how old these businesses are will kind of help me understand that aspect. And then understanding their profits will just allow me to understand if they're how big the company is and how influential it is on the city of Erie. And then when it comes to zebra mussels impact and economic questions, we have two examples here. in the last 2 years, what % of your total cost was used to repair damages caused by zebra mussels? This question in particular would be more gauged towards boat marinas, because I'm looking more for boat marinas to have to deal with correcting issues when it comes to their docks, boats, because the zebra mussels will attach to those surfaces, and they will then have to be repaired and fixed. And those are obviously going to be out of pocket for them, and so that their marina can keep on working. And then when it comes to boat repair shops, they will be more focused on, in the last 2 years, what % of your total revenue was used to repair damages caused by zebra mussels, because a lot of their clientele will be coming in with boats that are damaged by the effects of zebra mussels. So that's where I'm going to be gathering those two data points there. And then one other example I'm using here is in the last 2 years, when have you seen a majority of your zebra mussels damages occur? And this will kind of help me understand what season is big for zebra mussels, if it's in the spring or the summer, or if it's in the fall. So in the end, I hypothesized that zebra mussels are affecting businesses in the city of Erie, Pennsylvania, and that being able to bring that to the forefront of everyone's mind will help spark some studies focusing on how we can diminish their effect and possibly even bring their population to a halt in increasing and maybe even declining, so that we can minimize the effects in the future. And hopefully that will come to fruition. And then in the end, I just want to send a little acknowledgement out to my two professors that are helping me on this project, Professor Kelly Pierce and Professor Catherine Bender, as well as the Allegheny College IRB board head, Dr. Polson, who is looking over my surveys right now and hopefully will be approving them in a short time. Now I just want to open up to everyone, if anyone has any questions about my project.
Thanks, Steve. We're going to give it a couple moments just to see if any questions roll into the chat.
Awesome. Sounds good. Thank you.
So we have a question for you. How many of these businesses have implemented methods of dealing with these zebra mussels since they've been around here since the 90s, if any?
So to my understanding, businesses that have been dealing with this for so long have kind of just learned to work around it. Because they've been around for so long, they are a big issue, but a lot of businesses have come to the point where they've just learned to cope with it. It's kind of like a nagging issue that's always going to be there, but because there's no solution currently, it's just, no one's really been able to solve it. Some studies have been done that have come up with solutions, but one solution that was in the works was introducing the round goby, because the round goby is known to eat zebra mussels, but because it is also an invasive species, you have a lot of issues when it comes to introducing them as well. So just because there isn't really a good solution, a lot of these businesses have just kind of focused on just dealing with it. And I think that's the big issue, is I want to bring that to the forefront, that you don't have to just deal with it. We can find a solution. And that's what I'm hoping that my study will be able to do.
We do have one more question for you. Have you reached out to the Erie drinking water treatment folks at all? They have a lot of zebra mussel nightmares at their water intake tubes.
I have not reached out to them because I know that looking into, I know that they're the only water facility up there, and I kind of wanted to focus more on like branching out on getting a lot of data instead of just one specific data point. But I would definitely I would hope that my research would help them in some way, and hopefully if we can show that they're also suffering with this issue, that that will even help make it even more of a something we need to focus on.
Thanks so much, Steve. We're just about out of time, so we're going to go ahead and move on to our last speed talk of the session. Awesome. Next up, we have Kylie Weierbach from Allegheny College, who's going to be talking on using historic records and habitat suitability analyses for the reintroduction of lake sturgeon in Lake Erie tributaries. So Kylie, go ahead.
Thank you. I had a little bit of an issue yesterday with Zoom volume, so I want to make sure that you can hear me first. Okay, great. Cool. So I'm going to share my screen. Okay. And it should be good to go. Okay. Okay. Hi, everybody. My name is Kylie Weierbach. I'm a first-year master's student of biology at SUNY Buffalo State College up here in New York. And today I want to be introducing my thesis and some preliminary findings I have. And the title that I'm working on so far is Using Historic Records and Habitat Suitability Analysis for the Reintroduction of Lake Sturgeon in Lake Erie Tributaries. Also, full disclosure, I'm listed on the proceedings as being from Allegheny College, but that is my alma mater. I graduated earlier this year, but I am from Buffalo State. So. Oops. Okay. So the Great Lakes have a really complicated conservation history. They have been a major importance to the economy for decades, if not centuries, for the United States because of not just fisheries, but also because of shipping, obviously between the United States and Canada. And that has impacted a lot of the different species of fish that we have. My study species is the lake sturgeon, Accipenser fulvescens. It is a large, long-lived potamodromous fish, meaning it exclusively migrates within fresh water. And it is native to the Great Lakes region around here. It in particular has a complicated conservation history within Lake Erie. Originally, before European settlers got here, the lake sturgeon had a daily and spiritual importance to a lot of the indigenous peoples that lived around Lake Erie, such as the Ojibwe, the Potawatomi, and the Cree. But it became a nuisance species in the eyes of commercial fishermen in the 1800s because it had these rows of bony scutes across its body that were really sharp, so they would tear the nets if they got tangled in the nets, and all of the fish that the fishermen caught would be released. So the fishermen began to heavily fish the lake sturgeon itself, but not to use it, just to get it out of the lake, pile it up on the beach, and burn the carcasses, which by the end of the 1800s really made it a very rare species in the lake. But around that time also, it kind of reverted to this valuable species because the knowledge of using the entire body of the lake sturgeon for food and the hide for material and the oil for making isinglass and things like that kind of came back into the eyes of the public, usually because of Eastern European immigrants. So now it was a highly desirable species, but it was rare because it had been a nuisance species for so long. At the beginning of the 1900s, all the way through the middle of the 1900s, other environmental problems in the Great Lakes, mainly from development, such as pollution, dredging and damming of the tributaries, furthered the plight of the lake sturgeon in the decades that followed. So the past couple of decades, there have been a lot of research and reintroduction efforts and conservation efforts that have come to the foreground for the lake sturgeon. But it is believed that the quality, accessibility, and connectivity or proximity of habitat for lake sturgeon, especially the spawning and juvenile habitat, are still barriers to the fish's recovery. So the goal of my thesis is to assess 1, the quality, 2, the accessibility, and 3, the connectivity or proximity of habitat in Lake Erie tributaries for lake sturgeon using habitat suitability analysis. This technique combines multiple ranked environmental criteria that are important for the study organism, such as river substrate size, water temperature, and channel depth, as an example for the lake sturgeon, in a GIS environment, and produces a composite map showing areas that are highly suitable and areas that are very unsuitable for the goal, in this case, looking for habitat. So I would like to look at habitat for spawning and juvenile lake sturgeon, both of which are found in streams. And my results can inform reintroduction efforts across the states that border Lake Erie: Michigan, Ohio, Pennsylvania, and New York, which is important because this thesis is being funded by the Fish and Wildlife Service. Okay. To help break down the workflow of my thesis, I've been thinking of it in 3 general parts. 1, where in Lake Erie were sturgeon then, as in before they were largely extirpated from the lake. This part involved a historic record search to build a list of tributaries that used to be used by lake sturgeon. These tributaries may still be suitable today, or after decades of change both within and outside of the lake, they might be unsuitable now. This was my main goal for this summer. I started working on this project back in June, and I've majorly completed this section, which is why I put a check mark there. Number 2, where in historic tributaries could they be now? This part is the actual GIS analysis, bringing the known habitat preferences that I got from literature reviews and spatial data sets together with those historic records, which helped me build my list of the study regions essentially, and producing the suitability map that shows high and low suitability regions within each tributary. And so I'm moving into this step currently. And then the 3rd step will be where in Lake Erie could they be introduced later? This brings the focus back to Lake Erie as a whole. And it's the summary portion of the project in which I compare the suitable and unsuitable tributaries as a whole to each other on the lake, the entire lake scale. And I can make my final recommendations about where we could reintroduce lake sturgeon. Since I've majorly finished the first step of the process, I can show some preliminary findings. Across the 4 US states bordering Lake Erie, there are 22 potential tributaries that are known or thought to have been used by lake sturgeon in the past. 3 are in Michigan, they're on the left. 13 are in Ohio, 3 are in New York, and 3 are in Pennsylvania, but the validity of the Pennsylvania tributaries are the most dubious of the 22. And all of this makes sense so far, given that Ohio has the most shoreline of Lake Erie of these 4, while all the other 3 have somewhat similar amounts of shoreline comparatively. So here's the full list of tributaries of interest so far. Of these, only the Detroit River and Niagara River are definitively known to have lake sturgeon consistently spawning in them today, and a lot of research, a lot of population monitoring. And I've got pictures of them here. And also the Maumee River in Ohio has had reintroductions of juveniles in the past couple of years. They released 3,000 in 2018 and 3,000 in 2019, but they won't be reproductively active for a pretty long time. Also, I see the irony of putting the Detroit River on the right under New York and the Niagara River on the left under Michigan, but the shape of the lists and the pictures worked out that way. Okay. The immediate next steps include finding or collecting data for these tributaries of interest and considering constraints of these streams before I perform the actual suitability analysis. I have to ask these questions. Which of these streams are still accessible to lake sturgeon, and to what degree? Are there new barriers that haven't been in place in the history? Is the stream actually wide enough, deep enough, and long enough? Are these streams accessible in terms of spatial data? Could I access them by boat for side scan sonar, which is a field collection method of looking at the bottom of the stream to get a sense of how big the substrate is? Do spatial data already exist thanks to government agencies or other studies? And also, since we know that certain landscape characteristics within the whole greater watershed of the stream impact certain stream characteristics within the water, could I use surrogate landscape or watershed-based data sets in place of certain criteria to make the data collection easier? Also, feedback is pretty important to consider at this step as well. So I would welcome any comments, advice, or questions about my method so far from the audience. And that's all I have for you guys today, but I already have some people to thank. My advisor is Dr. Chris Panuto here at Buffalo State. Dr. Dmitry Gorski is from the US Fish and Wildlife Service up here in New York. He is the one that secured funding for this project. I also have a lot of people that helped me with my literature search, especially for finding the records of lake sturgeon use in tributaries. And you guys, the audience, because this is the first time I'm presenting this research to anybody, and if you'd like to provide any feedback to me outside of the chat, you can send an email to that address right there. Thanks, guys.
Thank you so much, Kylie. We're going to give it just a short moment to see if any questions roll in. So we do have a couple of questions that have come in. Do you think that commercial anglers might resist reintroduction of the sturgeon?
Netting is still used a lot in the lakes, but I think there could be a lot of overlap between commercial fishing and sport fishing. The lake sturgeon is a popular sport fish in other areas where it's not as threatened. People go out and fish for it every year and they want to get the biggest one possible. But I could foresee some pushback, especially since those nets, netting is still used, but there could also be developments in better netting that maybe wouldn't be so sensitive to the bony scutes of the sturgeon. They could also maybe just avoid fishing in the very few areas that lake sturgeon might be in the lake. But that's my guess for that.
So we do have another question. Are you considering biotic variables such as prey availability in any of your models?
That's something that I do want to look into as part of this step that I'm on currently. Step 2 is I really have to see just what is available and what is available consistently across all of my study regions. I can maybe use, I could certainly use like benthic records, and I can use community records for streams where that's available. And it's probably much more widely available for larger streams and streams that have already been studied for a lot of other reasons. But it might not be available for every single stream. But yes, that is something I would like to consider if it's consistent across the study region.
We do have a couple other questions for you. Are the sturgeons sensitive to pollution at all? The Detroit River and River Basin aren't exactly the most pristine watersheds.
Yeah, so my understanding so far is that we do know at least a little bit about the chemical habitat preferences and lethality and what kinds of things cause lethality for lake sturgeon. I know that one paper that I read was talking about water quality kind of as a mix between kind of the chemistry and kind of the turbidity of the water. And I think the general finding from that paper was that the turbidity didn't impact them too much compared to the 3 major ones that everybody always talks about for lake sturgeon, which is the substrate size, the water temperature, and the water speed. And then after that are generally things like depth and the length of the stream reach. But I haven't seen a lot of literature so far on their sensitivity to pollution. But I did watch a webinar recently that had some new sources that I hadn't seen yet. So I took some notes on that and I'll be reading more into them.
So we have one last question for you. Do you know if there's evidence of in-lake shoal spawning?
In-lake shoal spawning, yeah, there is. Especially near shore and at the mouths of these historic tributaries, some of the records or some of the best anecdotal evidence we have of sturgeon using these tributaries is actually just them being found near the mouths of the tributaries, and they might not have gone all the way up the tributaries to spawn. But yeah, the main spawning that we know of, there's spawning here in the Buffalo Harbor, and they've taken side scan sonar and they've gone and looked at the shoals, and that was in Neuenhoff et al. 2018. I believe is the main paper I read about that. So yeah, my focus is mainly within the tributaries, but depending on how much time I have and how much I can find, I might focus on near shore stuff as well.
Well, thank you so much, Kylie. We're just about out of time for this session. So with that, we're going to take a short break and we will resume with the next session at 1:50.
Welcome back, everyone. We're going to start our next session. I'm really excited about this session. It's about wetland restoration at the priority wetlands at Presque State Park. And myself, the Regional Science Consortium, as well as many other organizations have been collaborating for some time on this project. So we're going to hear all the different aspects of that project in this session. The first presentation today will be Jim Figge, He's the regional biologist with Ducks Unlimited Incorporated, and he will be talking about Ducks Unlimited Pennsylvania Conservation Overview.
Can everybody hear me? Can you hear me, Jeanette? All right. Thanks, Jeanette. Yeah, so my name is Jim Figge. I'm the regional biologist for Ducks Unlimited, but also several mid-Atlantic states. But primarily here in Pennsylvania, our focus is in wetland conservation in primarily our inland systems and coastal marshes. So I've been asked here today to give an overview of sort of the importance of wetlands to various habitat and wildlife species, as well as kind of giving an overview of Ducks Unlimited and some of our local programs that are involved here in the Northwest Pennsylvania. To start off, those who aren't familiar with Ducks Unlimited, our organization's mission is to conserve, restore, and manage wetlands and associated habitats for North American waterfowl. However, it's important to note that another portion of our mission is that these habitats also benefits wildlife and other people. And just an aside, how do we know this? You know, the Western Hemisphere is innately and well studied and known to have ties to wetlands and waterfowl. And how do you suggest this? Well, if you turn it sideways, it's a duck. So you just see for those that are less artistically inclined to view it, there's the duck for you. But in all seriousness, so with Ducks Unlimited's efforts, waterfowl is obviously the key importance to our mission. However, it makes up a very small portion of the benefits, both with species and wildlife people that benefit from this work. You know, it's well documented that over 900 species of wildlife depend on these resources, these wetland resources, throughout some point in their life cycle, 50% of which are threatened endangered species that depend on these and are almost innately dependent on these wetlands. As far as fisheries go, wetlands obviously create a lot of estuarine breeding habitat on the coastal marshes, as well as nurseries within smaller isolated systems. 66% of fish rely on coastal wetlands throughout some point in their life cycle, including those in Lake Erie. You know, more importantly for the people, 1 billion people eat fish as a primary source of protein. From a recreational standpoint, basically, here's a census from 2011 from the U.S. Fish and Wildlife Service. There's a lot of numbers and figures here, but essentially the big take homes is millions of users benefit and recreate and use these wetland resources, which translates into multi-billion-dollar investment in the economy due to these end users of wetlands. And then you can start talking about the water quality and quantity benefits, you know, from nutrient sediment cycling to from flood and erosion control, groundwater recharge, erosion and sediment control, things of that nature. So ultimately, waterfowl, at least from Ducks Unlimited's perspective and many others, are listed as an umbrella species for wetland conservation that has benefits to a whole wide variety of ecosystems, goods, and services, as well as benefits to wildlife. So can I get into our conservation programs here within Ducks Unlimited? Our efforts here at Ducks Unlimited are largely based on our science efforts. We're very much a science-oriented organization, where our target areas of interest are those of historic importance to waterfowl, as well as those of historic wetland loss and other social factors. And there's sort of a storm cloud map that kind of shows those highlighting overall areas that overlap with a lot of priorities, specifically for North American waterfowl in the breeding and non-breeding seasons. So you can see a lot here in the Northeast and prairies and boreal forests in Canada. So internationally, when we look at Ducks Unlimited, it's DU Inc., which here in the United States, DU Canada is our sister organization in the north, and DU Mac or DU Mexico to the south. Internationally, as of January 1st of this year, we've conserved just under 15 million acres in North America via investment of about $5 billion, which is all part and parcel due to a large membership partnership that we have, almost 1 million members, and as far as supporters and partnerships, it goes well beyond that. We also contributed that large amount of effort due to our efficiency rate of 82%, which basically means that of every dollar that comes in the door at Ducks Unlimited, 82% at least is put on the ground for habitat benefits, and less so into the normal day-to-day, keeping the lights on and staffing and whatnot. So a lot of it gets invested in the area. So here in the Northeast, our priority areas and our conservation mission is largely guided by what we call our Completing the Cycle initiative, which is a fundraising initiative that is directly in line with our conservation goals. And that is to provide food resources for these birds to basically make their annual cycle from the breeding grounds down to the wintering grounds and then back again. And what you're seeing here, these are our two storm cloud maps of priority areas and deliveries, and you can kind of see where there's overlap quite a bit. And since our sort of inception here in the Northeast, more in the probably mid to early 80s, we've done over 220,000 acres of conservation. More importantly, here within sort of my subregion of Pennsylvania and New Jersey, here's the conservation by the numbers. As of the end of 2019, we've completed 9 projects, altogether just under 1,000 acres, and invested $730,000. The historical numbers, almost 278 projects. We've conserved over 48,000 acres within these 2 states and invested over $21 million. So as far as projects go, we've got a whole host from acquisitions, restorations, enhancements, policy work, and things of that nature. But I'm really going to tackle into specifically our conservation efforts here locally within the northwest portion of Pennsylvania. And one of the first is our French Creek Wetland Initiative, which is a multi-county, multi-partnership effort looking at conserving wetland and riparian habitats along the French Creek, major corridors of the French Creek watershed. And to date, we've completed over 5 individual projects totaling about 640 acres, 540 which with wetland acres. And we actually have 2 projects that are no longer pending. They actually had completed as of a few weeks ago, and when the award was given, and we'll be closely acquiring that property here shortly. But to date, we've gotten a series of North American Wetland Conservation Act grants, Department of Conservation Natural Resources, and several others coming down through. So definitely at this point, we're in the millions of dollars of range of investment within the watershed. And we all look at this as a sort of a revolving lands fund, because due to the partnership between the various entities, the states, and nonprofits, as that money is leveraged from those organizations to federal funding, we're able to bounce that money back and actually make back almost all the money that we invest up front to protect a lot of these lands. So here's some various pictures of some of the properties that we've acquired over the last, let's say 5-6 years, most of which are on the main stems of the Cuyahoga Creek and French Creek. Cuyahoga Creek being an important IBA and important tributary to French Creek watershed. One of the latest ones, which is one I was originally saying was pending, but it's pretty much in completion, is our Curtis and Highback properties in Erie County, Pennsylvania. These properties will be added to one of the primary holdings of the Western Pennsylvania Conservancy. There you can see in the map, everything in yellow is pretty much existing protected land, and the black and the red are those properties we're going to be acquiring. Anything with the blue hash dashed solid or anything else is wetland and riparian areas that we're conserving with this effort. So just under 400 acres with these two acquisitions, funding coming from NACA as well as various sources from our French Creek Initiative match, DU, Western Conservancy, the state, Mellon Foundation, Richard King Mellon Foundation, and some private donors. Switching over to the restoration side, we've done a series of restorations in the Northwest. One of the latest ones that we're currently working on is what we're calling a celery swamp IBA acquisition, which is largely based around State Game Lands 151, which is labeled as the IBA sort of boundary, or a good portion of which. So we're actually working on a restoration on the existing State Game Lands 151, but we've recently acquired an additional property just north of there in the red, in the one for 130 acres, adding to the State Games. And that was this past winter that was transferred over to Pennsylvania Game Commission. But given the situation at the site, this was actually a former aquaculture site that has been left fallow for quite a few decades and has shown some chances of rebounding into some beneficial wetland habitat. However, without any intervention, it's not likely that we'll get the positive bounce back that we're looking for, at least to the extent that it's possible. Thereby, we're looking at sort of currently working on the survey work of these sort of individual ponds and whatnot, and looking at how we might be able to reconfigure those into, to turn them more into natural wetland manageable units. It's also important to note on this property, you can kind of see in the background, there's a forested wetland or swamp, if you will, that is currently, or hopefully one day, will re-support a growing heron rookery in the area. This is just a quick look conceptually here. We're actually in the survey and design phase right now, but just conceptually looking, these are for the areas in blue, obviously the main flow channels, and then the areas in yellow are what we're considering for the conceptual design units for these restored wetlands once the redundant levees from the aquaculture are pushed back in, raised back the elevation to the historic wetland elevation would have been just outside of the property boundaries. One of the last projects, and one that's been an ongoing one, and sort of, I guess my presentation is the introduction for, is sort of ongoing work at Presque State Park that's been going on for quite a while now, at least a decade or more. But as far as the youth involvement, we've been involved in a series of National Fish and Wildlife Foundation grants, obtaining money for restoration, enhancement, and even some monitoring work throughout the state park and surrounding areas. So the first phase of which was completed in 2015, which was mainly looking at invasive plant treatment and some initial monitoring and baseline information in the early 2000 to 2010 years. And that was all, like I said, completed in 2015, and ultimately led to the treatment of 964 acres of wetland habitat within the park, altogether with NIFWIF and Atlantic Coast Joint Venture funding and partner match. Second phase of which, each of these, as you'll see, is building on the last. So in 2018, we completed phase 2, which was basically everything from phase 1, the monitoring and the treatment, but we also started acquiring equipment as what that would enhance their ability to treat wetlands in the area, as well as some native propagation, collecting seeds, tubers, and other plant materials to reestablish some of these native plants that are still existing when they're in areas that have been recently treated or are on their way to actually being restored. So with that effort, we had another 442 acres treated, again with NIFWIF funding and partner match. And then phase 3, which is our current effort, and what you'll hear more about here in a little bit, is it started back in 2019, and again, took everything from phases 1 and phases 2, and then we added some additional features and whatnot. So with this effort, the idea was to try to establish some resource management type positions within DCNR to support some of this ongoing work, as well as increase the propagation efforts, We also are working with local universities to develop a strategic plan as far as a PhD student, and doing some remote sensing to determine some of this information, have better targeted efforts for some of the treatment. As far as the overall goals, we hope to treat at least 700 acres, 400 of which is untreated, 300 of which has been initially treated from past phases, and then another 30 are those propagation areas. And again, it's all part and parcel due to the investment by National Fish and Wildlife Foundation and all the partners matched listed below. So just to finish out here, just give kind of those that are not familiar with the site at Presque Isle State Park, those that can still drive around the wildlife drive, see pictures of this from time to time throughout the year. I'll say it's far less than it used to be, but just in comparison, right after treatment, this is what we've been seeing in a lot of these sites. Obviously, there's somewhat of a scar left on the landscape. However, I can assure you that that is short-lived. There's a site within less than a year, I think. The other one would have been fall of the previous year, and then spring that following year. You're starting to see a resurgence of a lot of those native species that are coming back, including, you can barely make it out, but broadleaf cattail is one that everybody thinks is common, but is in fact pretty rare in this area since there's been an overrun by invasive narrowleaf cattail. So yeah, there's a couple more pictures of the site that we've been working on, one of those anyway. I'll finish off just saying, thanks for all the partners that make this project, as well as all of our efforts within my particular region, so successful. And thanks for all of our supporters that make this work possible as well. And I'll kind of just leave it there for questions.
Hi, Jim. We're going to give it a minute for the questions on the chat box. But you had shown a map of Pennsylvania, New Jersey, and many different sites that you guys have projects on or have worked on. Have you noticed that other sites similar to the Presque Isle project, have you had success elsewhere with invasive species removal and restoration as well?
Yeah, so, I do a lot on coast, on the Atlantic side, dealing with a lot of invasive Phragmites out there. A lot of times we're doing sort of ecosystem-based restoration where we're using the environment to fight back on some of the frag, dealing with salinity, mainly looking at reintroducing tides in the areas that have been historically cut off. So we get a lot of success that way. I know some of my colleagues up in New York and New England are looking at utilizing various lake levels and management and some restoration techniques to try to create habitat out of a lot of this invasive narrowleaf cattail that has taken over a lot of the coastal areas, especially in Lake Ontario. And we see a lot in our mid-continent, a lot of our partners along coast of Wisconsin, Michigan, and those areas. But funny enough, what we call invasives up here, namely Phragmites, our counterparts in Louisiana, Mississippi, Texas, really love Phragmites because it's really what's only keeping a lot of their marsh together. So it's variable as to how our programs effectively treat or manage for invasive species.
Okay, well, thank you very much for the presentation. That was a good start to this session.
Yeah.
And we definitely know the impacts of high lake levels or high water levels. We've been experiencing that for a couple of years now. So that does impact our management for invasives. Thank you. All right, we're going to move on to our next presentation. Holly Bess is the park manager at Presque Isle State Park, and she is going to give us a resource management overview of 2020 season at Presque Isle State Park. Holly?
Thank you, Jeanette. I'm assuming you can hear me and see everything. Fantastic. So just to give you an overview, and also to kind of springboard off of what Jim has presented, we've been working on invasive species control in some way, shape, or form at Presque Isle State Park since the 1990s. And when the program first started, it was actually just a couple of interns working around in the field, hand cutting some plants. So you can tell, and you could guess that that would not be very effective or efficient given the amount of invasives that we have at this park. So it's important to start with, why is Presque Isle State Park important? There are a lot of reasons, actually. So it's important to preserve this location because we are the only beach, waterfront, shorefront, however you'd like to refer to it, in Pennsylvania. The Presque Isle means almost an island, and it is a recurving sand spit that juts out into the lake. It has Lake Erie on one side and Presque Isle Bay on the other side. So we're basically surrounded by water, and inland we have a lot of water. So we have a lot of environments that are not found anywhere else, specifically not found anywhere else in Pennsylvania because we're the only beach, but also not found in a lot of different areas of the country. So we also are home to all of the successional stages. So we go from the brand new beach as it is forming all the way up to the climax forest. So we have all those habitats and all those species, plants and animals that are found in those different areas and successional stages. Presque Isle State Park also, since it's located so nicely on the lake, is also a migration path for many nesting and resting birds and insects. It's pretty important if you think about where we're located across the lake from Canada. Any bird that's flying to us or from us from Canada is going to take a rest. They're going to rest either on Canada's land or on this area of Pennsylvania. So it's pretty important that we are here and we're maintaining those properties and habitats for them. We also are home to many, many, many species of special concern. This includes both flora and fauna. So all the plant species, fish, turtles, you name it. If it is rare and located in a sandy environment, we have it here. So basically, we're pretty important to maintain those areas for those species. We're also identified as an important bird area because of the many species that are visiting our shore, and also call this location home. The numbers vary a little bit, but we have at least 300 species of birds that are always on the park or passing through. We're also identified as a national natural landmark because of all the different aspects we have. And finally, depending on how you think about it, maybe most importantly, maybe least importantly, we are also the recipients of at least 4 million visitors a year. So that is our average. And the whole park is only 3,200 acres. So that's a relatively small space to have that many people. And I like to think of it as, we want to put our best foot forward showing these natural habitats off to those 4 million visitors. We don't want them to come here and see something that's been taken over by invasives. So that's why it's very important to manage these. So we do manage for special species, as I kind of mentioned. This is just a handful of the special species that you might recognize. Of course, we have the bald eagle. Bald eagle were present on the park about 50 years ago. They've just recently started nesting back on the park. So that is a success story that we're going to take and carry on. Also, osprey have just recently began nesting on the park. Back in the 80s, the park actually built hacking towers for them to build nests on, and they had not used them up until just a couple years ago. So that's a pretty exciting return as well. We also have up in the corner, the little sneaky little least bittern. It's a sneaky little marsh bird that Sarah Sargent will talk more about, I'm sure, as she gets into her presentation in a little bit. But there's an interesting story. So we're looking for the birds in relationship to what's happening with controlling the invasive species. We're finding them a lot more. We don't know which came first. So we don't know if we're finding them more because we're looking for them, or if they're actually here in greater numbers because of what we're doing. So we can't claim that as a win just yet, but what we can say is that what we're doing is not impacting their population because they're here and they have a healthy population. So we can take that. Also here in the center, we have a flower called the hairy cocoon. That's located in a dry, sandy area across from the lighthouse. So that's something that we protect. We make sure that we push invasive plants away from that plant so that it grows here. Once again, we have that environment here, the only environment like it in Pennsylvania, so that's pretty rare. The lupine is another flower plant, a pretty purple flower there that grows in that upland area where it's nice dry sand. So we want to make sure that we maintain that space for it to grow. We're working on trying to get the common tern to have some success out here on the park. It does visit Gull Point, and it is out there throughout the season. Common terns, they need to have like a critical mass. They have to have a large population in order to be successful with their breeding, to be able to scare off predators and everything. They haven't reached that just yet, but we do see them coming back every year, so they're trying. So we're just keeping our fingers crossed that we will be able to do that at some point in time. We also have the Blandings turtle at this park. This is the only location that is found within the state of Pennsylvania. Fish and Boat Commission is keeping a good eye on this species. They come out annually and check for them, put radio transmitters on them and see where they're going. They have successfully found female and male species. The thing that we don't know is if they are successfully actually reproducing on the park. So we don't know if they're a viable population. Because when they put on the transmitters, they have unfortunately fallen off before the females have gotten to what would be considered as the nesting area. So we don't know where they are nesting or if they are successfully nesting. So hoping to get some more information about that in the future, and everybody just keep an eye out for that little guy. We also are monitoring and managing for the piping plover. We have had success with them being back at the park, breeding successfully, fledging their young successfully. The young that were fledged this year, we have gotten reports that they have successfully migrated south. So from their little band combinations, we can tell that they made it. So that's great. We're hoping to see them back again next season to continue their population. So we're seeing a lot of success, and a lot of that is tied directly to removing invasive plants from the area and bringing back that native environment for them. So you might ask, what species are we targeting? Pretty much everything. So as we also have a ton of native plants and a lot of rare, threatened, and endangered species, we pretty much also host most of the invasive plants that are identified within the state. So a couple of the targets, garlic mustard, that is one that gets pulled out by our weed warriors, our volunteer weed warriors in the spring before the seeds go and fly all over the place. The other one that they'll manage is Oriental bittersweet by cutting some of the vines. Those are both pretty tricky things. They're very prolific, but we're trying to just keep them at bay. So that is kind of our goal with that. Honeysuckle, we do have 2 invasive honeysuckle plants on the park. The tricky thing with this is that we have to remember those 4 million visitors a year. And the honeysuckle makes up the majority of the understory of the park. So most people believe that if it's green, it's good. And when you're looking, just driving onto a park, you really don't want to see a whole lot of dead vegetation. So that's a balancing act with us managing the honeysuckle. We pick and choose where we treat those locations. We want to make sure that they don't look so bad. And also we put them in areas, we treat them in areas where we can replace them with native vegetation. So that's something we're chipping away at. And until we get a big, large project to deal with it, it's going to be a little difficult. Japanese knotweed is just a couple locations throughout the park. We're having, we pretty much have that under control. Narrow-leafed cattail and Phragmites. Those are our big number 1 and 2 species that Jim talked about in our wetland areas. The Phragmites has been very responsive, as Jim alluded to, with us, doing some treatments and using herbicide. The unfortunate next thing that happens after you remove the Phragmites is narrow-leafed cattail comes in. It seems to be a little bit more strong with withstanding the invasive control measures we're able to do and withstanding the herbicide. So it does take a couple years for us to get rid of that after we get rid of the Phragmites. So that's just something we're learning as we go. Purple loosestrife, people buy that and plant it in their yards still, unfortunately, but it really spreads a lot. It has a ton of seeds, spreads very prolifically. As the water levels have risen in the lake and all around the park and within the park, we have seen a lot more purple loosestrife germinating and kind of popping up everywhere. So we do work to remove that on the park by digging it up. That's our best way to actually physically remove it. We have also, in the past, added some biological controls in the form of some beetles, and they are still present on the park. We're going to try to get more of those, but we don't have a really good resource at this time to get them. Flowering rush, which is sad for me because I think it's such a cute little plant, is also invasive. And that's another one that is really coming on strong with the high water levels. It's really just kind of popping up everywhere. Even just along the roadside where we have had high water levels making little ponds and depressions, flowering rush is popping up in those areas. And then the last target we have here, just to highlight, is Ailanthus. this is a tree that we've only ever had a couple individuals on the park, and that's a good thing. It's something we're always on the lookout for, and we get rid of it as soon as we see it, because it is the host to the spotted lanternfly. It's one of the main hosts for the spotted lanternfly, which is known to be very destructive and an invasive beetle that we do not want to have here. So at the park, we do follow some best management practices. The whole concept here is to protect the best and preserve special habitats. So like I pointed out, the lupine, and Jim talked about native Phragmites and native cattail. We also have a cactus here on the park called the prickly pear. We also have that cocoon area. So any of those special habitats that don't exist anywhere else, we want to make sure we're preserving those the best we can and just finding the edge of those invasive species and pushing them back from those areas. So we push back that invasive edge and then just keep chipping away at it. We're also encouraging native plants to fill in, reeds and sedges. And then you'll also hear Jen Salem talk about how she's also assisting with that by putting in some native vegetation into those areas to kind of reclaim those edges and get them more native. So we just kind of think of it here as like peeling back the layers. And if you do go back into the lagoon area specifically, you'll be able to see the multiple layers and years of work that we've done, because you can see the succession of those invasive plants kind of dying back. And then you'll see another layer of healthy invasive plants that we'll get to next year. So it's a lot of job security too. So we do our treatments in a lot of different ways. People always ask how we do this. So it pretty much runs the gamut. The most precise surgical method would be glove of death. So that's why I have a picture of a cotton glove there. And that's by using a saturated cotton glove and actually applying directly to the stem of the plant. That's the most surgical method. It will only affect the plant that you actually touch. So that we use in areas where we are seeing some of these areas rebound and see a lot of natives coming back. We want to make sure we don't want to damage them in any way. So that's where we'll use the glove of death method. When we say we run the gamut, we've also had helicopters. So twice in history, we have utilized grant funding to have helicopters come and spray some of the areas that are just complete and total monocultures of invasives. Those targets have been the narrowleaf cattail and the Phragmites, mostly in the inland ponds and waterways, because we have no other way to get to them. They're not going to do any damage. There's nothing there but the invasive plants. So that's when you choose to use a helicopter. We also have within our arsenal a couple boats. We have a large pontoon boat we use as a work barge. We also have an airboat that we can spray from. We have a couple large tank sprayers. You can see that the gang kind of steps off the boat, walks into the water, goes into those areas to treat that leading edge and peel back those layers. We also will use backpack sprayers. That's one step up from glove of death. That can be a little bit more precise than using the large spray tanks. And you will see that the spray here in these pictures is blue. That's because we use an indicator dye. The dye is just basically a water-based dye that goes in and shows us that we are getting good coverage with our spray. So it makes sure that we are doing the right thing, getting good coverage, not having to go back, not having missed areas. So as Jim kind of said, we all have a role to play in what we're doing here. We have a lot of different partnerships. We have been very successful at getting funding. Most of our funding has come from the federal Great Lakes Restoration Initiative. The program we're working in with right now is Sustain Our Great Lakes from NIFWIF, as he mentioned. Ducks Unlimited is the administrator of the money. They are helping with being the applicant for this grant. So he's responsible for everything that we're doing. We're just turning in reports to him and saying that we're holding up our end of the bargain. We also have multiple partners with the Regional Science Consortium, California University of Pennsylvania. I know that Allegheny has also joined in. So they help with monitoring with plants, amphibians, reptiles, helping with water quality parameters, and also bats were added. So just kind of looking at everything as we're doing these treatments, what kind of impacts are we having? So Go Native Erie also is very involved in the project by propagating native plants and reestablishing those treated sites. Erie Bird Observatory, who you'll hear from, is assisting with monitoring all the bird populations. And also Go Native Erie and Pennsylvania Sea Grant are working together to support the Weed Warrior program, which is that group of volunteers that go out and help us with some of those species. So this year looks pretty different from other years, as most everyone else is experiencing with COVID-19. DCNR was unable to support the intern program for 2020, which is basically our staff for the resource management program. Every summer we hire anywhere from 6 to 8 interns to work just with the resource management control. So that wasn't going to work out because there was no way to safely mitigate having that many people, as well as having all the park staff be safe. So we did have a backup plan. We were able over the years to train some of our environmental education staff and get them certified as applicators and registered technicians. So that staff was able to do what they could, utilizing mostly the airboat to work in those internal waterways, also all the lagoon systems. And they were able to just work on those layers, just go in and kind of spray along the outside, push that area back. So that was pretty important. They also were able to, since they had some time, to utilize one of our UTVs and one of the tank sprayers to treat along the main roadway corridors. So as we said, we want to protect the best. We also want to stop any new little infestations when they start happening. So with the water levels increasing, we had those little pop-ups of plants, and wherever that was happening right along the main roadways, the staff was able to get those as well. So while we might not have been able to do as much as we wanted to do, we were able to get some pretty high priority areas and things done. So just to show you kind of a snapshot of what we did this year, these are highlighted on the map are all the areas that the guys worked in and where they were able to go. The numbers are a little bit misleading this year because of how we had to collect the data. The takeaways are the area treated was a little over 15 acres. And what that number means is that is the area that actually herbicide was applied to. So we applied herbicide to a total of 15 acres amongst all those work areas. The area protected number is probably high, but that's okay. So I'll explain why. Area protected is basically where we had boots on the ground looking for invasive plants. We reported that at 522 acres this year, which is definitely high. You can see on the map, anything that is shown in color was identified as an area protected. So a lot of the bodies of water were included. We can look at this two ways. Was someone, I know they had the boats in all these areas, so they were looking. Was all that area technically protected? Probably, because it's open water, so there's no plant growing there. But I do feel that this number was most likely pretty inflated. Typically, our area protected number is more closely aligned to about 350 acres, somewhere in that ballpark. So it's just what we have to work with for the data, but we do know that we applied herbicide to 15 acres. Those treatments occurred on 10 different treatment days. Basically, it was 2 staff per day for about a half a day before it got too hot to be outside. So as we move to 2021, we're kind of hoping to get back to normal, whatever that may look like after all this kind of shakes out. But we are looking for interns for 2021. If you would like to apply for one of those positions, it is to work in the field through the summer, as long as we're able to do so. Doing some invasive species eradication. You get to, if you want to apply, send your resume and cover letter to myself. My email address is there on the screen. And we have, for anyone who has worked with us in the past, we have changed the date this year. So due to some different hiring processes that are being done a lot faster than they were done in the past, the cutoff date is now January 31st. Before, it was the end of February. So I want to make sure everyone is aware of that I need to receive your application materials to my email prior to the end of January. So that is my summary on what is happening at the park and what has happened over the past year. If anyone has any questions, I'd be happy to try to answer them.
We do have a question for you, Holly. Do you know if there are any stands of the Pennsylvania endangered calms lobelia still on the park? It used to be present in modestly stable numbers at the northwestern part of Gull Point in the Sand Plain area.
That is a question that I usually refer to an expert on. So if we could put a bookmark on that question and ask Jen that question, maybe she can assist with that, because she is definitely the plant expert who is available on the call.
And one more question. Is there any plans, or have you previously used prescribed burns or fires to treat invasive plants?
Yes. So previously on the park in history, they have done prescribed burns a couple times. I have met with forestry. Just a little bit of history. The Department of Forestry controls all of the burn plans and the process. There's a pretty elaborate scheme to building a burn plan, and you have to meet a lot of different parameters. So we do have a burn plan, an active burn plan for Presque Isle State Park, as well as Erie Bluff State Park. We have a lot of challenges with doing a burn at Presque Isle, mostly because we have so many people. We have so many ways for the fire to spread. We don't have a lot of places where we can put good fire breaks besides using our own roadways. Unfortunately, there's a lot of power lines in those locations as well. And the other thing is there's always a balancing act with the humidity, level of precipitation, as well as the windy conditions that we get on the park. So we have not been able to find a lot of favorable conditions for burns at Presque Isle. The good news is we have gotten rid of a lot of the large stands that we were considering burning when we first started looking at those plans. Flip side, we most likely will be burning the fields at some point in time at Erie Bluffs. Those fields were planted when we took those out of crop production. We actually had a contract with a farmer who produced crops at those fields. Those are just cover crops that were in place. So those areas we do want to burn because that's how we're going to manage that area as a savanna. So most likely you will see in the future us trying to do some burns at Erie Bluffs. Probably not so likely at Presque Isle.
Okay, great. Thank you very much, Holly. That was a great presentation.
Thanks.
We'll move on to our next presentation. Jennifer Salem from the Regional Science Consortium is going to talk to us about plant propagation for wetland restoration on Presque Isle State Park. Jen, you can go ahead.
Hi, Jeanette. Thank you very much. So I guess before I'm going to share my screen and reply to that question about Calm's lobelia at the same time, if that's all right. And so I have not seen it. It's one of my wish list plants. So as you can imagine, because we have so many rare, threatened, and endangered plants on the park, I do have lists of things that I would like to see, and that is not something that I have come across yet. So let me pull this up and we should be good to go, hopefully. Okay, is my screen up? Okay, good. Okay, so this is my third year presenting about the Ducks Unlimited Wetland Restoration Project. And so I'm going to give you some background for those of you that have not heard my presentation before. And then of course, 2020 threw some exciting new updates and challenges at us. So I will also update on that. And then like Holly mentioned, I am now the co-director of the Weed Warriors program, so I'll touch on that briefly just to give you an idea of what that is if you're not familiar with it. So again, as our past two speakers have talked about, we have an awful lot of invasive species on the park. When Holly's team goes in and treats the areas, we're often left with pretty much like dead zones for a while. And keep in mind, plant seeds are in the seed bank and the soil, but you have a mix of the good stuff, native plants, and you also have exotic plants and then invasive species. And so what we wanted to do is try to restore some of the areas and do it in with a quicker method than waiting for things to come back and see what happens. Also, perennials can be a little fussy. Their germination is difficult most of the time or sometimes. And we also, it's like you're trying to wait for them to grow back, and it's a very slow process. So our solution was to select a few areas on the park that were previously treated to do some wetland restoration. So we'll talk about those. This is Thompson Circle, and I think this is a photo. I take a picture of it every year. I think this is a picture from this year. where you can see that it's very heavily vegetated now. In years past, it was pretty sparse after the treatment. And actually, Holly's team does an excellent job of going in and doing spot treatments on little pockets of invasives that are coming back. So if you look, I'm not sure if you can see my cursor, but if you look at the fallen tree that's kind of horizontal, in the front of the shot, there's some Phragmites that is growing in that area. That'll be treated. If it hasn't already been treated this year, that will be treated next year. And so we're going in and very carefully pulling out the invasives that we don't want, not literally, but figuratively, and keeping the native plants in there and trying to increase the populations of those native plants. So this is our Thompson Circle area. This is an overhead view, and just to orient yourself as to where this is on the park, it is between beach 10 and 11, and the big body of water that you see adjacent to that is Thompson's Bay. And let's see here. Here's Leo's Landing. This is the area that is on the bay side, Presque Isle Bay side of the park. And this is where that little observation platform known as the Feather is also present. So, and then this is the overhead view of this. So the bottom of your screen, that's Presque Isle Bay, and then where it's highlighted, that is the restoration area that we're working in. And then this is the peninsula neck, or the head of the peninsula. And this is right when you come onto the park on your right-hand side, or right adjacent to Presque Isle Bay. And here, we'll take a look at that. And also to orient yourself, if you look on the other side of your screen, you'll see a break wall and beaches. So it's on the opposite side. I like to joke around that I'm only on the beach about once every year because I work in the interior of the park or on the other side of the park. So, so our other solution to our invasive species problem is to do propagation in a way that has not been done, to my knowledge, at any other state park. And what we have is an on the park, for the park philosophy, which I will explain to you. That is basically, instead of buying things at nurseries and greenhouses and things like that, we actually grow everything for this restoration project in-house. So we're very lucky to have proper facilities, which I will talk to you about in a minute. But that means we harvest seeds. That's a fall, late summer and fall project for me. We do hardwood cuttings, which is an all winter long project. And we do that when the shrubs are dormant. And then we also do herbaceous cuttings, and that's a much smaller part of our project, but it is also something that's very successful. So we're taking everything from the park, we're looking at what's growing in each location, and then we're coming up with a species list that is very specific to each site. So we do have some species selection. I won't go into detail with what these are, but we do a mix of your carex and juncus, so your sedges and rushes. We do a lot of the hardwood cuttings that I mentioned, so those are your shrubs. And then we also do herbaceous, like flowering plants. One thing that we do pay particular attention to are pollinators. So you can see there's that's a European honeybee, but you can see that there's a bee enjoying one of our swamp milkweed in photo number 6. So, okay. So again, I had mentioned that we have an amazing facility to facilitate, sounds weird, our propagation. So we have the Regional Science Consortium Propagation Greenhouse, which is attached to the Tom Ridge Environmental Center. We also have a plant lab, which is where I'm doing a lot of prep work for the seeds and doing any plant research and things like that. And then we also have a cold frame greenhouse down on Presque Isle State Park, which is near our maintenance building. So this is what the plant lab looks like. This is during seed collection time when things are identified and photographed. And then they're processed and then put into a cooler just to put the things away for winter. And then this is our greenhouse. It's a small greenhouse, but it's perfect for propagation. This looks like a very wet time of the year. I'm thinking this is probably the beginning of March, but it's ideal. The conditions are ideal there for starting all these plants. And then we do have a cold frame greenhouse. It's basically used as a storage and distribution center. And what's cool about that is that because it is right on Presque Isle, we are very close to our restoration sites. So we load up trucks and we bring the materials to our sites. It's really a wonderful setup, and we're very fortunate that we have that. Also, because at the Tom Ridge Center, there isn't really an area to store all these plants, and we're producing thousands of plants a year, so it does take a lot of space up. And then we used volunteers and staff to do the installs of the plants. This year, of course, it's been very challenging because we weren't really able to take volunteers. Except this group is I guess they're my guinea pigs. I just had them come out a couple weeks ago, and we tried to do a socially distant planting. So if you were in the same household, you could be close together. And if you were not, you had to spread out. Everyone wore masks. Everyone wore gloves, but that's because it was kind of muddy out. And that actually worked pretty well. But we're dealing with smaller groups than we typically get. This was about 10 people. We've had groups of, 30, 40 people at a time that have come in to volunteer for us. Sorry, my lab doesn't recognize that I'm here. Okay. And then we do monitor the sites. I do a shoreline survey of the emergent zone. Bob White and I go out and do transects in a lot of the wetland areas. And we're looking at, not only are we documenting what species are we finding, but we're looking at what's coming back every year, what's thriving in an area, what populations are increasing, because that is going to guide us every year as far as what I decide to grow for those areas. So this is colleagues of mine, Sean and Sarah. We were actually looking for an invasive species that day. I'll explain more about that, but usually we're going in kayaks or waders to do a lot of our monitoring. And you might have seen this picture before. This is the reason that monitoring to me is so important, because we see firsthand what's coming back and what's not. We're going through a lot of acreage. We're documenting what we see. So we certainly don't want to stop monitoring in an area, because I feel like once that happens, then it becomes more of a challenge to keep those invasives out. Nobody's seeing them on a regular basis. Okay, so my updates. Well, like I said, this has been a challenge year, probably for everyone, but we weren't permitted to work in the Tom Ridge Center starting in about mid-March. So I had to come up with an idea. It wasn't a good idea, of how to grow plants for this project this year. So this is my garage, and I moved all the plant trays, which had of course just started to germinate, into my garage. I think it was it was right after St. Patrick's Day. And I tried to make a little greenhouse in there. So the middle picture, you can see this is when all the trays first came in. I didn't really know what to do with them, and we had to build a growing area with heat mats, with lights, with fans to try to replicate a greenhouse. It didn't work. very well, so we'll talk about that. But you can see that this is my March, April, and May. I would go out every day, multiple times a day, check the conditions of the trays, and then I do document everything very well as far as what's germinating, what's having successes and failures, and then eventually I'll get a % germination, probably by like June, I should know. So, okay, so what I did learn from trying to grow plants in a garage is that the germination was pretty poor. Trays that didn't already germinate in the greenhouse had a really tough time. I had really badly stretched plants because I could not produce the same amount of light that I had in the greenhouse here. And then my plants kept drying out. I was afraid to overwater because then I was worried about getting like fungal and bacterial diseases. So we were in dry conditions most of the time. Trays were either too dry or too wet. I ended up rotating them every couple of days to different locations to see if that would help kind of give me a more uniform growing condition. And I never really was able to achieve that. And then my seed coats didn't shed well. So we had a lot of issues with things germinating, but then dying right after germination. So anyway, and then also we had water everywhere. We had extremely high water conditions. And so a lot of the areas that were scheduled to be planted were not able to, well, we didn't really have the volunteers to bring in either, but even I couldn't get into some of the areas. I'll also mention another challenge that we had with the high water levels. So, okay, smaller area to plant in, not really having volunteers. My plants washed away a lot. And I was really frustrated with that because the high water levels, they weren't just maybe day by day changing or week to week. It was almost hour to hour. You'd go into a site, observe a site, it looked great, you'd plant, and then you'd come back later in the afternoon and things had already been uprooted and started to wash out into the middle of the wetland. So I had to come up with a way to counteract that. Also, things were kept in pots too long. So in my picture on the screen, you can see that everything rooted together, which makes planting a challenge. Even trying to get things out of the pots that was really, really difficult. So I'll show you my methods to keep plants in place. We first tried some fencing, which is snow fencing and silt fencing. That actually worked really, really well. But like Holly said, we have a lot of visitors. We want things to be aesthetically pleasing. We don't want to have fencing everywhere, especially in wetlands. Wetlands are really beautiful, but not with this fence. So we had to come up with something that was a little less hideous. And so I did some bamboo staking, and that worked really well as well. These photos are from a Thompson Circle, and they did keep plants in place. In fact, if you look at the very top plant that looks like sticks and mud, that's actually sediment forming all around the root ball of that plant, which was awesome. And there's a bamboo stake right in the middle of that. So, and then of course we did a lot of restoration field work and plant surveys. So that's just a couple photos of that. This is what a wetland planting looks like. Everything's loaded on a truck. Things are laid out and then planted in the ground. So this is both Thompson Circle and Leo's. And then the Weed Warrior program, again, that's a volunteer program. We had a challenge trying to manage the program ourselves without taking volunteers, but we still removed quite a bit of garlic mustard. That's a picture of Tom Cermak, my co-director, with his poison dart gun. We were shooting Oriental bittersweet. And we really rely on manual removal of invasives, but Tom has an applicator license, so he'll be working with some chemical for us. And then this is a great field opportunity for students. So Please, I have my contact information at the end of the program. Keep this in mind if you want some hands-on field work. You'll definitely get an education with this program. So hopefully we'll be scheduling for next year. I'm not exactly sure what's going to happen. And then my considerations basically are just saying that, we're learning from our successes and failures every year. We have great infrastructure and a great team, and so I couldn't ask for better coworkers and park staff. And then my updates real quick, hardwood cuttings 3560 for this year. Let's see here, a little bit over 50, what is it, 5160 for 2020. And then I did plant about 1,200, a little over 1,200 plants myself this year. Usually we're closer to 2,000, but still overall a good year considering. And just thank you to all our partners involved, Ducks, RSC, DCNR, Bob White, who's like a mentor to me and the person I like to fight with about plants, and of course our volunteers and staff that have helped. And so that's really everything. Any questions?
Hi, Jen. We have a couple comments in the chat. Very impressive work and good job. You've had a lot of obstacles over the last couple years.
And, it keeps the job interesting. So it's, I'm a positive person, so I try to turn it around and say, what can we learn from it? But there were times that it was very trying too. So. Yeah.
Have you altered the plants that you propagate from over the years, from things that were not successful to what is more successful?
Mm-hmm. Absolutely. And that's one, I do keep a grower's log. And if I find that something is either germinating very poorly or not germinating at all, or if we just don't have success when it is planted, if it's maybe slow growing or it has a weak root system, we just take it off the list. And I'm always trying to come up with new things to add that we're finding in these restoration locations. So every year I try to grow a core group of things that do really, really well for us. And then I try to add some new things in just so I can learn from that basically and see if we're going to be able to turn that into something that's viable for the project.
And one more comment, maybe you could consider planting floating marsh islands that are tethered to the benthos, which can then return to the benthos as the lake levels recede. So it sounds like a fun winter project. Get you out of the garage.
I know. And really, any suggestions are welcome, because that is something that I think we're going to be dealing with for the next couple of years. And like I said, you hate to grow plants and then have everything kind of wash away. So anything that can be used to stabilize the plants, I'd be welcome to suggestions.
Okay, well, thank you so much. That was a great presentation.
All right, thank you.
And we will move on to our next presentation. This one is by Amber Stillwell from the Regional Science Consortium on the monitoring surveys in priority wetland areas on Presque Isle State Park. Amber?
Hi, everyone. Oops, that's a sneak peek. Thank you so much, Jeanette. As Jeanette said, my name is Amber Stillwell, and I am the Regional Science Consortium Research Coordinator. Sorry, I'm a bit distracted here. I'm pulling this up. Okay, so I'm going to talk to everyone a little bit about the monitoring that we've been doing in these restoration areas that you have been hearing about. And I'm excited to share some updates with you, as well as 2 new projects towards the end of my presentation that we worked on this summer. So you'll see a lot of really nice photos that I took this summer. This one in the back that you can see is a view of Thompson Circle in the rain, and I thought it was very fitting for some of the field work that we did this year and all of the different environmental parameters that we encountered as we were out there. So before I get into my projects, I want to talk a little bit about how important monitoring is. the success of these projects is truly demonstrated not only by acreage and by plants planted, which are extremely important, but also by the abundance and diversity of the plant and animal species in those ecosystems after they have been restored. So the monitoring aspect of this project allows for multiple metrics to be evaluated, and that increases the potential level of success of this project. So you're going to be hearing from me as well as several other scientists who've been monitoring things like birds and bats and all sorts of things. And they all really contribute to the overall success of this project. And this presentation will mostly focus on some animal diversity and abundance in these priority wetlands that we have been restoring. So the monitoring locations that we have been working in include the neck of the peninsula, Leo's Landing, the marina on Presque Isle State Park, and Thompson's Bay. And those are the primary sites that we've been targeting for fish surveys and amphibian surveys, as well as macroinvertebrate surveys. We also surveyed freshwater mussels this summer, and those survey sites differ a little bit, and I'll explain that why when I get to that part of my presentation. So I'd like to begin with amphibian monitoring, and you can see a photo here of our lab and field coordinator, Sean Dalton, placing a song meter on a tree in Leo's Landing. Song meters are placed at each monitoring location for at least 3 nights per week throughout March and the summer and into September. The audio files that we pull from those evening recordings are then processed using SongScope software, and that enables us to determine the presence or the absence of amphibian species in each of these wetland areas. And just to give you a quick introduction to how we analyze this information, you can see the call profiles of the various amphibians on the bottom, in the bottom photo. The American bullfrog has a very loud signature that is bold and quite extensive, whereas the gray tree frog has more of a trill looking signature where it has all of those parallel lines throughout the frequency. And then you sometimes have situations where you record multiple amphibian species at the same time, like the third photo there that shows a gray tree frog and spring peepers chorusing all together. So due to the fact that the monitoring audio files are so large, we haven't yet been able to look through all of the 2020 monitoring files. So I wanted to give you an update from the 2019 season. And this is best represented in this pie chart because we're looking at presence or absence of species. And this is an average of all of the species that we have found throughout the entire monitoring season of 2019. You can see that the species that we record most frequently are spring peepers and the gray tree frog, but we also get American toads, Fowler's toads, green frogs, and bullfrogs. Unfortunately, we did not capture any northern leopard frogs, although we did visually confirm them at several locations. So in 2020, we were able to start monitoring earlier in the season. We began in early March instead of in early April in hopes of catching those northern leopard frogs on our audio files. And I'll update you on that hopefully next year. So to move on to our fish monitoring, we used a combination of trapping and seining for 2020. The trapping was new to me and to this project, and it was very exciting to be able to do some monitoring in the sites where we cannot use a seine. So for those that are unfamiliar, a seine is a large net that has weights on the bottom that 2 technicians will pull through the water, and then you count the fish that are captured within the net. In some of these wetland areas, there is far too much debris and vegetation to pull a net through the water. So fish trap is much more applicable. So we specifically used a fyke net in these more internal wetland areas. And In fact, on our very first day in June, when we deployed the fish trap, we caught several spotted gar. And you can see our education and outreach coordinator, Sarah Magian, there holding a spotted gar. Her face is covered by a mask, but she has a huge smile on her face. It was a very exciting day. They were congregating in Leo's Landing area to spawn, and we had never before been able to capture them. And in the upper photo, I just want to acknowledge our interns that we had this summer. 2 of them are not pictured here, but our full-time interns, Madison Heater and Joey Forish, were very, very helpful in obtaining all of this data throughout the summer. So the results of our fish monitoring over the last 4 years are very interesting when we consider the species abundance and richness and diversity. So this is a chart showing you the annual fish abundance in by species in each year of our monitoring. So you can see that in some years we recorded that species in all 4 years, maybe in similar quantities, but in other years we only captured a few species or none. For example, we did capture an Iowa darter in 2017, 2018, and 2019, but we did not capture Iowa darters in 2020. So something like this I can attribute to the fact that we used fish trapping a little bit more than fish seining in 2020. So that could indicate that we actually need to do a combination of both of these techniques in order to get more accurate results, because catching darters with a seine is much more likely than catching them in a pike net that's stationary for 24 hours or for 8 hours. However, with other species, we had great success. We caught a lot of yellow perch in every year, with it being a little bit lower in 2020. And we caught a lot of bass and a lot of pumpkin seeds and a lot of bluegill and banded killifish almost every year. So this is our just general species abundance as it varied from year to year. So we can also look at the variation of species richness and diversity over the last 4 years. So in 2017, looking at the species richness, we had 15 observed species. In 2018, we had 1 less, and in 2019 and 2020, we had 16 observed species. The diversity follows a little bit different of a trend. 2017 was our most diverse year, followed by 2018, and then 2020 was our 3rd most diverse year. So this indicates to me that we need to monitor a little bit more frequently. Although we deploy the fish traps at least once per month at each location, and we use the seining, we definitely need to increase that deployment and to ensure that we are getting the best representation of the fish communities. So I'm going to move on to one of the new projects that we did this summer, and that is the freshwater mussel monitoring that we did. And this photo in the background is a photo of Sarah Magian and Sean Dalton, both snorkeling, looking for mussels in Thompson Bay. And the lines that you see in the water are the transect lines that I'm going to be talking about. So let me first introduce you to why we decided to study this this summer. So there are 2 proposed causes of population decline in freshwater mussel species. Freshwater mussels, also known as freshwater unionids, used to be found in high diversity in population all over Presque Isle State Park. It was really known as a habitat reserve almost for these freshwater mussels. So they were found here, but nowhere else. And so a lot of studies occurred since 1909, but they were sporadically occurring throughout the 1900s and into the 1990s, 1993, and then with 2013 being the last time, many of these sites were surveyed. There are a lot of survey gaps between the sites. So a lot of the sites do not overlap their monitoring from study to study. So we had a lot of information to kind of sort through and determine which sites would be the best to monitor. So we tried to kind of stick somewhere in the middle. We chose monitoring locations based on previous studies and whether they were from 2013 or from 1993. And throughout all of these studies, they all consistently showed that sensitive species of freshwater mussels have been declining. And they were found to be, in the 1990 study, less than 1% of the populations identified. In the 2011 study, they were not found at all. But like I said before, the same monitoring sites were not looked at. So there, like I started out saying, there are two proposed causes of this population decline. And one of them is the introduction of the invasive dracenid mussels, or the zebra and quagga mussels. They were first found in the Great Lakes, and they tend to outcompete native freshwater mussels for the same food sources and habitat. The second proposed cause of population decline is due to round and tube-nosed gobies, which were also identified in the Great Lakes in 1989. And they tend to feed on the larva or the glaucidia of the native mussels because they're free floating in the water before they find a fish host. So not really knowing which proposed cause is the correct one was a really large driving factor for us to begin this study. Not only does the presence of freshwater mussels indicate that our restoration is working, but it also will give us a clearer idea of what is happening to the freshwater mussel community based on both the invasive plant species, but also these other invasive aquatic species. So the methods for consisted of combined methods of snorkeling and tactile searching. And we surveyed areas of 100 by 20 foot transects. And what we did was we strung up a 100-foot line and had 2 technicians at a time wearing snorkel gear and scuba diving gloves. And they would have a 25-foot transect and swim out and back until they covered the entire area. We surveyed only 3 sites this summer, and I'll explain a little bit as to why that occurred. But we surveyed 3 sites, and we did 3 surveys throughout the entire summer. So 1 survey at each site. The sites consisted of Perry Monument, Leo's Landing, and Thompson Bay, and that was based on the previous literature. We collected the freshwater unionids or freshwater mussels from locations where they were found. We tagged them and processed them by determining their length and their shell length and their shell width and some other parameters and identifying them. And then we returned them to the water body. So this is a map created to show the freshwater sampling or freshwater mussel sampling that we did. And you can see that from my original map, the sites pretty much line up. But the most important thing is to point out that at Leo's Landing, the bottom pop-out square, it shows 2 separate monitoring sites. And that's because we unfortunately didn't find any freshwater mussels at Leo's Landing at either site. We started with 1 site, and then we decided to go to move the transect to another site when we found nothing, and we found nothing at either transect. We also surveyed at Perry's Monument, like I mentioned before, and at Perry's Monument, we found shell pieces and whole shells, but no live specimens. And at Thompson's Bay, we were very, very lucky. We found some freshwater unionids that were alive, and we were able to tag them and get the information and identify them. So at Thompson Bay, we found several eastern pond mussels, which are not which are not very, they're not endangered, they're not a very like sensitive species, but it was very, very exciting to us to even find any in the first place, because the thought had been that these species had been completely extirpated from Presque Isle. So finding them was a victory for our restoration project. And like I said, at Perry's Monument, we found whole and broken shells. At Leo's Landing, we found no shells and no live specimens. What we did find at all three sites was a high population of round gobies and zebra and quagga mussels. So to wrap up the freshwater mussels monitoring, I just want to say that we need to continue these surveys in the future and determine where we can find other freshwater mussels, because if they're at Thompson's Bay, they have got to be in other places. And we need to look more heavily at the relationship between round gobies and at the invasive mussels, because we're still not sure what that relationship is. So we'll be investigating that more in the future. And then my last project, I know I'm running out of time, I'm sorry about that. My last project this summer that was new was macroinvertebrate monitoring. And this is something that has been done by the Regional Science Consortium before, but not specifically by me. And so I'm very excited to be able to continue this work. We want to look at the water quality at these priority restoration sites. And we use 3 separate techniques of bottle trapping, sediment sampling, and dip netting. We took samples at 4 sites 1 time per month from June to September, and these samples have been preserved for identification over the 2020 and 2021 field season. Excuse me. For those that do not know, macroinvertebrates are an excellent indicator of water quality, and they are the backbone of a lot of the bird and fish species that can be within these wetland areas. So determining their presence and their species abundance and diversity will be incredibly helpful. And these are just the locations where we deployed those bottle traps and did the dip netting and the sediment collection. And I need to thank all of our wonderful project partners: Ducks Unlimited, Go Native Erie, California University of Pennsylvania, Pennsylvania DCNR, Erie Bird Observatory, and the Weed Warriors, and our RSC team, and especially our interns, Joey Forish from Penn State and Madison Heater from Allegheny College, because without them, we definitely would not have been able to accomplish all of these projects this summer. Thank you, and I will take any questions.
Great presentation. Thanks so much, Amber. We have a comment for you. Great work. I'm so proud you know your stuff.
Thank you.
And I think that one of the important parts of this session, this whole wetland restoration project, is the amount of monitoring that we are doing. That really kind of justifies the success of the different wetlands that we are restoring and looking at. Okay.
Thank you.
We're going to move on to the next presentation. Thanks so much, Amber. And our next presentation is by Sarah Sargent from Erie Bird Observatory. And she's going to talk about the Waterbird monitoring at Presque Isle State Park associated with the Wetland Restoration Project. Sarah, go ahead.
Okay, can everybody hear me okay? No. Yes, okay, good. Oh, everybody else is muted, yes, okay. Very good. Yeah, my name's Sarah Sargent, and I've been working on Waterbird monitoring at Presque Isle State Park, or that's what I'm talking about. I too am a partner on the same project that the previous speakers have been talking about. It's funded by Sustain Our Great Lakes through the National Fish and Wildlife Foundation at this point, and overseen and managed by Ducks Unlimited. I have also worked on the marsh birds last year for our presentation at this symposium. I talked about the marsh bird monitoring we're doing. I will not be talking about that today, but we did continue with that this summer. And so that is breeding birds during the breeding season in the summer in the marshes at Presque Isle. But this is the first time I've talked about the waterbird monitoring. And this is a new phase or a new program within the bird monitoring work that we're doing for this project. That started with this latest round of funding. So our first season of conducting this monitoring work was in the early spring of 2019. So that's what I'm going to talk about today, not the marsh breeding bird work or piping plover monitoring, which I think Holly also mentioned, that which we also participate in. Briefly first, Erie Bird Observatory is a new, young organization that started in 2018, and our mission is to foster enthusiasm for birds and conservation through ornithological research and public engagement. And so primarily we do our public engagement through our bird banding station. And unfortunately this year we were not doing any public bird banding, but we also incorporate volunteers into some of the monitoring work, and we did have volunteers participating with this waterbird monitoring work that I'm going to talk about today. So the waterbird monitoring, as I mentioned, it started in early spring of 2019. The season lasted 8 weeks in the spring, and in the fall it lasts 12 weeks, October, November, December. The reason, I think I have, yes. First of all, I'm going to talk about this. What are waterbirds? Many people know waterfowl, and waterfowl are the ducks, geese, and swans. However, we expanded, instead of just talking about waterfowl and monitoring ducks, geese, and swans, we are also including this other set of bird species that are ecologically similar. So they look like ducks, but they don't quack like them, as the saying goes. So that the loons, for example, on the lower right, down here is a picture of a common loon. It kind of looks like a duck. And in fact, hunters occasionally accidentally shoot them. Oops, I'm trying to see my other screen. But they are not ducks. They're unrelated. They're in a different family. They're in a different order of birds, in fact. The loons and grebes are in different orders than the ducks. And so there's these other birds that are similar, and we are including them. They feed in a similar way. They're using resources to some extent overlapping with what the waterfowl are doing, and they're sort of functionally similar. We'll be referring to water birds, and that includes this set of species, anything within these groups that occurs at our sites will get counted if it falls into one of these groups. And the question is, and Amber was just mentioning this too, why do monitoring at all? Particularly birds that are easier to monitor than many of the things that she was talking about. It's hard to monitor some of those less conspicuous organisms, but birds are very conspicuous and they're easy to count. And a lot of people enjoy bird watching, and so you can recruit volunteers to assist. And so birds are a good thing to establish long-term monitoring programs with, because they are conspicuous, easy to count, and people like to go out and count them or watch them at least. And of course, with any monitoring program, you assume that the abundance of your target species is going to be reflecting the productivity and the quality of the habitat. So that particularly if you're using birds, in this case waterbirds, as a monitoring tool for keeping a finger on the pulse of the quality of the habitat that's available, and hopefully with improvements in the habitat, we expect there then to be an uptick in the numbers of birds counted. And so it also is very often you get a lot of insight into the function of the ecosystem by figuring out or observing changes in different species composition, for example. And that's another thing that Amber was mentioning with the different fish species and other things. Same thing with the birds. And so that if the certain types of ducks decline and other types increase, you presume that that means that's reflecting a change in the food sources available to them. You can infer about the functionality of the ecosystem is reflected in those numbers. However, as I will discuss a little bit later, there's not always a direct connection between numbers you count and quality of the habitat, because other things, there are a number of factors that are affecting populations of these species, not just the habitat availability or quality. So yeah, long-term monitoring, the reason to do, so that's monitoring, but then what about long-term monitoring? Long-term monitoring is what our goal is with the bird programs, is to do long-term monitoring, because over time, if you continue to do monitoring over longer periods of time, you get much better tracking of what's going on. Most grant funding programs are rather short in cycles, and to really get at whether something is increasing or decreasing at a global or a large continental scale, you really have to do long-term monitoring, and particularly for things for which their populations can run into the millions. It can be hard to detect changes over a short time, short scale, small scale. And one of the most conspicuous examples of this probably was this recent science paper that came out last October, a year ago now, and that they estimated, based on long-term monitoring of bird populations, that approximately there's been a 29% loss in avian abundance since 1970, which is when some of these long-running bird monitoring programs began. So close to 3 billion birds, that's sort of the tagline that came out. This is what this is the graphic from their paper. Since 1970 through close to almost 2020, there's estimated 3 billion birds lost in these monitored populations. That's why it's important for us to monitor populations, so we know. Otherwise, we just don't know what we're missing, and we can't do anything about it if we don't know anything about it. So our objectives are to establish long-term monitoring program for waterbirds at Presque Isle. The steps we went through with the support from this funding we have is to set up a series of locations and a protocol, and then to enlist and train volunteers, and then go out and collect the surveys and then manage the data. So that's sort of the steps we were going through. Ultimately, of course, the goal would be to link the bird abundance with habitat management actions so that we can hopefully see the results of the habitat management on the bird populations. Well, it'll take a little while to get to that, but we're getting the monitoring in place ultimately to be able to do that. We selected 16 sites at Presque Isle. And they're shown here. I'm trying to get it where I can see both. Okay, so we have 16 sites. The head of the bay, Leo's Landing. This is Swan Cove and the Lily Pond. We have Marina Lake here. We have West Pier. This is the Low Bridge site. This is boat launch 4 looking into the marshes. Misery Bay, Niagara Pond, Horseshoe Bay, North Pier, Beach 11, Thompson's Bay, and then Thompson's Pool. Sunset Point is here. Whoops, Beach 10, Sunset Point. This was last year. We've now established another walking transect that we do along Sidewalk Trail. What you end up having to do is count birds in flight after you flush them from along here, but it gets more into this interior area, which is at least adjacent to some of the habitat treatment areas. And then we are also now including a goal point segment as well. So those are our sites that we're working at. And for the volunteers and also our staff, we have locations that we go to and for each site, and scanning. You're supposed to do a complete count of any waterbirds seen within this area from these viewpoints. So we have these sites. The protocol essentially is just go out there and look at everything that's you can see, count up each species and record them, and record the amount of time you spent doing it. So just for example, the Leo's Landing, here's a similar diagram for Leo's Landing. In some of our sites do face outward into the bay and or the lake. And so we have tried to say, to delimit an area, we want people to only count the ducks within a particular area. It is a little hard to do that on open water, but we're trying to give people guidelines, and especially our staff who does count a lot of counts, to have guidelines on exactly how far out to do the counting. The Marinas Lake, again, is one where we have a bunch of different locations we look into it from. So we're getting a pretty complete count of whatever is in there. But Launch 4 is a really cool one because it is one of the sites that has most interior marshes. And I will say a lot of this vegetation that's out here in the months that we are doing the survey work, a lot of that vegetation has died back and isn't there because it's a lot of emergent spatter duck and some stuff like that. So this is actually, there's a bigger view here now than suggested by this picture. This is what we call Thompson's Pool, Thompson Circle Wetland. I guess I heard several different references to it with the other monitoring work and the vegetation restoration. It is a site that we do bird surveys at because I know it's of particular interest. And it's very visible from the road along here. So it's a nice one. We also get back in here and look in from there. So this is a repeat of this slide. The spring, we did the 8 weeks of last year, October through December, we did the 12 weeks. As with other people, this year got cut short. We were interrupted by COVID. We did 3 weeks of March of spring of 2020, but we stopped after that, and we asked our volunteers not to go out. We are conducting season this fall, and but we haven't done as much of a volunteer push as we did last year, but we are recruiting hunters to observe from their hunting blinds. And so we hopefully will get a lot of duck counts contributed by the hunting community from where their blinds are. And we have, of course, information on which blinds, where the blinds actually are. They don't correspond one-to-one with our counting sites, but we are covering our count locations ourselves. So just to get into a little bit of results, we've had 38 species. Some of them are relatively uncommon. We only get, there we go. We don't get that many of them. So they don't contribute much towards the total waterbird use days. Waterbird use days is something that waterfowl biologists use, sort of a cumulative use of a particular patch of habitat over time or during a season. So if you had 100 ducks there for one day, that's 100 waterbird use days or duck use days, often they count. So what that gives us is a measure of, we can count up across species and over time for a whole season and say this, Thompson Circle wetland got X number of waterbird use days that we observed, we estimated, because we can also interpolate between counts if we don't count every day. So we can calculate how much the habitat is supporting ducks. We can compare different sites. And then over time, of course, we'll be able to compare 1 year to the next year. So that's our tool that we're using is our waterbird use days. And again, it's sort of, we interpolate, if we count one day and there's 100, the next day there's only, 2 days later, there's 50. That means we estimate the in-between day as having 75. We would add up across those 3 days and get a total cumulative waterbird use days, some number. So just to give you an initial taste of some of our results, the species codes across the bottom are these 4-letter codes that bird people like to use. And here's total waterbird use, or total number of WUDs, waterbird use days. And this is a comparison of cross species between the spring and fall seasons. And so this is not a timeline, this is just across different species. You can see the spring numbers in blue for particular sets of species are definitely higher than the fall numbers. So canvasback is this one, redheads, ringneck ducks, and the scaup. Greater and lesser scaup are very, very similar in appearance. You can separate them if you're close enough, but in many cases with at Presque Isle, looking out into the bay in particular, if they're further away, you cannot tell the difference at a distance, so we combine them into a scaup species. And that tends to be a lot of what we're counting. So there's a set of species here that are primarily diving ducks that are definitely more abundant in spring than in fall. Now, there's a couple factors going on here, though. We have a fall hunting season that occurs. And so many ducks are less apparent and less likely to be counted in the fall because they are shying away from the areas where the hunters are. And this also has affected our participation by volunteers, and that they are, we have fewer volunteers in the fall than in the spring. I'm losing track of how much time I have. Okay, just a taste of some of the other results. We can also get the pattern through the season. This is from spring of, thank you. This is from spring of 2019, and you can see we didn't count on weekends. There's little gaps here, but you can definitely see that this third week of March is really peak season for the birds. Same thing happened this year, except we don't have the whole tail end. We only went up through about the third week. And then here's our fall seasons, daily water use, waterbird use days, with and without gulls. If you looked at that slide 2 slides ago, the gulls, we have a lot of gulls, and we have more gulls in the fall than in the spring, and they tend to add up to a lot of waterbird use days. And so we wanted to separate out the gulls, and that's what this shows. Again, this reflects the interference from hunting. Okay, so just to start wrapping it up, response to management, it's way too soon for us to be able to make any kind of conclusions about duck or waterbird numbers being responding to management actions at this point. We don't actually have good comparable before numbers from prior to the management work that's been done in these marshes. I will say the visibility into those interior marshes is way better than it used to be because the frag made it virtually impossible to see. And the diversity of the plant structure is also really helpful, I think, for a lot of species. It gives them a lot more habitat if you get things broken up into smaller patches, and a lot of interstices of patchiness is very helpful to the waterfowl and other birds as well. Now, I just wanted to mention that although we make the assumption that the habitat quality is going to be reflected in the bird numbers, there's not a direct connection. There are other factors. Hunting in the fall certainly is driving ducks away. They're not using the habitats, the nearshore habitats that are exposed to hunting in some of those more sheltered locations close to shore, which they would normally be using. They're just not using them because the hunters are there. And then I'll also say that in the early spring, in years when we get ice cover, but on the Bayside, we've been having ice cover no matter what, at least to some extent. Ice cover definitely affects the distribution of the ducks and whether or not they're using the spots that we have as our count sites. I'd like to say, a big thank you to our volunteers. They've submitted a lot of checklists, a lot of hours. Last year, I haven't summarized, we did have some volunteers this spring and we do have some volunteers this fall, but not very many. And but like I said, we're trying to recruit the hunters, have them contribute. It's a little too early to count those up, but I want to thank all the volunteers who have helped did help in 2019. And also want to acknowledge all the partners that you've been hearing about, Jim with Ducks Unlimited, the Regional Science Consortium, and all their people. We also had really good support from that Northwestern Pennsylvania Duck Hunters Association, both last fall and this fall, with trying to get their membership involved in helping to support this habitat work. And then State Park and the funding program, and then Conavera, who supplied lots of these photos, including this nice picture of a grebe. And that's it, I think. Thank you. And I am available for questions if there's any time left. Thanks.
Hi, Sarah. We have a quick question for you. Where do the ducks go the fall that they get chased away by hunters? Do they leave the area and not come back?
No, I don't think they're not really leaving the area, but they are definitely hiding. We don't know. We don't have radio trackers on them. Maybe someday we can figure that out exactly where they go. But they are very cautious about being in areas where they can be hunted, and they are moving around the landscape at bigger scales. But then they are also finding locations that don't have as much hunting. So, for example, Gull Point or the interior marshes, or for the diving ducks that are really good at being out in open water, they're going offshore into the lake.
Okay, well, thank you very much, Sarah. That was very informative. That was a great presentation. Thank you.
All right, thank you.
And we will move on to our next presenter, Kelly Pierce. And she is with Allegheny College. She will be talking about the summer bat communities in Presque Isle State Park. Kelly, you can go ahead and start sharing your screen.
Okay, welcome everybody to my talk today. Like Jeanette mentioned, my name is Kelly Pierce. I'm an assistant professor at Allegheny College in the Environmental Science and Sustainability Department. Today, I'm going to be presenting on work that we completed this summer on summer bat community composition at Presque Isle State Park. And I'd like to also just thank my co-authors on this, Sarah Till from Gannon University, Katie Brozel, Bailey Kozala, and Matt Lazeski, who helped collect this research this summer. And so just a little bit on historic knowledge on bat communities at Presque Isle. It's really an understudied area in terms of bat communities. There was some information conducted by Anderson from Gannon University using acoustic detectors and mortality counts. This was related to the wind turbine that's located at Tom Ridge Environmental Center. And so the goal of this was just to determine if there was an impact on bats in this area due to that wind turbine. And they found that it really didn't impact bats, but they found a decent number of bat species located through those acoustic monitoring. Terry Lebdell also is a local bat person. He has done opportunistic monitoring of roost locations at various places on Presque Isle. A lot with those species that hibernate or go into torpor in the winter time. A few other studies that have looked at distribution and migratory bats, particularly the silver-haired bats, occurred almost 10 years ago in Long Point, Ontario. Those are really looking at migratory bats like the silver-haired bat, for example, and not necessarily the bats that are inhabiting Presque Isle throughout the year. So there are a variety of likely summer residents at Presque Isle State Park. These are bats that live here in the summertime, including the little brown bat, the northern long-eared bat, the big brown bat, and the tricolored bat. Those are year-long residents, but we also have 3 summer residents likely at Presque Isle State Park, including the hoary bat, the silver-haired bat, and the red bat. And so these species migrate south starting, about a month ago, early October, mid-October. They start to migrate south, but they do inhabit this area in the summertime as well. And again, those migratory bats are indicated by the red stars. The big brown bat is the most common species. They are also the species that has been least impacted by white nose syndrome. Researchers aren't exactly sure why, but maybe because they're a little bit larger, so they have a little bit more energy that they can expend. But other species, the Myotis species in particular, have been really hard hit by white nose syndrome. So this study took place at 3 locations on Presque Isle. Our most southern site was Leo's Landing. Our site there in the middle of the isle is Old Gaswell Trail, pretty close to the marina. And then we also did some sampling towards the end of the summer at Thompson's Circle. Really what we're looking for when we're doing the bat sampling is an area that is clear for the path, for the bat to fly through. We're trying to catch the bat while they're feeding at night. And really what we're looking for is an open flyway with canopy cover over top and then trees that line the sides as well. Again, we really want to pick areas that kind of funnel the bats into these feeding areas. Near water is a benefit. Many of these bats are flying at night to feed on insects, and insects are really drawn towards these water areas. And so you can see our site there at Leo's Landing. We have our net set up there on that top left picture, and then we're setting the net up there on that bottom left. That's our site at Thompson's Circle. So this summer we were able to get out for 10 sample days. We initially had planned on more, but our summer funding was held up because of COVID. And so we did the best that we could with what we had available. And so we had 10 sample days between June 16th and August 1st of 2020. And our sample days, or our sampling method, is essentially setting the net up prior to sunset. And so once the sun sets, we raise the net, and typically we stay out 3 to 4 hours after the net is raised. And so usually our sampling times start around 9:30 p.m. and end around 12:30 a.m. or 1:30 a.m. depending on our success and temperature as well. Bats tend to not fly when it's colder outside, and so we tried to pick nights that were warmer, and additionally pick nights that had really low precipitation chances, because they really don't fly when there is precipitation. And so you can see the setup. We use what are called triple high mist nets. And so these are essentially 3 nets that are put together. Each one of these we call a shelf. They're about 3 meters tall. So overall, we're about 9 meters in height. And this also enables us to get higher up in the canopy, but can also limit the sites where we're able to set up this mist net. And so it's a really fine netting system. Doesn't harm the bats when they're able to when they fly in to the net. A few of the tools and the measurements that we take when we capture a bat. We of course use protective equipment. Our Tyvek suits are pretty much have been standard since white nose these to ensure that we're not carrying white nose from one location to another. This summer we also, as I handled the bats, used an N95 mask. There was some, concern about humans transferring the coronavirus to bats themselves. And so any work that was conducted with bats had to follow protocols that were established, which included using N95 masks. And so that protective equipment was used. Also, what I call extraction tools. I use actual batting gloves. And so these bats, they eat insects. They eat insects with these hard exoskeletons. And so they can do a lot of damage to your fingers as you're handling them. And so I use batting gloves and latex gloves over that, which I of course, change in between bats. We take the right forearm measurement of the bats. This helps to, if there's any question between bat species, forearm measurements and weight usually help to eliminate possibilities. And so we use a measuring tool to get the right forearm length of the bat. The weight of the bat, again, another way to differentiate between species. We use a UV light that helps to illuminate white nose spores. So white nose spores, that fungus that lives on the bat, illuminates in an orange color if those spores are present. And so we use the UV light to detect white nose. And also a headlamp not pictured helps to look for wing score. And so for wing score, what we're looking at is there any damage that indicates that during the torpor period that the bats were itching at their wings, and so there we're looking for discoloration and scarring, using the headlamp as a tool for that. So our results, we captured 15 bats throughout the season. So overall, effort-wise, it was about 1.5 bats per night. The old gas well trail was the only site that we captured bats in. And so our efforts there was about 3.75 bats per night. We captured primarily big brown bats, 14 of them. We captured one little brown bat as well, but it escaped before we were able to take any measurements or even sex the bat. But we did capture one little brown bat as well. Our big brown bat, in terms of the demographics, they were all adults. So which isn't that surprising given our time frame of the trapping. They were all males as well. And I'll get into the discussion of why potentially they were all males. On average, the forearm length was about 4.4 centimeters and the weight was about 21 grams. Big brown bats on kind of span from about 12 grams to 25 grams. So we were catching relatively larger big brown bats. All the bats that we captured appeared healthy. They were all determined to have a wing score of 0, except one bat that we determined had a wing score of 1. It had some light damage and depigmentation. And so you can see that picture. This was not taken this past summer, but previous bat research. You can see kind of the method that we use to determine the wing score, where we shine that headlamp through the wing, again, looking for scarring and depigmentation, which is an indication that over the winter periods, those bats are itching themselves and tearing up their wings. We also didn't detect any white nose spores on any of the bats that we captured. We did incidentally capture 4 southern flying squirrels. So these are these nocturnal rodent species that kind of glide through the air. They do a lot of damage to nets. And so we were lucky to get them out pretty quickly, but we did, those are always fun to capture as well and see close up. So a little bit about why I think that we captured primarily big brown bats. Number one, they're the most common species in Pennsylvania. And again, the big brown bats there are pictured on the right side. They're also early flyers. They come out early on in the night, really soon after sunset. And so it may be that we didn't sample quite late into the night, maybe extending the samples 5 hours. We could increase the potential to get other species of bats. But big brown bats are also generalists. They roost in a lot of locations, and particularly the males roost together in the summertime. And it might be that that old pond or the gas well trail is quite near a bachelor pad of these big brown bats. The little brown bats, they are a species that has greatly declined due to white nose syndrome, and they really prefer really hot, dark retreats like barns. And so that could be why we didn't capture more of those species, why those species were represented in our samples. We detected a few other species though, which was kind of surprising. It could be due to our setup locations. Leo's Landing, I didn't realize how breezy it was until I was out there at 10 and 11 o'clock at night. And so in the future, I don't think I'll sample there again. It's just a little bit too breezy for the bats to fly around. Other bat species like the tricolored bats really prefer rocky cliff areas, so it might be unlikely that they inhabit those northern parts of Presque Isle State Park. Hoary bats really prefer carnivore species, so it might be just not available habitat to have a diverse group of bats. And also we were ultimately limited in our sample days, so having more sample days, of course, might increase the probability of getting a greater variety of bat species. So if this study continues in 2021, I would like to add additional study sites. Through this past summer, I learned of a couple additional suitable areas for mist netting, including the Dead Pond Trail possibly and near the Big Pond. Also, again, close to those wetland restoration projects. But I'm interested to hear if others have additional ideas or other possible sample sites as well. Also, I'm interested in using acoustic detectors to set out during our mist netting as a complementary sample device, although I would be interested in having a summer research student analyze that type of data, because as Sean and those know who do acoustic sampling, it is intensive on the back end. So I'm interested to possibly add that as well to the project. So I would just like to thank the Presque Isle Wetland Restoration Project, including the funding sources such as Ducks Unlimited for the equipment. Also, thanks to Amber Stillwell for just being a great help in organizing and ordering all the equipment. Because of the COVID regulations, each night she set out my equipment outside, I picked it up, I set it back out for her. And so there was a lot of coordination on each sample night. Also, the Pennsylvania Game Commission for the permitting process, and the PA Department of Conservation and Natural Resources, specifically Holly Best. She helped me arrange or informed the rangers every time we would be out at night. And also the rangers that stopped by to check in on us at 1 a.m. and 2 a.m. while we were leaving the park. They were all really nice and interested in the project. And then just thanks to the volunteers that also helped. This project was really fueled all by volunteers, volunteering their time and their efforts. And so I just really want to say thank you and appreciate that. And so with that, I'll take any questions.
Thanks, Kelly. That was a great presentation. And we're so happy to have you as part of the wetland restoration group and doing the monitoring. Thank you. One quick question for you. So you mentioned that certain species of bats favor specific habitats. So when sampling, would you only expect to see one species at a certain location, or would you expect to see a diverse number of species?
Yeah, that's a great question. So typically you still get 3 to 4 different species a night when you're mist netting, even though you're just in one location, assuming that that habitat is available, you know, within, I would say, a half of a mile or a mile. Bats do fly long distances at night to feed, and so they will traverse a variety of habitats in order to find food. And so in my experience, even though we sample in one location, we do typically get a variety of species.
Okay, great. Well, thank you so much for the presentation. Okay, we are going to move on to our next presenter. Sean Dalton with the Regional Science Consortium is going to be discussing the aerial drone surveys of shoreline change and primary wetland habitats on Presque Isle State Park in Erie, Pennsylvania. Sean, I'm going to turn it over to you.
Is that any better? All right. So my name is Sean Dalton. I am the lab and field coordinator, as well as GIS specialist for the Regional Science Consortium. Today, I'm going to talk about some aerial drone surveys of shoreline change and primary wetland habitats on Presque Isle State Park that we did this summer. This ended up being quite an adventure of a project with COVID and things that happened this summer. But we got a lot done, and I'm really excited to talk about it because it was really just a fun experience. Before I get into anything with the drone, though, I am going to give everyone a brief natural history lesson of Presque Isle. I promise I'll keep it really brief. I'm only going back 23,000 years. So with that, Presque Isle and Lake Erie as a whole, this whole region of northwestern Pennsylvania, was shaped by the Wisconsin glacial episode in the late Wisconsin Age, which was about 23,000 years ago. It's only named Wisconsin Age because that's where the geologists studied the rocks where they learned about this point of Earth's history. So the southern lobe of that glacier carved out the Great Lakes. The whole ice sheet as a whole was called the Laurentide Ice Sheet. Specifically, the Lake Erie lobe carved out Lake Erie, very aptly named. And so one thing to keep in mind, when a glacier pushes down from its original snowfield, it'll pick everything up that's in its way. Nothing really erodes in the same way that ice does. But then also uniquely to ice, once it's done, it just drops everything right where it was holding it. So if you look here, I have a figure of the Laurentide ice sheet, and you can see up in the top left there, about 21,400 years ago was when that ice sheet reached its max extent. And so you can see, it's kind of hard to see on here, but it went farther south than Erie, actually around Moraine State Park is where the edge of the glacier was named because of the glacial moraine, which is at the end of the glacier. But like I said, these glaciers will pick up anything and everything that are in their path. So if you're ever in the county, up on tops of hills, particularly down in Crawford County, you might come across giant boulders that are kind of out of place, not part of any other formation that you see around the area, and they're totally unrelated to everything. This is most likely a piece of Canada that was dropped so lovingly by the ice sheet all those many years ago, and it's still just sitting there. And so these are referred to as glacial erratics. They're just a fun little thing I like to point out. So that brings us to Presque Isle's geology. Just like the glacial erratic, Presque Isle was dropped unceremoniously out of this glacier as it retreated back northward. So Presque Isle is composed entirely of unconsolidated glacial sediment. And what that means is that it's quite literally a big sand pile. There is no Presque Isle formation. There's no underlying bedrock that gives the peninsula its shape. It has totally been formed and shaped by the lake itself. So the sediment, like I said, was just left in a ridge along the southern edge of the escarpment that would fill as the glacier melted to create Lake Erie. And it's just been pushed around ever since. So the original area that it was deposited is about 30 to 50 feet below the current lake level. That figure's a little old. It's probably a little bit deeper than that because we've had high water levels the past few years, but I'll talk about that later. So if you look at this figure here on the left, this is from the Army Corps of Engineers, and these are historical illustrations of how the peninsula's landforms have changed over time. So you can see several times throughout its history, it's actually breached and been an island. And then around 1926, you can see there it's closer to what we know it as now. And in that final figure, you can actually see it's estimated that around 1200, the neck of the peninsula was about where Walnut Creek enters into the lake now. And so the reason that the peninsula has been migrating, so to speak, along the shoreline is because of a concept known as longshore drift. So if you've ever spent any time on Lake Erie, you may know that most of our wind currents are most prevalently coming out of the northwest. So when you constantly have wind coming out of that direction, the waves are going to follow the wind. So the waves then will wash up on shore at an angle. However, because of gravity, the water will always take the path of least resistance back towards its main body and follows a straight line back down the slope of that beach. So if you were a little sand grain floating along in the water here, getting pushed by the current, and you got thrown up on Presque Isle shore, you would then wash straight back down into the lake, and the process would continue as those waves hit the shore at an angle. And so that's what leads to this gradual progression of the peninsula down the shoreline. All right, so enough natural history. Let's go on to the drone a little bit. So this is a picture of our drone. We've lovingly named him Desmond. It is a DJI Phantom 4 Pro V2. This is a very common drone that you can find in the civilian market. It comes stock fitted with a 60 frames per second 4K camera, as well as we purchased a secondary mount for it for a Mapir OCN Survey 3 camera. This is a secondary sensor that looks at specific wavelengths of light that I'll talk about a little bit later. But as far as the drone's operation goes, it's pretty simple. It comes right out of the box. You charge the batteries up. The propellers just clip right on there. It hooks into your iPhone and you can take right off. Now, drones have really been flooding the civilian market a lot lately, and what a lot of people don't know is that if you're going to use a drone for any sort of, well, any work that you can be compensated in any way, shape, or form, even if you're just putting videos on YouTube, you have to actually have a license for it. And there's all sorts of stipulations and things that you have to learn in order to do it. So particularly in Pennsylvania, when you're on state park land, the pilot in command of the drone has to hold an FAA Part 10A certification. And what this is you go and you take an 8-hour course that has a written exam 2 weeks later, and it goes over all the rules and regulations that you do, how to interact with air traffic control, how to input your flight plans so that you don't crash into an airplane accidentally, and other stipulations such as when you're operating, nobody that's outside of your crew can you fly the drone over top of, which can be a little bit of an issue when you're trying to look at things such as beaches. Also, since we're on the shoreline, you can't exceed certain wind speeds, because if your drone can't get back to you, that's kind of a problem for you and whoever unfortunately gets hit by it when it comes back down to earth, as well as you have to keep line of sight on it and airspace regulations. So I went at the end of February to take this course. It was a pretty straightforward course, an 8-hour day. And they said, all right, so in 2 weeks, you'll come back and you'll take your test and you'll get your license and you'll be all good to fly your drone this summer. And so that test date was originally scheduled for March 18th. And if anybody else feels that date's a little familiar, that is because that is the day that everything in Erie shut down due to COVID. So that day I was already, studied up to go take my test, and I got an email that I'd gotten my testing refunded. Turns out that FAA test centers are deemed non-essential and they weren't going to be doing that testing throughout COVID. So I didn't exactly have an end date that they gave me for when I might be able to take my test either. So that kind of started a gray period of perpetual studying and not really knowing when I was going to be taking the exam. But that eventually came to an end on, I believe it was July 27th. And so before this, you could think we were planning March 18th. We had set drone dates set for every month, and it was all planned. It was going to be great. And now we were at the end of July. We hadn't done anything yet, but hey, I got my license, so that was good. So then we had to prioritize our targets. And surprisingly, we got an amazing amount of work done for the little amount of time that we packed it into. Shout out to Amber Stillwell and Sarah Megan. I know you guys love waking up at 5:30 every morning to hear my weather report, whether or not we're going out. So if you can kind of see here on this map, along this shoreline here is the entirety of where we were able to drone throughout the summer, as well as we got one of our wetland restoration sites here at Thompson Circle. So another thing that I'll mention about the peninsula is you may or may not be aware of this, but we're in very close proximity to the Erie International Airport. International airports have pretty tight restrictions on their airspace, and it actually extends all the way until about this point here on the park, just past these two square waterworks ponds. So if you want to operate a drone within this area, there's an altitude regulation of 50 feet, as well as you have to call air traffic control beforehand to let them know that you are going to be there. That's not a big issue. However, operating at 50 feet when you're looking at wetland areas, particularly those that are skirted by trees, you start to run into some collision issues. So we decided to prioritize first areas farther away from that protected airspace. The other thing to mention is, so we wanted to look at erosion as well as our wetland habitats. And depending on the time of year, depends on, what the plants are going to look like. In late July, the plants are, they're still photosynthesizing and producing, but they're not at their full health that they would be normally earlier in the season. So if you want to look at something like their infrared reflectance or things like that, they can already start to be showing signs of stress that wouldn't be visible to the naked eye per se, and you might misconstrue as stress from an herbicide application or something like that. So instead of prioritizing the wetland sites first and possibly getting confused by any results that we got there, we started looking at the shoreline first. Specifically on the shoreline, we were looking at areas that we've previously sampled for tombolo buildup, as well as other erosion-heavy locations along the shoreline, with the ultimate goal being to document the entire shoreline. So here's a photo of us out on the beach bright and early. Like I mentioned before, one of the stipulations when operating under FAA Part 107 is you cannot operate over people that are not directly involved with your operation. As you can imagine, Presque Isle State Park beaches in the summer are a very popular place that people like to be. So we really had to schedule to be there as the sun was coming up, because if you're not, then there's people on the beaches, and there's no way you're going to be able to manage 50 people on the beach and tell them like, oh, I want to fly my drone. Like, you know, can you go somewhere else? So it worked out that we were able to go out there super early, just as the sun got up, and we were able to get a lot of those flights done. This picture over here to the right, you can see is another area just by, I believe, the Beach 7 Waterworks area. This is a pedestrian bike path and walkway. And the past few years, we've seen pretty heavy erosion along the lakeside of Presque Isle for a number of reasons that are currently hypothesized. For one, we've had very high water levels the past few years, as well as we've had pretty minimal ice cover the past few winters, which, believe it or not, the ice actually does act somewhat as a shoreline armor for when we have that increased precipitation during those winter months. If that's not there to protect from the wave action, those waves just continue all winter and will eat away at the shoreline. These tombolos are another feature that we were looking at that form on the beaches. These happen as a side effect of the breakwaters attenuating the waves. So when the waves come into those openings in between the breakwaters, that area that's in the middle there kind of becomes a slack water. And so the sediment that's held in those areas starts to drop out and actually build out a land bridge. So here's a photo of the wetland habitats as well. This is the Thompson Circle area. I'll show this a before and after of the satellite imagery that I compared this to later, but I will note that the resolution of the camera is great. We get about 1.7 centimeters per pixel. So if you zoom in on this photo, Amber and I actually are in it. I won't tell you where, but you could zoom in and you can see right where we're standing. It's that clear. So here's some of our results of the drone flights. So this is the Beach 9 area, or Pine Tree Beach. And so if you look here on the bottom, this is a 2012 satellite image. This is the most recent satellite image I was able to get of this beach face. And at the top here, this was just at the start of August. We were able to go out and get a flight of the Beach 9 area. So just if you look at the two differences, you can see just how greatly the shoreline has changed. And one thing that I do want to point out about this is you can see that this tombolo here is grown and connected all the way to this breakwater here. That might not seem like a huge issue, but what actually happens is that this will now start acting as a groin almost. If you're not familiar with a groin, it's an anti-erosion measure that's constructed generally. It looks like a stack of rocks just extending straight out into the lake, sorry. And what happens is the idea behind this is that it cuts off that longshore sediment drift that I spoke about earlier. So as the sediment is being pushed downshore, it will start to build up in these tombolos, and effectively it will cut this down current portion of beach totally off from a sediment source. So you can see that in this photo, how we've got a lot of sand on this upcurrent side of it that's building up, but you can start to see how the lake is starting to erode on this beach, and really on this side, it's starting to get almost to the dune line. So at the park, they'll go in and they'll dig those up with an excavator, and then that's put back into sand replenishment sand. So this is another area that there's a lot that can be said about this picture. So at the bottom here, this is of the beaches just east of Beach 10 out towards Gull Point. The bottom here is the 2012 image, and the top here is the 2020. So the most striking thing about this I can point out is that you can see the difference in how far the shoreline has receded. I measured this within GIS and it's about 70 meters. So it's a pretty substantial loss of shoreline to the south. However, I don't want to like go and say that it's all doom and gloom and the peninsula is eroding away, because the peninsula's always been doing that. And so it's gradually migrating. So this sand isn't gone. I would hypothesize most likely that it's collecting over here at this down current end of Gull Point. This is also the farthest that I've gotten down the shoreline so far with the drone. This was just, I think, Friday of last week, actually, that we went out and we got this imagery. So as we move further down, I predict that the whole end of Gull Point is going to look totally different than what the 2012 satellite imagery is suggesting it. Some other things I'd like to point out about this photo is that this was a previous breach point that we had measured. The lake did breach through at that point into Thompson's Bay. I think at our shortest measurement, we had 6 feet of sand there. So you can see that's considerably extended now. So it's interesting that the shoreline has been pushed farther south, yet there seems to be an accumulation of more sand. You can also see here the change in the 2012. A lot of this gray shoreline area is actually old Phragmites that had been sprayed as part of this same restoration project. So you can see here in the difference where the mosaic and cuts over the satellite imagery, just how much more open water is there because that Phragmite has been controlled. So this is our Thompson Circle wetland area. You might be looking at this thinking like, oh, well, the landforms kind of look the same. There's not really a lot of erosion there. That's because there's not. That wasn't really our real goal with looking at this site. That secondary sensor that I mentioned that is on the drone as well looks at specific wavelengths of light for looking at plant health, actually. And so the way of doing that is through what's called an NDVI, or a normalized difference vegetation index. What this is an indicator that you can put remotely sensed data such as satellite imagery or drone data into, and it will assess whether or not it contains green vegetation and moreover how healthy that vegetation is. So due to photosynthesis, plants absorb a lot of visible light, but they reflect a lot of near-infrared light. So if you use a near-infrared sensor coupled with a visible light sensor, plants have a very, very distinct signature that you can pick out from that. And then using the values that are coded in that from the drone, you can then create an index that shows you approximate plant health. So this could be used within those restoration areas to perhaps when the vegetation at the start of the season is very healthy, we can go out and we can do a flyover and see how everything is. Then DCNR might go in and do a spray treatment, and then we could do another flyover and actually see, even if the plants themselves to your eye look totally green and still normal, with this camera, they might look red and just totally different from everything else. So you can more accurately apply your treatments, as well as you can go back and have super accurate touch-up spot treatments if there's any areas within stands and things like that that you missed. So these indices are normally performed using an RGN sensor, which is red, green, and near infrared. However, for our drone, we got a new sensor that's kind of an improvement on the original red, green, near infrared setup, in that it's orange, cyan, and near infrared. So if you look at this graph here, this is the normal wavelength distribution that an RGN sensor would have, and so this is the OCN down below. You can see everything shifted a little bit lower in wavelengths, as well as things are made a little bit wider. And I know this graph doesn't really make a lot of sense, but if you look in the imagery that it spits out, so at the top here you have your traditional visible light spectrum. This is what the indices are normally performed with using the RGN sensor. And so you can see in this lower OCN sensor just how much more contrast and detail there is. It cuts out a lot of the soil noise and yellow areas, which will be very useful in our wetland areas because there's quite a mix of soil and healthy vegetation. So the image processing for the drone is super straightforward. We use a program called Maps Made Easy, which is an application for iOS. I download it right on my iPhone, and it literally brings up a satellite map, and I can use my finger to click and draw a map, and it puts in a programmed flight path for the drone, and I literally press upload to drone, and the drone takes off and goes, and it does it. All those images are collected and stored on an SD card that's on the drone itself. Once it's done with its mission, it flies back, lands, and I can take that SD card, and there's an accompanying software in browser from the company that everything uploads. It's specifically tailored for a DJI drone, and the imagery is auto-mosaicked for us. In our experience, it says it can take up to 48 hours, but every time that we've processed it, we've had our results after lunch from that morning that we've processed. So it's a very quick turnaround, and it's very simple to use. That being said, we've had a lot of difficulties. Weather can be very unpredictable, particularly in Erie, Pennsylvania. Along the lakeshore. Sometimes what the airport's reporting is not always indicative of what it looks like when you're down on the beach. Also, like I said, the inability to operate over people unaffiliated with the mission, it's a little tricky. We just got to get out there really early when we're going for the beaches. Battery time is another concern. The drone for flight time, depending on conditions, can go anywhere from 30 minutes to 40 minutes or so. We have 3 batteries. So we're restricted to probably about a 2-hour time period in the morning that we're able to do droning. But that hasn't really been a problem for us yet. Wind speed, like I mentioned before, the drone weighs less than 4 pounds. So a stiff breeze will quite literally send it flying. And the controlled airspace of the park. Time of year, like I mentioned, the leaf on, leaf off vegetation stress. Next year, I really want to get out there earlier and get some leaf on stuff before any spraying or anything's done, so we get a nice, healthy pre-season picture of what it looks like for when we go out and we get another follow-up pass after treatments are done. As well as COVID-19, like I mentioned, it kind of delayed that test for a considerable period of time, but we passed and we're all good now. So the future plans for this are to build a up-to-date database of shoreline erosion conditions, which we can then draw comparisons with. We have our RSC buoy system that's in the lake for the entire boating season. So any changes that we notice along the shoreline, we can compare with changes that we may see in the buoy system, maybe tease out any trends from that data. As well as the nearest satellite imagery I could get was 2012. So the ability to go out there and be able to get an aerial pass of a site in minutes is it's pretty awesome. As well as those NDVI analyses of the Ducks Unlimited habitat restoration areas for that more accurate application and assessment of the treatment. Also possible bird monitoring is one of the things that we may do. I'm currently in talks with Sarah Sargent about, in the next coming weeks, actually doing flyovers of some of the interior lagoons to do duck counts. So that will be an interesting thing that we'll see how that plays out. We can use it to choose new sample sites that we can't get to if we don't want to trudge through the woods or any neck deep water. And like I said, just the ability to have an aerial photograph of your sample site the afternoon that you took it is just amazingly powerful. And when you compare it to how much satellite imagery costs, when previously you had to call up NASA or someone to get a Landsat pass of your sample site, it's just a very powerful tool to now that we have access to. So with that, I'd like to acknowledge all of our project partners: Ducks Unlimited, Pennsylvania DCNR, Go Native Erie, California University of Pennsylvania, as well as Erie Bird Observatory. And I'll take any questions that you may have.
Sean, that was a great presentation. Thank you so much. Real quick, we have a question. Will the drone imagery data obtained this year be publicly available?
Yes, it actually is. So all of the imagery that we collect on that Maps Made Easy site is publicly available as well. So you can actually go on there and look us up and you can see what we've done. That being said, it is available only for a limited time. But if you're interested in seeing the data that we have, feel free to reach out to me, and they're GeoTIFFs. They can very easily be shared, and yeah, so absolutely.
Great, thank you so much. That was a great presentation.
Thanks.
And so we will be transitioning to the next session. We're just going to take a 30-second break and we will be back. And thank you again for joining us. Stay tuned.
Hi Hassan, how are you doing?
Well, thank you. How about you?
I'm doing well also. Welcome everyone to our last session of the evening. My name is Amber Stillwell and I'm the research coordinator of the Regional Science Consortium. For those watching out there, we are running about 7 minutes behind, so just stay tuned, and if you are waiting to watch someone, just jump on and make sure you stay on because they will be coming. So with that, I would like to introduce Hassan Alhelo. He is a graduate student at LECOM and he will be presenting about the excitotoxic effects on development of auditory brainstem neuron morphology and function. So Hassan, please take it away.
Thank you for the introduction. So as it was mentioned, my name is Hassan Alhelo. I'm a second-year medical student at Lake Erie College of Osteopathic Medicine, and I'll be presenting a project which was submitted earlier this fall for publication and from the lab. So it's called Excitotoxic Effect on Development of Auditory Brainstem Neuron Morphology and Function. So a brief background on monosodium glutamate, better known as MSG. There has been evidence of excitotoxic injury in neonatal exposure. So basically, right after birth, during the early stages of life, where neuronal cell death in multiple regions have been shown, and smaller brain and body mass have been evident, along with degeneration of the retinal ganglion cells in the eyes. And there's also been shown lower density of neurons in the Purkinje cells in the cerebellum, as well as in neurons in the cerebral cortex in the brain. More specifically, we are going to be focusing on the auditory system. So the degeneration of the cochlear hair cells have been shown to be affected in previous studies, with fewer neurons in the spinal ganglion, as well as increased thresholds to the clicks and tones when ABRs were performed. A brief overview of the ascending auditory pathway. In our lab, we discuss different regions of the pathway, and previous studies from the lab in Foran et al., for example, we looked at MSG effects in the ventral cochlear nucleus, which is in the medulla of the brainstem, as well as the MSO, LSO, MNTB, and the SPON, which are nuclei in the superior alveolar complex, which is present in the pons of the brainstem. Almost all but the LSO have shown significant decrease in the number of neurons in MSG exposed animals in the previous studies. So we've been taking the study a bit further and looking at further connections between the neurons within the pathway. So the hypothesis of this project is that MSG exposure will result in the non-uniform loss of neurons throughout the auditory brainstem pathway, with the most drastic loss in the cochlear nucleus and the superior alveolar complex, and that there'll be an impact in thresholds and latencies of the click-to-evoked responses, with a possible difference that will be exacerbated with age. So to start off, we looked at the temporal bone, more specifically the spinal ganglion in the cochlea of the rat models, and as you can see, after we decalcified and sectioned the brains the spinal ganglions, we stained them with hematoxin niacin, and were able to visualize the spinal ganglion themselves. And as a result, show that the MSG exposed rats actually had larger neurons, but their density was lower, with a significant change between the shape morphology distribution between the stellate and round oval neurons, where there was an increase in the stellate neurons, as well as a decrease in the round oval shaped neurons in the MSG exposed. We also performed brainstem sections and stained them with the Genscher stains, and we'll look here at the posterior ventral cochlear nucleus, as well as the MNTB. And here we're looking at the connection between the globular bushy cells and the PVCN towards MNTB neurons, which are direct connections. And as you can see in the data, that the GBC, both the GBC and MNTB neurons density decreased in the MSG exposed rats. But in addition to that, the ratio also increased in the MSG exposed rats due to the drastic decrease of the GBCs for the MNTB GBC ratio. So where it would be between 1 to 1.53 in the controls, the ratio increased to 3.53 in the MSG group due to the decrease in the GBC neurons. We also looked at the neurons of the lateral lemniscus, and we more focused on the VNLL, the ventral nucleus, as well as the dorsal nucleus and the DNLL, and looked at the MSG exposure effects on the morphology and density of the neurons there as well. And looking at the DNLL first, we see that there's a decrease in the number of neurons in the nucleus, but there was no change in the distribution of the shapes of the neurons, whether the morphology, whether it's overall or whether their shapes were stellate, fusiform, or round. There was no significant difference between the two groups. There was also no difference between the distribution overall. The ventral nucleus of the lateral lemniscus, on the other hand, also showed a decrease in the number of neurons in the MSG group, but also showed an overall difference in shape, where the neuron size was actually also smaller. But there's also, again, no significant difference in the distribution of the shapes themselves. So between the control and MSG groups, there was no actual significant difference between whether there were more or less stellate neurons versus round neurons versus the fusiforms. Looking further up into the ponds, we're looking at the CNIC now, and the inferior colliculus, and we're comparing again with the given sustained, the controls versus the MSG to look at the density as well as the morphology. And as you can see here, there are varying shapes, various shapes and morphology between the two, where the arrowheads versus the double arrowheads and the single arrowheads all show different shapes, and there's an increase in variation in the MSG group on the bottom right. And the bottom figures are the zoomed in versions of the black box and in boxes A and B. So the results for these images, we were looking at density, and we saw that there was a significant decrease in the MSG group compared to the control group. And unlike in the DNLL and BNLL, there's extensive difference in the overall size of the neurons, as well as within specific shapes, where even the stellate and the round neurons themselves were significantly smaller in the MSG group versus the control group. The number of neurons between the two groups also shifted, as you can see in graph G, where there was the soma area increased and decreased in the MSG group towards the left. And in figure H, you could also see that there's a significant increase in the percentage of neurons that are stellates, and a significant decrease in those that are round and oval, with no change in the fusiform. The next step is where we looked at the ABRs, which are basically auditory brainstem responses. We used live animals for this portion. Looked at animals at 50 days old as well as 150 days old, and compared the reflexes, the responses to auditory clicks. So in figure A, we're looking at a single control, and figure B, we're looking at a single MSG exposed animal, and the average tracing in C at the bottom shows a total of six the recording of 6 rats from each group, and showing that there's a difference between the groups. And in D, we could also see here at the age of 150, a single control animal and a single MSG animal represent also in the blue. And in F, at the lower bottom right, you also see that, again, there's a average of 6 traces from each group from MSG and controls. And you can see there's also a difference over there between the groups, seeing that tracings do not actually overlap with one another. So look at data specifically. MSG exposure actually showed that there was abnormal responses at the age of P50, where there was significant increase in latency in waves 2, 3, 4, and 5, where there was also an increase in the intervals between waves 1 and 3, and the wave amplitude was actually that was picked up was significantly low in the MSG group, where thresholds higher and the wave amplitude was lower in the MSG groups. The P150, which means 150 years old, also showed abnormal ABRs, but it only showed it in waves 3, 4, and 5, where again the MSG exposed rats showed increased latencies in those 3 waves. By looking at the intervals in the bottom left, you'll notice that it's not only in the intervals between waves 1 and 3, but also in waves 1 and 5, where there was a significant increase in the interval duration between the clicks. The wave 1 amplitude was also significantly lower in the P150s, and the threshold of response was also higher as well in MSG group again. Finally, this graphic on the left shows the ABR results of the control and MSG groups exposed animals in comparison to the number of neurons. And on the right, you'll see that there was no significant correlation between MSG-induced wave latencies and the percentage of the neurons lost in each brainstem. So basically, the overall takeaway here is, even though we were able to show the neurons to latency differences, there was no loss of neuron. The loss of neurons does not predict any ABR latency or amplitude differences between the two groups. So basically, the summary of this project is that the neonatal MSG exposure would result in significant loss of neurons in the brain, the midbrain, with several synapses beyond the inner hair cells and the spinal ganglion neuron terminals, which has been proven in previous studies. And that there are varying effects on the density, size, and morphology of the different auditory neurons, with severely impacted neurons in the VCN and the ventriculus, but with unaffected neurons in the lateral superior olives. And that the abnormal ABRs when assessing both potentials were present, but there was no exacerbation of the effect of MSG with age. Thank you.
Hi, Hassan. Thank you for that wonderful presentation. We're going to wait just a few moments and see what questions roll in from the YouTube chat.
Of course.
We have a question coming in. Will you be continuing this research into the future? And if so, what are your plans?
So this is up to the lab. We are in the lab. I'm part of the Auditory Research Center. So obviously we're going to continue working on projects in the auditory system itself. Expanding further on this project itself, I'm unsure about, but I'm certain that we're going to continue working on looking at morphology and neurons within the connections, because we are constantly trying to figure these things out. So yeah, it's this project was based off of like multiple things we found out before. So obviously, I believe that we're going to build up on it.
That's wonderful. We do have another question. What are the theories of the actual chemical causes of neuronal issues?
That's a very loaded question. So specifically for, in this case, we were looking at the excitotoxic effects of the glycinergic synapsing, where excessive synapsing would end up leading to the death of neurons. There are a lot of chemical effects that could affect the neurons. It's an extremely broad question. Pretty much anything could affect it, and other things cannot. I'm not completely vested in that. I cannot answer that with that uncertainty.
That is not a problem. Thank you so much, Hassan. I hope you enjoy the rest of your evening, and we're going to move on to our next presenter.
Thank you.
Okay, everyone. Up next, we have Allison Burchell. Allison, are you with us?
Yes.
Hello. Allison is a graduate student from LECOM, and she will be presenting about the investigation of a long-range glycinergic projection from brainstem interneurons to the thalamus. So Allison, I will let you take it away.
Okay, can everyone see my screen? Yeah, okay. Sorry. Okay. I'm sorry, something's happening whenever I'm trying to share my screen. Okay, can we see the screen now? Okay. So hello, my name's Allison Birchall, and I'd like to thank you all for being here to see my presentation on the project that I've been working on for my master's degree. I'm a second-year medical student at Lake Erie College of Osteopathic Medicine. So to begin, I'd like to have an overview of the ascending auditory pathway. Sound comes in through the ear as an air pressure wave that hits off of the tympanic membrane, and this will vibrate the middle ear ossicles that act as a transducer to turn this air pressure wave of sound into a fluid pressure wave of sound within the cochlea. This fluid pressure wave of sound excites the inner hair cells that send an action potential along the auditory nerve. The auditory nerve will project to the dorsal and ventral cochlear nucleus of the medulla. From the cochlear nucleus, there's a bilateral projection to the superior olivary complex located within the pons. From the SOC, there's a bilateral but mostly ipsilateral projection through the lateral lemniscus to the inferior colliculus of the midbrain. From the IC, there's a bilateral but again mostly ipsilateral projection to the medial geniculate of the thalamus, which is the major auditory processing center. From the thalamus, there's a projection to the auditory cortex. So this is the known pathway, and in previous experiments within our lab, we have found what we think is a novel pathway, and that is one that goes directly from the medial nucleus of the trapezoid body located within the SOC directly to the medial geniculate of the thalamus. And what's really interesting about this direct pathway from the SOC to the thalamus is that it's bypassing both the lateral lemniscus and the inferior colliculus, which is known to be an obligatory auditory relay center. So my project is working on characterizing this novel pathway, as well as predicting what its role in the ascending auditory system may be. So this is a GAM sustained section through the pons, and what we have is the facial nerve here, and located directly medial to the facial nerve is this collection of 6 nuclei. This is the superior olivary complex. My project is focused on the most medial nucleus of the SOC, and that is the medial nucleus of the trapezoid body, or the MNTB. So the MNTB is one of the 3 primary nuclei within the SOC. In this image here, it's again a GAMP-sustained section through the pons, and outlined in white is the MNTB. These purple cells within the outline of the MNTB are principal neurons. The medial nucleus of the trapezoid body is essential for sound source localization and temporal processing of complex sounds, such as vocalizations. So in this image here, I'll show you the input and output of an MNTB principal neuron. The MNTB principal neuron receives glutamatergic excitatory input from the contralateral cochlear nucleus globular bushy cell through the calyx of held synapse. This principal neuron of the MNTB then acts as an interneuron, changing this excitatory input into an inhibitory glycinergic output locally to other nuclei within the SOC, as well as to the lateral lemniscus. We hypothesize that these principal neurons of the MNTB have a homogeneous, long-range glycinergic projection to the ipsilateral ventral medial geniculate body that bypasses the CNIC, the central nucleus of the inferior colliculus. And what's really cool about this is that the literature had stated that these principal neurons of the MNTB only project locally. So for us to find this long-range projection all the way from the pons to the thalamus is really neat. So for our experiments, we use male Sprague Dolly rats that are aged P50. And as you can see in image A, this is a depiction of the rat's skull. And through precise coordinates, we'll drill a hole where this X is in the rat's skull so that we can access their thalamus. We'll then use a retrograde track tracer called Florgold and inject that into the thalamus. We're specifically interested in injections into the VMG because this is the site of the thalamus that receives most of the auditory input. After the injection, the rat survives for 6 days, We perfuse the rat and section the brainstem, and then perform immunohistochemistry. So these are some of the stains that we do on the brainstems. I'd first like to show you the neurotrace red stain. It stains the Nissl substance of all neurons. So you can see in this image, all of these little red dots are neurons. And I've highlighted with these yellow arrows specifically principal neurons. They have a round to oval morphology and a central clearing around the nucleus. We also do a calbindin stain. Calbindin is a calcium binding protein that is specific for principal neurons within the MNTB. So we can see that these same 3 principal neurons that I pointed out on the NTR picture also take up this calbindin stain, meaning that they are principal neurons. And then finally, we look at the fluorogold uptake of the MNTB neurons. So this is the retrograde tract tracer that's injected into the thalamus. And if we see it staining neurons within the MNTB, that means that they project to the thalamus. So what we can see is that these same 3 principal neurons that were highlighted in the other 2 images have this blue color, so they uptook the fluorogold, meaning that these principal neurons of the MNTB project to the thalamus. So we wanted to make sure that the MNTB does not project to the central nucleus of the inferior colliculus. We performed 5 injections of the retrograde tract tracer fluorogold into the CNIC, and then looked at the percentage of fluorogold labeled neurons in the contralateral and ipsilateral MNTB. What we found was that there was a significantly higher percentage of fluorogold labeled neurons in the ipsilateral MNTB compared to the contralateral MNTB. However, the median percentage of fluorogold labeled neurons after an injection into the CNIC was only 0.77%, which does not constitute as a projection. So we can say that the MNTB does not project to the CNIC. This is contrasted to what we found when we did the retrograde tract tracing injections into the thalamus. We had 6 injections into the ventral medial geniculate of the thalamus and 4 injections into areas surrounding the VMG, such as the brachium of the inferior colliculus or the dorsal medial geniculate. And when we looked at the percentage of fluorogold labeled MNTB neurons after these injections, we found significantly higher percentage of fluorogold labeled neurons after injection into the VMG compared to an injection that was in a place other than the VMG, with the median percentage of fluorogold labeled neurons being 23.07%, meaning that this is a projection. So here I would like to highlight a 200 nanoliter injection of fluorogold into the VMG. This is a picture that I took from the microscope of the injection site where we put the retrograde tract tracer. And then here is a depiction anatomically of where the retrograde tract tracer was deposited. This is a section through the SOC, and the lateral MNTB is outlined. The section stained with the neurotrace red stain for nerve. And then you can also see the fluor found that 86.36% of neurons that project to the thalamus are principal neurons. We were also interested in looking at the glycine receptor density in the medial geniculate body. And this is important because if the MNTB projects to the medial geniculate body of the thalamus, there should be glycine receptors because the MNTB uses glycine as its neurotransmitter. So what we have here is a confocal image of the MGB with outlined areas of interest. This teal color is the glycine receptor, and the pink is neurotrace. So when we looked at the optical density of the glycine receptors, we found that there was significantly higher glycine receptor optical density in this area under the VMG compared to a blood vessel, the MMG, as well as the DMG. Also, it's noted that the VMG does have a high percentage of glycine receptors. So in conclusion, we have shown that the principal neurons of the MNTB do indeed project to the medial geniculate of the thalamus. We've also shown that this long-range glycinergic projection is a direct projection to the thalamus. It does not synapse within the CNIC. So what could the function of this long-range glycinergic projection to the thalamus B. And we hypothesized that these glycinergic principal neurons within the MNTB are essential for generating offset responses within the auditory thalamus. And an offset response marks the end of a sound stimulus, and it's essential for sound duration encoding and has a profound importance in speech perception. So I'd like to say thank you for Dr. Kaleza and Yusra for supporting me and just helping me and guiding me on this project, and I would be happy to answer anybody's questions.
Hi, Allison. Thank you very much.
Yeah.
There's one question. We'll wait for more to come in, but the question is, if you project this to humans, would you find the same result? And if so, do you think that that would have any impact in issues with hearing and things like that in humans?
So it was recently found actually by Dr. K and another researcher that there is an MNTB in humans. So it was disputed for a while whether or not there was an MNTB. And I'm not sure what the function of the MNTB in humans would be, because rats they can hear a lot more sounds than us. So maybe this role of the MNTB in a rat is more important than what it would be in a human. But there's still more work to do on what potentially the MNTB's role in a human would be, but it would be hard to measure because we can't do the things that we're doing with rats with humans.
Thank you. You have a couple comments. Great job, and very interesting presentation. But I don't see any other questions coming in. So thank you so much, Allison. Very wonderful job.
Okay, thank you.
And with that, we will move on to our next presenter. Next up, we have Robert Waters. He is also a student at LECOM, and he will be giving a speed talk on the effect of drawing microbiology concepts on short-term retention before and after interrupted learning. So Robert, I will turn it over to you and you may take it away.
All right, thank you, Amber. So like she said, I'm going to talk about drawing and microbiology and how that relates and how that can potentially enhance student learning and retention. So in medical school, especially the preclinical years, our students have to learn copious amounts of information in a short period of time. And at the same time, they're learning multiple subjects, potentially in the same day. So it's we need to find the most effective approach to teach these students this information in the time period they have to learn it. Previous research, especially in like the undergraduate level, has found that drawing does enhance learning and retention, but there's limited studies that actually looked at more advanced topics that we would see in the medical school level or the graduate school level. So one approach we want to take is looking at the drawing and if we can incorporate this into our micro education. Also, with that in mind, since we do know in our program at least that maybe in the morning you might have micro and then you might have physiology followed by micro again, we do have the aspect of what we call interrupted learning, where you learn micro to begin with and then you start learning about some other kind of subject like physio or biochem, and then you go back and learn micro again. So can giving students the opportunity to draw, can that help with them retain the information when they start learning about different subjects? So that's one of the things we want to look at as well. So as part of my first aim in my dissertation, what we're looking at is specifically the drawing aspect of this. And what we did is we got an N of 82, and they consisted of graduate students and medical year 1 students at LECOM. So first we get these students come in and they take a questionnaire to give an idea of what their preferred learning style is or what they think their preferred learning style is. And after that, we would randomize the two groups into two different rooms. One would be a read and draw group, and the other one would be just reading the text group. Each group had 15 minutes to go through the text and or draw, depending on which group they're in. If they were in the drawing group, they received a drawing prompt, as well as a figure legend and a background to draw on that had the virus already there. The text consists of the adenovirus replication cycle. Previous research has found that when you do give students guidance, it does lead to benefits in their drawing, as opposed to if you just give them a blank piece of paper, it can lead to more confusion in that way. So after they completed their drawing or completed reading the text, they then received post-test number 1, all about adenovirus questions, 10 questions on the replication cycle. After that, they then received text number 2. Both groups did not receive any drawing prompt for text number 2, just to read the text, highlight, take notes. And text number 2 was specifically looking at Staph aureus' quorum sensing mechanism. So two totally different concepts. So one was adenovirus in the first text, and then we go on to quorum sensing. After they finished reading that text, we then move on to post-test number 2, which consists of 10 questions of quorum sensing, and then 10 questions about the adenovirus replication cycle, 10 new questions. So we didn't reuse questions like that. There were 10 brand new questions. And that was to gauge our interrupted learning approach. So here's just a background of one of the student drawings. So this is what they received. They would receive this background and like the pre-drawn virus there, and then they would draw out that replication cycle. And they would also receive a figure legend for what each part of that virus actually represents. So first off, we looked at the immediate performance on drawing versus text-only groups. And what we found was in that first post-test, specifically looking at the adenovirus, the group that actually drew out the adenovirus replication cycle performed significantly better on post-test number 1 related to adenovirus questions. We moved on to test number 2, post-test number 2, which looked at the overall performance between the drawing and text-only group. This is the test where we had both the quorum sensing and adenovirus questions on there. We found that between the two groups, there was no significant difference on their performance. But when we looked at the interrupted learning effects on adenovirus questions, we actually, in post-test number 2, we actually found that the drawing group performed significantly better than the text-only group, indicating that they were able to retain that information a little better than, or significantly better than the text group. And then we also looked at student performance on quorum sensing topics just to see if there's see if they just by default learn better by reading text between the two groups. And once again, we found no significant difference there. So kind of a little control there to make sure that neither group's better at reading text and perform better that route. Like I said, in our questionnaire, we looked at a couple different things. I didn't include them all in here due to the speed talk, but a couple of things we asked was, I like to draw out concepts while studying. We found no significant difference there, indicating that both the groups do like to draw while studying. And then we also looked at reading text in order to learn new concepts. Once again, we found no significant difference between the two groups there. So in conclusion, what we found was drawing immediate effects do have benefits for the students. We found that when the students drew out the adenovirus replication cycle, which is a pretty complicated replication cycle, they performed significantly better on that post-test than the ones that just read the text. We also found that when interrupted learning was introduced, the students who ended up drawing during that first post-test or that first text actually performed significantly better on adeno-specific virus questions during post-test number 2 after the interrupted learning was applied to the scenario. Also, between text-only quorum sensing performance and the overall performance, there was no significant difference. And we found no significant difference between, in all the questions we asked, between the students' perceived learning styles. What we can take home from this is drawing can potentially be used as a potential supplement material that we can incorporate into our lectures. So after they learn about, say, an exotoxin, we can then have them draw out that process in order to reinforce that concept. So future research, what we plan on looking at, we currently just finished up looking at online drawing workshops with our first year medical students during the core micro program. Currently collecting data on that, just finished that up actually on Monday. So right now we're getting the process of getting that all collected and start to analyze that information. Followed by, depending on what that looks like, introducing what is drawing companion in the fall of 2021 for the MS1s, which will consist of multiple different drawings in immuno and in microbiology. And lastly, looking at the long-term effects of drawing as well to see if there's any benefit as far as long-term retention in these concepts when you actually have them draw out this material. And with that, are there any questions?
Very nice job, Robert. Thank you so much. Well, I'm waiting for the chat to populate on YouTube to see if anyone has questions. I have a question.
Okay.
I was wondering if you think that the same results will correlate to other fields of study.
I think it depends on what you're specifically looking at. I mean, like anatomy definitely could, that's definitely a image-based program or a hands-on approach where you need to look at those structures. So I really think it depends on what you're looking at. Obviously, trying to apply it to like a literature setting may not be as effective. I think in sciences in general, though, I think it could be useful in multiple different disciplines in science.
Very cool. Thank you. Okay. Oh, we have one more question. Do you find different results from different age groups of people?
So as far as the previous research I looked at, they didn't find any like different results as far as whether it significantly helps the students. So I looked at some studies that looked at elementary school students. Some were like middle age to high school students and also undergrad students. And they all found that there's benefit to that. It's just the main issue is how they actually apply the drawing. So how much guidance you actually give the students, whether or not you give them that figure life and whether or not you just give them a big piece of paper and say, here, just draw it out and see what happens. It really depends on that guidance, which is why there is some conflicting results, is because different studies will give students different amounts of guidance, which can lead to different results. But overall, it does look like it benefits multiple different age groups as far as enhancing that learning and enhancing that retention of that concept.
Very, very interesting. Well, thank you so, so much for all of your work on this.
Thanks.
Okay, everyone, we are a little bit ahead of schedule. So we are going to take a break until 5:10 and then come back to hear from Lauren Avery. Hello everyone, welcome back. We are ready for a speed talk from Lauren Avery, who is an undergraduate student at California University. She will be talking to you about the potential discovery of new antibiotics in soil. So Lauren, I will let you take it away.
Hi guys, so I'm Lauren and I'm here to talk about my research study I did. So I test for antibiotic producing microorganisms in a soil sample. So antibiotic resistant bacteria have caused a shortage of viable antibiotics, which has led to the antibiotic resistant crisis. Antibiotic resistance is caused by bacteria developing the ability to resist antibiotics, which then are unable to inhibit their growth or kill them. Unfortunately, there has been a lack of new antibiotic development from the pharmaceutical industry, which caused there to be a shortage of antibiotics that most bacteria aren't resistant to. The shortage of novel antibiotics is a problem because treatment for an infected individual are more difficult to overcome, and those infected individuals can also spread antibiotic-resistant bacteria to healthy individuals. So antibiotics can be discovered through many ways, but one way in particular is through the isolation and screening of soil bacteria. And the steps are pretty simple. So first, bacteria are cultured from a soil sample using serial dilutions. Next, individual colonies will be screened for their ability to inhibit 3 different types of pathogens. And then lastly, any colony able to inhibit any of the 3 pathogens is an antibiotic producer. And my hypothesis is that isolated bacteria from the soil sample will produce non-novel antibiotics because non-novel antibiotics are hard to discover. Like I mentioned briefly, a series of 6 10-fold serial dilutions will be performed first to quantify the biomass of a soil sample. The soil sample used in the study was collected from California University of Pennsylvania. 100 microliters of the first 4 dilutions will be plated on an LB agar petri dish as shown on the slide. Then once the colonies start to grow, a master plate was formed by picking 12 unique single colonies from any of the 4 dilution plates and patching them individually on the square grid of the master plate as shown on the right of the slide. Next, all isolates on the master plate were analyzed to determine whether they were gram-positive or gram-negative using differential and selective media. The 2 media I used was McConkey agar and mannitol salt agar. So any growth on the McConkey agar suggests that bacteria are gram-negative because the media inhibits the growth of gram-positive bacteria. Also, if the bacteria cause the media color to change from the original color as shown on the slide, bacteria are not lactose fermenters because the magenta color of the media indicates that the bacteria can ferment lactose. Mannitol salt agar preferably selects for gram-positive bacteria, but gram-negative bacteria are able to grow on the media as well. And any bacteria that are able to change the color of the media to yellow indicates that the bacteria can ferment mannitol, as no color change means that the bacteria are unable to ferment mannitol. And lastly, all isolates from the master plate were screened with 3 different types of pathogens: Staphylococcus epidermidis, Pseudomonas aeruginosa, and Bacillus aureus. 3 LB agar petri plates were streaked all over with each individual pathogen. Then each isolate from the master plate was patched in its corresponding grid square. So here's my resulting master plate. Isolates 10 and 11 did not grow, so they were not analyzed. I also looked at the morphologies of all of the isolates except 10 and 11, but there was not a lot of differences between all the isolates, so more analysis of the isolates needed to be performed. So I looked and analyzed the results of the differential and selective media tests. So here are the results of those. So on the left are the results from the McConkey agar plates. Isolates 4, 7, and 12 were the only ones that grew on the media, which suggests they are gram-negative. Isolates 1, 2, and 3 had a slight color change around the edge of the plate, which suggests they might be able to ferment lactose, but the color change isn't big enough to give a solid answer. And on the far right are the results from the mannitol salt agar plate. Isolates 1 to 9 were the only ones that grew on the agar, which suggests they could be gram-positive. These two isolates also had a media color change to yellow, which indicates they can ferment mannitol. And lastly, these 3 plates are my results of my isolates from my master plate for their ability to produce antibiotics against any of the 3 pathogens as shown on the slide. For a bacteria to produce antibiotics against a pathogen, the bacteria would produce a halo around itself, but unfortunately, none of my isolates were positive for any production of antibiotics. So I want to remind you all that the purpose of the study was to discover potentially new antibiotics from soil bacteria, which could supplement for widely used antibiotics that bacteria are commonly resistant to. The results of the differential and selective media test, as I mentioned previously, are important because gram-negative bacteria are more resistant to antibiotics because they have 2 membranes in their structure, and gram-positive bacteria only have 1 membrane. In the picture on the right, I put stars next to the membranes so you can see where they are. Again, all of my isolates from my master plate were negative for antibiotic production against the 3 selected pathogens. Altogether, these results show that even though microorganisms that produce antibiotics are ubiquitous, there is a strong urge of more help to discover them. There is a slim chance that a novel antibiotic will be found on the first couple rounds of screening, but with more help from people all over the world, novel antibiotics can be found at a faster rate. For example, the Tiny Earth Project is a global network of instructors and students that focus on discovering antibiotics from soil. If any new antibiotics are discovered, the bacteria responsible for producing the antibiotic would be isolated to form a pure colony. Then the information would be gathered and put into a database. In conclusion, there is only so much time until all bacteria become resistant to all available antibiotics used today. So new potential discoveries are needed to combat the problem of running out of viable antibiotics. Any questions?
Wonderful job, Lauren. Let's give it a few seconds to see if any questions come through YouTube. Okay, we do have a question. Where do you see this research going into the future?
So I hope to do this again, but with a different soil sample so that maybe I could discover new novel antibiotics or being close to discover like an antibiotic, but maybe not novel, but just to discover like any new antibiotics that could be helpful in the future.
You may have said this at the beginning, I may have missed it, but where did you take your soil samples from?
So they were taken at California, University of Pennsylvania.
Do you think that might have any effect on your results?
Maybe if they use any kind of like herbicides or anything around, because they were taking around like, a bush. So if herbicides were used around that bush, maybe it would have caused like the bacteria around there to like not have any antibiotic production in them.
Very cool. Well, thank you so very much, Lauren. We are going to move on to our next presenter. Next up, we have Michaela Kling. She will be presenting a speed talk, and she's also from California University. And her speed talk will be about the antibiotic resistance in environmental bacteria. Michaela, take it away.
Thank you so much. Okay, let me get my PowerPoint up. Alrighty, can you guys see that? Awesome. All right, so hi everyone. My name is Michaela Kling. I am a junior biology major at California University of Pennsylvania. And today I'm going to be talking about a little bit of the research that we've been doing in my microbiology course, and specifically with the antibiotic resistance in environmental bacteria. And so this is pretty important because there is an antibiotic crisis that is happening worldwide. And pretty much what that is that we're finding that a bunch of antibiotics are becoming resistant to the things that they are fighting against. And this can be due to the overuse or misuse of antibiotic medications. And there's actually been a study done where there have not been any exceptions to this so far. So all antibiotics that have been here so far have actually been into this crisis. And so we need to try to find new viable options to combat this crisis, and so then we can actually keep with these antibiotics. And the main thing that I was really interested with this study is that are undergraduates able to find antibiotic producers on their own campus? Can you utilize the classroom to actually combat this worldwide crisis that's happening? And so my hypothesis was that there will not be any organisms capable of producing antibiotics in the environment at my home university of California University of Pennsylvania. And so to do this, we actually collected 1 gram of peat soil sample, and this was collected behind our biology building at Cal U, underneath a bush, just like Lauren's, and we completed serial dilutions through this. And from these serial dilutions, we got single colonies. We were able to put those on a spread plate and make master plates through that. And each lab bench got their own master plate. And so from there, they were able to use that throughout the entire experiment. And through their own master plate, we actually looked at the colony morphologies of each of the isolates. We did selective and differential medias, as well as actually finding if there's any antibiotic producers through the spread patch antibiotic production. So our first step was to calculate the colony forming units. And to do this, like I said, we grabbed 1 gram of peat soil and we completed serial dilutions through this. And we actually had 4 plates constructed, as you can see in the picture. And from here, we used the single colonies to create everyone's master plates. And I'll continue with that here in a little bit. But also, we were able to calculate the CFUs, or the colony forming units. And for myself and my lab partner, Abigail King, we actually used the plate 10 to the -4th, which had 150 colonies on, and that had a CFU of 1.50 times 10 to the 6 CFUs per grams. And so from these single colonies that I mentioned earlier, we were able to create these master plates. And Abby and I actually got this master plate 1. And on this, as you can see in the picture, there are 12 isolates and all 12 grew. Now, I do want to note that both of these pictures are of the same master plate. They are just from different viewpoints. So then it was easier to see because we are learning remotely this semester. So we were not allowed in lab. And we actually got these pictures from our wonderful professor. And from these isolates, we were able to look at the morphology. And as you can see, everything that we looked at. And through these pictures, you can notice that there are a bunch of similarities. There's not many differences and there's not a lot of diversity. So a lot of other tests had to be done. But I will also say that there were a couple limitations with these morphologies themselves, especially with the surface elevation, because since we couldn't move the plates around, it was very, very difficult to see how raised they are off of the plate themselves. So after these master plates, we actually also did the selective and differential medias. We actually got to pick 2 out of 3. We either got to do the mannitol salt agars, the eosin methylene blue, or the McConkey agars. So my lab partner and I chose the MSA and EMB plates. And the MSA plates primarily grow gram positive. However, it is not a perfect test. You can grow gram negative. It doesn't fully inhibit it gram negative, but that's why we do extra test to see everything that we might get. So primarily you do get gram positive, and as you can see, we do have some growth on the top picture. And you're also able to see if they are mannitol fermenters. If they are yellow, which we have a lot of yellow, they are mannitol fermenting. If they are red, they are non-mannitol fermenting. So in the bottom picture, we have our EMB plate, and this type of plate grows the gram-negative. So anything that is grown on the plate is gram-negative, and it does do a better job of inhibiting the gram-positive. So it is a little bit of a better test and inhibition through the two plates. So we do have a lot of gram-negative growth on here, but it also does show lactose fermenters. So if it has a slight purple tint, then it is a lactose fermenting. And a majority of our isolates between the two plates were gram-negative, and they were mannitol fermenting. However, this doesn't fully show if they're antibiotic producers or not. So then we completed our antibiotic production, which we did this through patch plating. And pretty much what patch plating is that you take a bacterial tester and you put it on the plate to create this lawn. And then you take your isolates and you place them on top of the lawn. And this layering can actually cause the isolates to grow and even possibly form clearings if they're antibiotic producers. So we screen against 3 different organisms: Staphylococcus epidermidis, Bacillus aureus, and Pseudomonas aeruginosa. And you can see them in the bottom picture there. The top picture is actually a zoomed in version of the staph one, but I do want to note that all 3 of the plates did show that lawn-like structure that you see in the top picture there. So we did notice that 2 isolates grew, however, only 1 produced clearing, and that one that produced clearing was isolate 11 on the staph plate. I do want to say that I definitely think that more experiments and testing should be done to see why the other isolate did not produce clearing, and see if any other type of bacteria or anything can work with this, and see if other isolates can grow from other types of organisms. But for now, the isolate 11 is an antibiotic producer, so it is positive for that. And there are a couple future directions that we can go from there. First of all, we can identify the positive organisms taxonomy and basically see if we already know about this antibiotic producer or if it's a new strand, and we can go from there. We can also test for the other antibiotic producers based on other pathogens. So instead of the staph, bacillus, or pseudomonas, then we can do something else and see if any of the other isolates grow, or if, say, isolate 10 does grow the clearing that we wish to see. And then finally, we could also test soil samples from different places. That could be in other places at Cal, in the state, in southwestern Pennsylvania, in the US, out of the US, whatever. But I really want to do that. I think that that's pretty cool, that you can maybe see the similarities and differences between different areas, and it also just helps to get a wide range of variety of possible antibiotic producers. So in conclusion, again, there was an antibiotic producing bacteria found in the soil sample on master plate 1 on the staph plate, which was isolate 11. But mainly, I was really happy that we could find that students actually can find antibiotic producers on their own campus. So this meaning that you can utilize the classroom to actually do this and contribute to this worldwide crisis that's happening. And you can use a bunch of numbers, you can combat it with numbers. You can just get a lot of people to do this with different types of classes. And I think that that's absolutely amazing. And I think that that's probably my favorite, I should say my favorite part, but that's probably one of my best things out of this whole entire experience. Are there any questions?
Very nice job, Michaela.
Thank you.
I'm going to monitor the chat to see if any questions come through, but I have a question for you.
Yeah.
If you were to choose another location on campus to do this same study, where do you think you would choose it and why?
So I would probably choose it. So we have a really big river that runs around campus. I would probably choose it closer to there because there wouldn't be as much human intervention, there wouldn't be pesticides and mulch and different things like that. So I think that you might be able to find more antibiotic producers there, or at least a different variety of it.
Great, thank you.
You're welcome.
We have a question that came in from the chat.
Mm-hmm.
It says, why did you choose the MSA and EMB augers over the other available types of auger?
So the MSA does just do the gram-positive, while the EMB and the McConkey do the gram-negative. And pretty much you could have done either the McConkey or the EMB. I felt personally, my lab partner and I felt like everyone was going to do the McConkey plate. So we wanted to be different and do the EMB and pose ourselves a little bit of a challenge so we weren't like everyone else. But you could have picked either the McConkey or EMB. It would have proven the same results.
Very, very good. You have a couple comments. I just want to let you know, great presentation. It's tough to run experiments on campus or off campus, so fantastic job. And I would agree with that 100%, so wonderful job.
Thank you so much.
Okay, Michaela, we are going to move on to our next presenter of the evening. Next up, we have Heaven Cole, also is presenting a speed talk from California University at Pennsylvania, and they will be presenting about uncovering innovative antibiotic-producing bacteria from soil samples. So take it away, Heaven.
Hello, everyone. My name is Heaven Cole. I am a student at California University of Pennsylvania. I will start sharing my screen now. You may notice that my video is in black and white, and you may be asking yourself, how did she do that? How do I do that? The answer is that I have no idea how to stop doing it, so I cannot tell you how to do it. So I hope we can all enjoy. Yes, thank you, thank you. So I hope we can all enjoy this black and white theme I have going on. Thank you. So again, I am a student at California University, and I bring you this presentation with great gratitude to Dr. Volkanist, who had worked very hard with us and with Tiny Earth to make it possible for us to do this experiment. Today, we will be uncovering innovative antibiotic-producing bacteria from soil samples. Now, to do this, I would like to begin with a bit of history, going back to 1928. I would like to, except my mouse has stopped working. We go. We're going back to 1928, when Alexander Fleming left a petri dish of bacteria next to a window uncovered. Sometime later, he came back to the petri dish and he noticed that there was mold growing in the petri dish, and that this mold excreted a sort of juice, mold juice, if you would. And where the mold juice was, that bacteria was not growing. The bacteria's growth was being restrained. This mold juice was penicillin, which today can be used to treat such things as gonorrhea, pneumonia, strep throat, and other such. The penicillin was first mass-produced in 1941 when resources were available to do so. The dilemma that we are discussing today is that by 1942, the first penicillin-resistant germ was identified. And this antibiotic resistance is a very great concern that we have. For one, because of how fast it develops, and for two, because of how slow we have been in developing new antibiotics in order to combat this. On the left, you can see a quote directly from the Center for Disease Control and Prevention, or the CDC website. Antibiotic resistance is one of the biggest public health challenges of our time. So I have gathered you all here today to ask the question, can we meet a growing need and benefit humankind from our own backyards? The answer is yes. Yes, we can. Technically, though, not my backyard, not this time. No, instead, it was from the California University of Pennsylvania's backyard. What happened was that soil was taken from one of the, by one of the academic buildings. It was Frick Hall, to be precise. You may have no idea what that is. And from the soil, 1 gram of soil was taken, mixed in with water, and diluted at least 4 times, and then spread over these 8 dishes, 8 plates you see on the left. One of those plates was picked, and from that plate, we took the 12 different bacterial colonies you see on the right on my master plate. When you look at the master plate, you can see that there is incredible diversity. The isolates are wonderful. Some of them are raised, some of them glisten, some of them look rough, some of them look smooth. Indeed, that morphology is gorgeous. So you would expect that these bacterial colonies would all be very different. Well, going onward, committing more experiments, gathering more results, we find that actually these isolates were not that different, not just yet. Here we went through a number of different differential and selective media. On the left are the mannitol salt agar plates, or MSA, agar, MSA plates. And the purpose of the MSA plates is to isolate gram-positive bacteria, or to allow gram-positive bacteria to grow and not allow gram-negative bacteria to grow. Also, the plates are supposed to turn yellow in the presence of mannitol fermentation, or when mannitol is broken down by that bacteria. And mannitol is a type of sugar. Looking at the left, you can see that there's growth. Possibly you cannot see it well, but there was. I assure you, there was growth. What you can also see is that they are both very pink. No mannitol was fermented here, my friends. On the right, you can see the eosin methylene blue agar plates, or the EMB agar plates. And what they are meant to do is that they are meant to isolate gram-negative bacteria. And they will turn green or kind of brown purple when fermenting lactose. And lactose is another type of sugar. It's actually found in milk. And looking at these plates, you can see that there's definitely very good growth in some of these isolates. I'm thinking specifically 1, 3, 4, and 7. And then you can see growth in other of the isolates as well. Looking at them, you can also see there's no green, there's no purple. No lactose was fermented here, my friends. The result, as I wrote them down, was that growth was apparent in 6 of the 12 isolates on the EMB plates and 9 out of the 12 of the MSA plates. This does not mean that some of the colonies were both gram-negative and both gram-positive. It means that further tests would have to be conducted in order to verify the colonies. However, I have been told that EMB plates are considered to be more trustworthy, so we are going to assume at this time that 6 of the 12 isolates are gram-negative. Now, looking at these, you can see that that beautiful diversity you saw on the master plate is not reflected here. However, moving on, there is no however. They continue to not be that different. Here we see that we screened for the antibiotic production, and this is through patch plating, where a toothpick was taken to our isolates from the master plate, and they were scraped over these 3 different bacteria that you see listed at the bottom. And these are proxy for very infectious bacteria that you may know of. And what is supposed to happen is that if the bacteria from our isolates produced antibiotics, then there was supposed to be a clearing where the bacteria listed under these 3 petri dishes would not grow. Looking at these 3 petri dishes, you can see that there was growth. There was indeed a great amount of growth in some of them. So in short, it appears that my experiment specifically did not produce antibiotics. As a matter of fact, in the total 216 bacterial isolates created in our microbiology class, only 7 produced antibiotics. This may be disheartening. However, consider my isolates came from 1 gram of soil, and all 216 isolates were taken from the same location on the California University of Pennsylvania campus. Imagine what would happen if you took your soil samples from your backyard and from your neighbor's backyard and from their neighbor's backyard. The number of antibiotic producing bacteria you could find could be incredible, and those antibiotics hiding in your garden could help our generation overcome antibiotic resistance present in different bacteria that are causing us great issues. Thank you for coming to my speed talk. Are there any questions?
Very nice job, Heaven. Let's give the chat a couple seconds to populate. Good. Okay, we have a question that's actually from within our room here. We're wondering, if you repeated this in a different season or over all 4 seasons, would you find, or would you expect to find different results?
It depends on how deep you dig. Some bacteria are not very cold friendly, some bacteria are not very heat friendly. So depending on how deep you dig in order to avoid frost, or even where exactly you are digging, I imagine desert versus an Antarctic tundra would get you different results. Yes, depending on how deep you dig, perhaps seasons would affect your results.
Wonderful. Well, thank you so very much, Heaven, for joining us. We are going to move on to our next presenter.
Wonderful.
Thank you. Okay, next up, we have another speed talk from California University of Pennsylvania. And I would like to introduce Abby Reichelderfer and Raylan Graybill, and they will be presenting the contribution towards antibiotic discovery through bacteria in soil collected in Western Pennsylvania. So Abby and Raylan, please take it away.
Hi, so like stated, we are here to present our contribution for antibiotic discovery to you guys today, and our sample just so happens to be taken from our hometown here in California University of Pennsylvania. So let me start by saying why we're doing this research. Well, antibiotic resistance is becoming extremely prevalent worldwide. It's prevalent in our agriculture, ecosystems, and more importantly, our public health. So why is this still an issue if we know that it's a problem? Well, there's a steady increase in antibiotic resistance. However, there's a lack in antibiotic discovery. In fact, the last class of antibiotics was discovered in 1987. So we're here to say that crowdsourcing and the use of undergraduate research like we have done can lead to the discovery of potential antibiotics. So we're just going to briefly discuss the methods that we took in order to get our results. First, our professor, Michelle Volkanis, collected soil sample here behind our Frick building, which is the biology building here at Cal U. And we were able to then perform serial dilutions and get colony plates. From those plates, we were able to extract using a pick and patch method to obtain 12 isolates. We were then able to perform selective and differential tests to see how our bacteria metabolized, as well as to determine if they were a gram-positive or gram-negative bacteria. Finally, we were able to screen against different pathogens to see if our isolates were able to produce antibiotics.
So first off, Dr. Volkanis, our professor, collected our soil sample from California, Pennsylvania, behind the biology building, about 2 inches deep. We obtained colony counts for serial dilutions, 10 to the -4th to 10 to the -7th. We did not do any serial dilutions from before that because the amount was just too hard to justify. We calculated a CFU of 1.50 times 10 to the 6th CFU per gram. We inquired 150 isolates for our 10 to the -4th serial dilution, 72 isolates from our 10 to the -5th dilution. We obtained not 5 5 isolates from our 10 to the -6 dilution, and 9 from our 10 to the -7 dilution. We did have some human error possible that happened to our 10 to the -6, because that is why we got a higher number for our 10 to the -7. So after obtaining our 12 isolates, we used pick and patch method to obtain these. And after doing so, we were able to examine colony morphology. There was minimal variation in our isolates. Why is examining colony morphology important, you might say? Well, because by examining colony morphology, we were as easily able to tell that our isolates were not the same based on our actual visual cues. So next, after doing that, we analyzed the selective and differential media on McConkey and mannitol salt agar plates. McConkey plates select for gram-negative bacteria and differentiates between lactose fermenters. For our mannitol salt agar plate, it is selecting for gram-positive and it differentiates between mannitol fermenting. We did receive lactose fermenting on our RG7 plate, and our mannitol salt fermenting was on isolates 4, 7, and 11. We had 3 gram-positive isolates and 8 gram-negative isolates. One inconclusive isolate that we did have, though, was our isolate 11. We were unable to tell, but we could definitely tell that it was mannitol fermenting.
So this is where we finally detected our antibiotic producers, and we got one hit. It was RG11 isolate, and this was an antibiotic producer against Bacillus aureus. So overall, we screened against 2 gram-positive pathogens. This was Staphylococcus epidermidis and Bacillus aureus, the one we got the hit on. And we also screened it against 1 gram-negative, and that was Pseudomonas aeruginosa. So now I want to reflect a little bit more on whether we supported our hypothesis or not. So overall, we did this in a class setting. So me and Abby personally, we were able to find 1 antibiotic producer out of our 7 particular isolates. The class, however, was able to isolate 7 antibiotic producers total out of the 216 bacterial isolates we observed. So ask yourselves, if our class alone was able to find 7 total antibiotic producing bacteria, imagine if more undergraduate universities contribute to this. And that's exactly our objective with crowdsourcing. We believe that if everybody takes initiative and produces their information and research to these crowdsourcing platforms, like the Tiny Earth database that we use, then we can contribute to finding more antibiotics since we are heavily in need of them. And now I just want to conclude with where we can go from here. Since we were able to get 1 isolate, we want to determine if that isolate was gram-positive or gram-negative, since we were unable to obtain a strict read. We also then want to classify its genus and species. And finally, we want to screen this antibiotic producer that we found against other gram-positive pathogens to see if we can get this one to produce antibiotics against other pathogens. This concludes our presentation. At this time, we can take any questions.
Hi guys, very wonderful presentation.
Yeah.
I have a question for you. I don't see anything coming through the chat, so I'm going to ask my question and we'll see if other questions come through. So you mentioned that you want to test your isolate against other pathogens. Do you have any idea or a prediction of what pathogen you would choose to test it against, or a variety of pathogens?
That's absolutely a great question. So we learned that our isolate was able to produce against produce antibiotics against gram-positive pathogens. So we just want to collect a varying amount of other gram-positive pathogens to test it against and see if there's a correlation that it produced antibiotic. It's an antibiotic producer against those specific gram-positive bacteria.
Okay, very good job.
I do not see any more questions coming through. So we're going to move on to our next presentations, but thank you so much. You guys did a wonderful job.
Thank you.
Okay, everyone. Up next, we have presenters Kendall Leek and Alex Lear, presenting also from California University of Pennsylvania. They will be presenting a speed talk about the undergraduate research of the antibiotic crisis. So Kendall and Alex, please take it away.
Thank you. I'm going to start screen sharing.
Hi, thank you for having us.
Okay, hi everyone. Today we're going to talk about the undergraduate research of the antibiotic crisis. I'm Kendall. And this is Alex.
Hi, I'm Alex.
So I want to start by talking about what the antibiotic crisis is and just in general what an antibiotic is. So antibiotics are a medicine that prevents infections caused by bacteria. Most antibiotics come from microbes and they are used to kill bacteria or keep them from reproducing or copying itself. We use antibiotics in medicine and surgery, and it has extended the life expectancy. And there is now an issue that has come upon us that against antibiotics, and it has caused major concern in the global health. So why? The answer is that there's a bacterial resistance occurring towards antibiotics, and it can affect many individuals of different regions and different ages. So now I'm going to talk about what causes these resistance to, like, bacterial resistance to antibiotics. The first one is mutations. So when bacteria multiply, errors occur, creating mutations which then cause a change in the nucleotide sequence of the genes. One of the most well-known is antibiotics are being overused and have caused evolution to occur and resistance to the antibiotics. And they are also being inappropriately prescribed, and they are extensively used in agriculture. And this has become a problem because when they give the animals the antibiotics, we consume animals. So, and then another reason is that they, due to economic and regulatory difficulties, newly developed antibiotics have not been able to be produced, which has caused major concern to microbiologists and infectious disease specialists. Now I'm going to talk about why this is an important topic. Well, antibiotic resistant diseases have been spreading across the world, like I said, and infections that are antibiotic resistant have now become a burden to the health industry and have become a costly issue. So resistance to antibiotics will be an overall threat to the global population. So I'm going to talk about some things that we did in our experiment. So here's a little picture to our right of like the steps of the experiment. And to our left, I have just it written down in word form. But in week 1, there was, we collected soil, well, Dr. Valkanis collected the soil from California University behind our biology department. And then we performed serial dilutions, which then we took 1 gram of the soil and transferred it into 9 milliliters of solution. And then it was transferred into a 110 dilution. And then 5 of those dilutions were performed and then plated to obtain single colonies. The plates were then incubated at 30 degrees Celsius for 3 days. We needed to isolate the colonies due to the dilution plates mixing over time, so we used the pick and patch method, which involves picking an individual bacterium colony, and the bacteria were picked from dilution plates using a toothpick and patched onto a new AR plate, which we call the master plate. After the plates were picked and patched, the individual bacterial colonies were then organized by number to which isolate they were, and then we characterized these plates.
So from week 3, we now analyzed the bacteria using McConkey agar plates and mannitol salt agar plates. These are differential and selective medias. The McConkeys present with pink colonies and red media, which indicate gram-negative and lactose fermenters. If opaque colonies appear on yellow media, this is indicative of gram-negative and non-lactose fermenters. And if there's no growth, then that's indicative of gram-positives. The mannitol salt, if you see opaque and pink media, that is indicative of gram-negative and non-mannitol fermenting. Growth in the yellow media indicates gram-positive and mannitol fermenting, and no growth is indicative of gram-negative. And that was what we did over the course of week 3. For week 4, we did patch plating, which included the escape pathogens, Pseudomonas aeruginosa, Staphylococcus epidermis, and Bacillus aureus. And what we did was we coated plates, and from there we took from our master plate and put on bacteria to see if any of them were antibiotic producers, and we'll go over that more, but if there was a clearing around it, it tested positive. If not, then it was negative. We also did calculations for the colony forming units, and ours was 1.96 to the 10 to the power 6 colony forming. And we analyzed the colony morphology about the size and the shape and the texture of each colony. And that really gave us a better understanding in helping identify what colonies we had been growing. You can see on our master plate. So our results concluded that in the MSA plates, most of the isolates were opaque with pink media, indicative of gram-negative and non-mannitol fermenters. In isolates 1, 2, and 12, there wasn't any growth, and in isolate 9, there was growth, indicative of gram-positive bacteria that were mannitol fermenters. And we concluded that, as you can see within the yellow media. In the MAC plate, there was no growth for 1, 7, 9, and 10 through 12. However, 6 was positive for being a lactose fermenter, and the rest were weakly positive. So from there, after we screened for our antibiotic producers, as you can see, the total prevalence, however, was 0%. We didn't have any great clearings or anything like that. We did have some other students in our class that did really well and found antibiotic producers that will go on to further analyze them, but our plates were not successful. So to conclude, there weren't any antibiotic producers that we found, unfortunately, but as an undergraduate program, we can contribute greatly to finding antibiotic producers, and it's really important to do this mass examination on what we can find, especially in soil, since that is the greatest biodiversity that we have. And then from there on, I'll let Kendall go.
Yeah, so like Alex said, this is it's like a very important topic, you know, because we use antibiotics every day, and it is important that we find new ones because of the resistance that is occurring. So if we said that if we were to further extend this experiment, we could possibly test against eukaryotic organisms to see if they would be harmful to them. And that's all we have today. Thank you.
Hi guys, wonderful presentation. Thank you so much.
Thank you.
I have a quick question for you.
Okay.
I was wondering what you thought about if you repeated this experiment but used a surface water sample instead of soil, what would you expect to find?
I would anticipate finding different types of maybe antibiotic producers. We talked about acid mine drainage, particularly in our class, and I thought that was a very interesting sample to collect from, because, you know, bacteria that would be able to grow and that would be very unique and produce very different things than just bacteria found in soil. That's a very good question, and I would expect different results, and it would be nice to sample from different areas, not just soil. We figured that soil was the best because it does have the greatest biodiversity, but definitely water samples would be interesting in the future, for sure.
Thank you.
Thank you.
We have one question coming in. Give me one moment.
Sure.
Okay, so the second question for you is, would you expect to see a difference in the bacteria on the surface level versus a couple centimeters or a couple inches down?
Absolutely. We also got, I'm sorry, go ahead, Kendall.
You can go ahead and talk. It's okay.
So we also got to learn about the different levels of the water. So if we are talking about water or soil, we know that bacteria, different types, like to live in different places. So bacteria that really rely on oxygen, we would expect to be, you know, toward the surface of the water, and bacteria that did not necessarily really want that oxygen to be lower even. And same with soil, we would expect that same result, you know, because there are different conditions in the soil the further you dig down and everything. So yeah, that's also a very good point. I think it would be important to sample from different layers and to even maybe get other students in that focus on agriculture, maybe to like assist us, like, you know, maybe you should try this type of soil, maybe you should try this deep. And I think we could benefit from other students in the biology department as well. So.
Thank you guys, very wonderful presentation. Thank you for joining us.
Thank you.
Yes, thank you so much for having us. It was a pleasure.
Good. Well, we are going to close this session for this evening. And I would like to remind everyone a couple things. First off, tonight at 6:30, we have a guest presentation by Brian Gula and John Laskos, who are Pennsylvania DCNR Environmental Education Specialist. They will be presenting from 6:30 to 7:30 about resource management at Presque Isle State Park. So please tune in back on reachscitv.com to see their presentation at 6:30. Another reminder, we will be beginning early tomorrow. Instead of starting at 10, we will be starting at 9 a.m. So please join us at 9 a.m. for the welcome, and we will have 2 sessions. And then the sessions will be followed by the student award ceremony at 1 o'clock tomorrow afternoon. So we will see you back here at 6:30 for our guest presentation, and we'll see you again tomorrow.